There was something.
I think it's now
Back
moving.
working. Yeah, let me turn that on this. Awesome. We'll get there. It's long. Yeah, okay.
Perfect. Okay. So over 12 years ago, much less painful than yesterday, at least.
2 minutes, not 20 minutes.
All right. So over 20 years ago, the first draft of a human genome came out, and a little snippet of it looks something like this. In the words of Dr. Eric Lander, genome, bought the book, hard to read. Despite being challenging for us mere mortals to understand by looking at, these long sequences of A's, C's, G's, and T's provided a crucial roadmap for the next generation of high-throughput biochemical assays. Here are an example of 4 such assays and an enhancer. Just by looking at their readouts, we can see that at this enhancer, there's probably the binding of OCT4, SOX2, and NANOG, but not necessarily the binding of KLF4. Clearly, these sorts of readouts are invaluable for us to understand what's going on in the genome. However, relying solely on these readouts is kind of reminiscent of the parable of the blind men trying to describe an elephant based on what they felt. Just because we understand what is happening and where in the genome doesn't mean we understand why it is happening there. And so enter sequence-based machine learning models. There are many different internal computational units that you can use, convolutions, transformers, whatever, and there are many different precise formulations of the exact task that the model is trying to solve. But they all have a commonality in that these are models that take in DNA sequence, like on the left, and they make predictions for these sorts of readouts of interest. But in my view, the predictions from these models are kind of the least interesting aspects of them. After all, most of these assays are already genome-wide. There is no more secret genome that we need to make predictions for. Rather, the strength of these sorts of machine and deep learning models is that we can run them backwards. We can get them to explain why they made the predictions that they did. And we can see here that the reason that this model was predicting the binding of OCT4 is because this sequence had an OCT-SOX heterodimeric binding site there. And it's just able to highlight that and say that that's what's driving model predictions. These same ideas can also be used in the design space. In a similar setting, imagine that you're midway through this design space, you have some sequence that you're messing with, you have a machine learning model that's making predictions of interest, and you have a desired output from that model. Maybe you're trying to design an open chromatin region, maybe you're trying to design a promoter or whatever. You take your sequence, you run it through the model like before, you get some prediction, and rather than running that output through the model in order to highlight the nucleotides that are driving model predictions, you now take the difference between the actual output and the desired output, and you run this difference backwards through the model. In this manner, you're not highlighting the nucleotides driving model predictions, you're highlighting the nucleotides that need to change in order to get your desired objective. And so these ideas of understanding and design go hand in hand when we have these sorts of machine learning models. And I'm going to share 2 research vignettes that I hope highlight that. The first is going to be some older work of mine where I introduce this model called Dragon Fruit that allows us to understand the drivers of chromatin accessibility at single cell resolution. And then I'm going to tell you about some more recent work that came out this year about a method called Ladidi that uses these same deep learning methods in order to allow you to design DNA with very specific characteristics. So let's jump right into the first one. Over the past decade, improvements in single cell technology have really increased the scope, the number of cells, the number of reads per cell, just the general quality of single cell data. So here is going to be an experiment. And so these single cell experiments have now really allowed us to look not just at discrete, groups of cell types that might be in a tissue, but even at entire continuous processes. And so here I'm going to show you an example of a fibroblast to iPSC reprogramming trajectory. This was data that was originally collected by Mo and was initially analyzed by Chirag, who has now moved on to greater things at Genentech. On the left-hand side here, we're going to have a cartoon schematic describing what's going on in the experiment. In the middle, we have everyone's favorite UMAP made up of the single-cell ATAC-seq data. And on the right-hand side, we have RNA-seq data from paired experiments. So this isn't multi-ohm data. This is just from paired specific time points along the trajectory. At the beginning, we have just pure dermal fibroblasts just hanging out being fibroblasts. Then they're infected with 3 Sendai virus vectors containing the 4 Yamanaka factors. After just 2 days, we can see that quite a bit has happened, including the emergence of a population of cells on the left there that exhibit very high concentrations of these Yamanaka factors. After 2 more days, it seems like that population has kind of slid into a population that simply has high relative to endogenous expression of these factors. And then we can see that more continues to happen over time. At around day 12, we switch out the media from fibroblast growth media to iPSC growth media, causing this big jump. And we start to see the emergence of pre-iPSCs, cells which aren't quite iPSCs, but exhibit many of their characteristics. After 2 more days, that flashes out. And then we leave the cells for 2 weeks. And we get a population of pure iPSCs over here. Just by looking at the dynamics here, not to infer too much from it, but we can get a sense for some of the trends that are going on in the data. The first is that we have a no-factor uptake trajectory. These are cells that never really encountered the virus and just lived as fibroblasts for the entire time. The second is the primary reprogramming trajectory. These are cells that began as fibroblasts, got reprogrammed, and ended up as iPSCs. But if we look a little bit more deeply, we can see that what initially happened is that these were cells that were infected by the virus that then spewed the 4 Yamanaka factors, severely changing the trans-regulatory environment. Then those factors went and they bound to the sites in the genome, according to motif presence, spacing, affinity, and other properties of syntax, cis-regulation. So I kind of think of it kind of like a partial differential equation that was at equilibrium initially, and then you poked it, and it had to oscillate quite a bit before it reached the new equilibrium, iPSCs. But despite that continuous nature, that's not really how most people actually model this sort of data. That's not how we modeled it initially when we did it. First, what we did is we clustered this into 15 different clusters. These weren't necessarily meant to represent different cell types, but just kind of different landmarks along this continuous process. We then aggregated all the cells by cluster into pseudobulks. We organized these pseudobulks according to some notion of pseudotime, and then we were able to inspect loci to see dynamic activity with respect to that pseudotime. And we can see here the nanog locus opening up as the cells get reprogrammed, which makes sense because nanog is one of the primary drivers of the reprogramming process. So I'll let this animation finish one more time. So if we wanted to really model this at single cell resolution, we have to go back to the data and think about how we would do that. Really, the underlying data is a super sparse matrix that looks like this, where each one of the cells is a different row, each one of the columns is a different locus or position in the genome, etc. And the values within it are the amount of chromatin accessibility, number of reads or processed reads, etc. If we want to build a model, first we're going to need a representation of our locus. But just like all other sequence-based machine learning models, we can just use DNA sequence. Then we need some sort of representation of our cell. This will be a vector representation that I'll get to later, but it can, for now, I'll just say that it can be quite flexible. Then we need a target to predict. That's going to be the chromatin accessibility at base pair resolution. If you've dealt with single cell ATAC-seq data before, though, you know that the profile doesn't look quite this dense. It looks very sparse. And so what we do is that we dynamically pseudobulk. We basically sum reads across similar cells in order to share statistical power. Then for our modeling part, the exact details of the layers that are being used don't necessarily matter, but if you're familiar with the BPNet model, we basically borrow that architecture. So we have layers of convolutions and dilated convolutions, but that doesn't incorporate the cell representation. In order to do that, we need to introduce a new component, the controller network. And the way this works is that we take our vector representation, pass a few layers of neural network magic that extract the meaningful features from this representation. And this is the key point to how Dragonfruit works, and it's why Dragonfruit is different from other methods that are out there. We are then predicting the parameters of these internal convolutions. We're not learning a representation that is also passed into a second component. We are actually predicting the parameters of these convolutions that are about to be applied to the sequence. And so the learning task quite literally becomes, we are learning how to use the cell representation in order to process sequence in a cell-specific manner. So you might ask how this works. Well, we start off and we have a cell representation that for the cell we're trying to make predictions for. We run it through this network and we get predicted sets of parameters for the convolutions. Now that we have predicted parameters for the convolutions, we have a full model. So we can just take our sequence and we can run it through and get predictions for what the profile of the dynamic pseudobulk looks like for that cell at that position. We can then calculate our loss, run it back through the model. But when we encounter a predicted parameter, we're not updating that directly. Rather, we're going down into the controller network and updating the parameters there. We are learning how to process the cell representation in a manner that, when applied to the sequence, gives us correct predictions. And so we're kind of learning a conditional distribution here instead of a joint distribution. This has two important points to keep in mind. The first is that once the dragon fruit model is trained, any analysis that you're currently doing at bulk resolution can now be done at single cell resolution. If you want to calculate variant effect, if you want to calculate feature attributions, if you want to look at marginalizations, anything you're doing at bulk resolution, you can now do at single cell resolution, because conceptually you just do it for each model, one model per each cell in there. It takes a little bit of time, so you may not want to use every single cell, but in principle you can. The second, and I said I'd get back to this, is that the cell representation can quite literally be anything. It can be derived from your data. If you have multi-ohm data, you could use gene expression values there and still make predictions for chromatin accessibility. If you have concentrations of various surface markers, et cetera, if you have spatial coordinates, whatever you have. If you like some deep learning model that learns, that learns embeddings from your single cell data, you can use that. It can be anything that you think faithfully represents your cells. So, does this work? Let me demonstrate it on a region whose significance will become apparent in a second. On the left-hand side, we have reads from our experiment. This is for one specific locus. So each row is a different cell in our experiment grouped by cell type. And each column there, it's kind of hard to see, is a different base pair in our 1 kV window. So you can see that there are a few reads. There aren't no reads, but there aren't a lot of reads either. And when we pseudobulk, not much is happening. When we applied dragon fruit there, we see that dragon fruit doesn't really predict that much is happening either, and likewise, the pseudobulk is pretty flat. Right after infection, we see that a lot has happened. Not only is there much more accessibility overall, but that it's structured in a way to form peaks, that we're seeing peaks at specific nucleotides there. And when we run dragon fruit, we see that not only are we also getting much more chromatin accessibility, but those specific base pairs are now predicted to be higher as before. The really interesting thing about this locus, though, is what happens next. We can see that chromatin accessibility fades. This is what's called a transient enhancer. This is a region that is not accessible in fibroblasts, and it's not accessible at iPSCs. If you only looked at the endpoints of this process, you would miss it, but it becomes temporarily accessible during the reprogramming process. And so we think that it may be very important for driving reprogramming forward. But like I said before, the predictions from these models aren't the main purpose for using them. After all, the experimental data showed us what I just told you. The real strength is being able to use these models to explain why that is the case, why this is a transient enhancer. And basically, immediately, we can pull out an explanation. If we look at feature attributions for the pseudobulks, we can see that there are quite a number of motifs that are being identified by the model as driving predictions here. On the left, we have KLF, and when we can not only look at this at pseudobulk resolution, but we can sum these attributions and display for every cell in our experiment how strong that motif is driving model predictions. So we can see a KLF, which makes sense. KLF is one of the 4 Yamanaka factors, and so shortly after infection, it kind of comes and opens this region. The most interesting thing here is actually this OX-SOX site. It is a very weak OX-SOX site. It is so weak that under normal concentrations of OCT and SOX, they aren't really going to bind there. Rather, what you need is ectopically high levels of OCT and SOX, like say a virus that's just spewing this factor everywhere in order to get any type of binding there. Then there's a stronger KLF site here, and there's an NFI motif. NFI is part of the fibroblast program, and so it's the reason that there was any accessibility in these sites initially. But you can see that we can derive these sorts of explanations for what's going on, not only immediately for this locus, but basically for any locus that we care about. We can just apply this genome-wide and get explanations for every single accessibility site across the sort of continuous process like this and connect it to the underlying biology. What this allowed us to do is kind of look at some things that may have been unexpected. And so I'm going to share a few things that we found that I thought were really interesting. The first is that there were some peaks that seemed to move along the genome, which isn't necessarily something that I thought was possible. This is a site that's normally inaccessible in fibroblasts. Shortly after infection, it becomes very accessible. Look at the grid lines to kind of see what's happening there. You can see that after it moving a tiny bit, in the next, it moving a little bit more, moving a little bit more. And then when we get to iPSCs, we can see that it really has moved around 200 base pairs from where it originally was. How do accessible sites move along the genome? They don't have wheels. Well, when we use attribution methods, we get an explanation for this immediately. Initially, there are two KLF binding sites that are blowing the region open. But as the concentration of KLF diminishes in the cell, the right-hand flank starts to collapse. At the same time, as TED, and ZIC proteins start to be expressed by the reprogrammed cells, we see that they start to raise the accessibility on the left-hand side. And so the collapsing flank on the right-hand side with the increased accessibility on the left-hand side gives the illusion that this is a site where the accessibility is moving. But with these attributions, we can see that's not really moving. It's just that different motifs are being utilized. Sometimes when people analyze single cell data, they focus on variable regions. And I think that this is a mistake. Maybe sites that are never accessible, you can just toss out because they're not that exciting, but people also toss out sites that are always accessible because they think it's something boring like CTCF that's really driving activity. Here's an example of why. Here we have both the experimental data and the predictions show that this is a site that is just uniformly accessible, but the reason why changes. Initially, this is a site that is accessible because of an AP1 motif. AP1 proteins are highly expressed in fibroblasts, but that starts to diminish over time. And as OCT and SOX start to get expressed, the OCT and SOX motifs take over. And so even though the accessibility of this site stays the same, the reason why changes. And when you exclude these types of regions from your analysis, you are systematically removing regions that evolution says have to be active across the entire process. They intentionally have put motifs from multiple cell lines in the same spot to make sure that the region is accessible across the entire process. So we can even then just factorize this region based on the attributions in the tier. We can see that there's the regime where accessibility is being driven by AP1 motifs, and there's the regime where it's being driven by the OCT-SOX motifs. That's kind of the last example. We wanted to return to the nanog locus, which was what I was showing in that GIF at the beginning. We see that there are a few of our favorite players here. We have OCT-SOX, KLF, and etc. There's a ZEB motif. ZEB is a repressor of chromatin accessibility. And so we can see that what's happening is that it's desperately trying to keep the region closed as the high concentration of KLF comes in and kind of bursts it open. So we see some negative attributions from the ZEB. But across models, we notice there's a little bit of fuzz here. And because, deep learning models are perfect, we decided we wanted to look into it a little bit more. And what we found is that what's actually happening here is that this is an array of weak KLF sites, and it is overlapped with an array of weak ZIC sites. ZIC is a protein that gets expressed in iPSCs, and so as the KLF gets diluted out of the cell, it goes away, and the same nucleotides that were previously being bound by KLF are now being bound by ZIC. And so I'd really encourage you to not think about the genome as basically places where nothing is happening except for a box, which is a distinct binding site. Rather, you should think about the genome as filled with these sites that are potentially bindable by so many different proteins, especially when you start looking at the weak affinity regime. So one of the things that I'm not going to get into a little here for time reasons is that we basically were able to build a local pseudotime clock based off this, based on whether or not the KLF nucleotides were being used or the ZIC-only nucleotides were being used. We created a local version of pseudotime, and this is work that is being continued by Danila Bredkin as a postdoc in Onshaw's lab. All right, so I shared a brief vignette about using dragonfruit in order to understand genomic activity. And now I'm going to talk a little bit about using the same machine learning models for design. So at a high level, imagine that you're midway through the design process. You have some sequence that is being designed with specific characteristics. You have your machine learning model that you are using to guide the design, and you have your desired output. You are running your sequence through the model. You are comparing the predictions from the model to the desired predictions from the model. and you're about to run that backwards. But you now encounter a problem. The gradients that we're running backwards through the model are continuous. We saw when we looked at the attributions that the characters had different heights, and the higher the character, the more important that was. But now we're looking at a discrete choice, whether or not this would be an edit or not. How do we deal with the gradients that we're passing through the model being continuous and the underlying DNA sequence being discrete? This is where we introduce a method called liddy. The way that liddy works is that it turns this discrete optimization task into a continuous optimization task that can be solved using off-the-shelf optimizers. And so in the end, it only ends up being a few lines of code once you use this little trick. And the little trick is basically that you learn an underlying weight matrix that you use in order to sample an edited sequence from. Let me walk you through that. So in this first position, we see that we have a G. At this point in the design process, the DD doesn't really care. It has low values for any of the possible nucleotides. And so from the addition of the initial sequence and this underlying weight matrix, we end up sampling a G because, the initial sequence is a G. At the next position, we see that the initial sequence was an A. The DD really thinks that it should be a T. And so from the addition of these, we end up sampling a T, an edit. We can then go all the way down this process at each position, either sampling the original sequence or sampling edits. But in the end, we end up with a discrete sequence that can then be run through a machine learning model. We can then compare the predictions to the desired output, as I've been saying over and over, and we can run the gradient backwards through the model. But when we encounter the edited sequence, we can go straight through it using the Gumbo softmax reparameterization trick, and we can update this underlying weight matrix. So that's fairly straightforward, that we are only trying to optimize this underlying weight matrix, but the loss that we are trying to optimize is a little bit more complicated. It's made up of two terms. The first term here is the output loss, which is kind of the classic thing that you talk about when you do design. You're trying to make the machine learning model predict the thing that you want it to predict. And so the loss is going to be how far away you are from the thing you're trying to predict. And the second is this novel input loss. We are trying to do so using as few edits from an initial template sequence as possible. And so we want to do this making as few changes to this sequence as possible. And so a difference between liddy and other sorts of design methods, that many design methods focus on de novo design, where they want to, from scratch, create something. Here we are trying to edit an initial template sequence using as few edits as possible. Let's just skip that. So we then went and, as a proof of concept, we went and applied it to a model that makes predictions for the binding of GATA2, BPNet model. The initial sequences had predictions that are the black dotted line, so GATA wasn't really binding there, and we wanted to turn it into a really strong, a site where GATA bound really strongly. And when we ran our procedure, here's where the sequences ended up. They're not quite on the cyan line, which is where we wanted them to be, because remember that we're optimizing a mixture of two terms. We could have gotten closer to the cyan line, but that would have required too many edits. And so we're fine being simply near the cyan line here. What was really interesting to me is when we look at the actual edits that liddy made. If I told you that, in order to get GATA binding, it made 5 edits, you might not be particularly impressed because you know that the motif that GATA binds to is GATAA, which is 5 characters. But if we look at the exact positioning of the edits here, we see something really interesting. Only one of the edits happens in that core GATAA motif. We're changing the orange ones are the edits, and the A that is there is the proposed one within the core sequence. We are then spending 3 edits, making the flanking regions higher affinity, and we're spending 1 edit, that G on the very right-hand side, adding in a tau binding site. Tau is a cofactor of GATA that really increases the strength of the binding. And so what liddy did here is that it found a region that was predisposed to GATA binding, and it focused on making its edits there because of this input loss. Because it could only make a few edits, it chose to target a region that was almost GATA, rather than just adding in a GATA site anywhere. And when we use a pruning algorithm subsequently, we can show that with only 2 edits, we're able to do this. This isn't just a one-off, that in the post that I have outside, I show how we can add in CTCF binding using only 3 edits, because it finds a region that is almost CTCF. I have a hypothesis that I'm not very well equipped to study, but my feeling is that the reason this is happening is because our genomes are graveyards of past viral infections and transposon integrations and that sort of things, where we had to mutate out the functional regions of those sites in order to live. And so our genomes are filled with almost binding sites. And with Ledidi, we can bring them back to life. So something I want to emphasize at this point is that Ledidi is a designed method. It is not a machine learning model. Sometimes when people publish papers saying they do design things, what they really mean is that they have published a machine learning model that make cool predictions, and then they apply off-the-shelf design methods to it. Ledidi is a design method, which means that it can be paired with any machine learning model, anyone that's been published already, anyone that you have in your lab, etc. And so to demonstrate that, we set up a design task, a standardized design task, where we wanted to take regions that were 50th percentile in terms of predicted activity and make them really strong. We want to make them 99.5th percentile. Percentiles because we're about to use a bunch of different models whose dynamic range are very different from each other. And so when we look at our friend BPNet making predictions for GATA2, we can see that Ledidi is able to very well design edits that achieve our 99.5th percentile task. We can then apply this out of the box with no changes to the default settings across different models, across different proteins, and we see that we get edits that are just as good. We can then look at 4 different models that make predictions for chromatin accessibility, and we're working just as well. We can look at models that make predictions for transcription and transcription initiation, just out of the box. We're doing quite well. We can look at MPRA activity from this Malinois model. We can look at enhancer activity according to the deep store model. Across all of these models, Ledidi just works out of the box. So one of the strengths of Ledidi is that it is very fast and that we are able to leverage this speed in order to simultaneously use multiple models for design. We can use models to make predictions for vastly different sorts of things in fairly straightforward manner. Basically, you have your sequence, you sample your edited sequence from it, you pass that edited sequence through each one of the models, you compare the predictions from those models to the desired predictions from any one of those models, you calculate the gradient with all of those, and you combine those into a single gradient update that is then applied to the weight matrix. There are some interesting technical details here, but these don't even have to have the same receptive field. So you can take very wide models and very small models and combine them. It's not the case that you're limited by having everything have to be the same. It is quite flexible. And so the question becomes, which multiple models should we use for design? At the time that I was doing this work, I was a visiting scientist at the Research Institute of Molecular Pathology, where Alex Stark, who invented StarSeq, is a scientist. And so I was contractually obligated to use his deep store models in order to do design. And so we ended up trying to design cell type specific enhancers using these deep store models that he had already trained for other projects. To be clear, these aren't models we trained for this. These were models he just had laying around. And so specifically, we wanted to try to design enhancers that were specific to S2 cells or specific to OSCs using these 2 models. But remember that one of the interesting things about Ledidi is that we use an initial template sequence in order to make our edits. And so there's choice as to what we should use for our initial sequences. So we started off with just regions that were not enhancers in either cell line. This is kind of a classic task, just take regions that are non-functional, turn them into enhancers in one or the other cell line. And so when we turn those regions into S2 specific, when we turn them into S2 specific enhancers, to be clear, this is real StarSeq data. These aren't predictions here. We can see that we get a range of activity in S2 cells while limiting the activity in OSCs. When we take those same regions, and we try to turn them into OSC specific enhancers, we see the transpose of that. When we try to make them active in both cell lines, we see that we're now following along the diagonal. You may be looking at this and be like, Jacob, that's not particularly impressive. What's going on in that bottom left there? Seems like there's quite a bit of activity there. An aspect of this experiment that I withheld from now is that we didn't just try to qualitatively design cell type specific enhancers, we also tried to quantitatively design enhancers of different strengths. And so when we bin these according to the desired strength, we see that we were able to achieve that goal with target strengths yielding enhancers of increased activity. When we look at the activity in OSCs, we're able to limit the activity in the off-target cell line quite well. across all of these. When we look at the OSC specific enhancers, we see the reverse, where we keep the activity in S2 low across all of these categories, while allowing the OSC activity to get quite high. And then potentially the cleanest results happen when we want them to be active in both cell lines. So returning to this idea of being able to choose the initial regions, we then expanded our search. We chose regions that were initially peaks in S2 cells, originally enhancers in OSC, peak enhancers, the same thing here. Originally enhancers in OSCs, enhancers in both, and then Alex required that because he was paying for the experiment, he wanted me to run random sequence through just to see what would happen. So those are the 5 initial pools of sequences that we used. The most interesting to me were the ones where we started with an enhancer in one cell line and were able to edit it to become an enhancer in a second. And so I'm going to show you a single example of one of these regions where we were able to take an enhancer that was initially in OSC and turn it into, I guess that's actually the opposite. Highlight the wrong arrow here, but it was initially an enhancer in S2 cells, and we turned it into an enhancer in OSCs. Because that involves 2 operations. The first is that we have to turn off the activity in the initial cell line. And the second is then add in the activity in the new cell line. That's more than what we were doing before, which is just adding in activity in one cell line. So we can see that the reason, so this is attributions from a model, but this is also backed up by experimental data that I'm not showing here. We see that the reason that the model was originally predicting S2 specific activity is because there are these 2 GATA sites here. GATA is an S2 specific motif, and so it makes sense that that's what's driving S2 specific activity. When we try to edit this to become a weak OSC enhancer, we see something really interesting. We make 10 total edits, of which 6 are shown here. There's 1 edit being made to knock out the GATA motif on the left-hand side, 2 edits being made to knock out the GATA motif on the right-hand side, and then 2 edits doing something, which we'll see in a second, in the middle there. When we want to take this same region and turn it into an OSC enhancer with middling strength, we see that we're making the same set of edits on the left and the right-hand side to knock out the GATA motifs, but now we're making a single edit to add in a traffic jam motif in the middle, which is OSC specific. And finally, we can see that when we want to make this a really strong OSC specific enhancer, that we're adding in quite a few more edits here, but that we're adding in a second traffic jam motif right next to it, which is pretty common in OSC cells, that if you want to have OSC specific enhancers, you add these 2 traffic jam motifs right next to each other. But what's interesting to me here is we're adding in the second traffic jam motif kind of on the dead body of the GATA motif that used to be inducing S2 specific activity. Something I found really interesting about the first and the second edit here is that we needed 2 edits to get to weak activity, but we only needed 1 edit to get to middle kind of strong activity there. And that flipped a few things in my head because it meant that we needed 1 edit to add in the traffic jam motif and a second edit to make that motif weak. Things aren't necessarily linear. We need fewer motifs to have weak activity and more edits in order to achieve constantly stronger activity. Sometimes there are these non-linear things depending on the sequence that you start off with. And so this choice of an initial sequence is quite important. So I only have a few more minutes, so I thought I'd just at a high level conceptually talk about some of the other things that we ended up doing. We ended up designing accessible sites, but we controlled the reason why they were accessible. So we had a Chrome BPNet model that predicts accessibility, and we said, I always want you to predict high accessibility, but then we had a battery of BPNet models, and we said, I want you to predict a high value and everyone else to predict a low value. And we were able to design sites that had uniform accessibility, but were predicted to be controlled by different transcription factors. Then we flipped this idea on its head. So far, we've been using different models that make predictions for different sorts of things. What happens when you have multiple models that make predictions for the same thing? This happens quite frequently when you enter a field for the new time. You see, for the first time, you see that there are many models. Each person kind of uses their own model, but many high-performing models exist for the field, and you don't know which one to use. Well, why don't you use all of them? You can have all of them trying to work simultaneously to achieve the same objective, and that way your designs don't overfit to the parameters of any specific model. So I shared with you 2 vignettes that I did about research that I've been doing over the past few years. But at a high level, I'm hoping that this talk isn't really so much about those specific research problems, but about the high level idea that about using machine learning for understanding and design in genomics. The real point here is that deep learning models are not just uninterpretable black boxes that we should try to avoid when we can, but really they are indispensable for our understanding of genomics as well as future research as a community in this field. And so if you agree with me, if you're interested in the sort of work, as David alluded to, I am currently recruiting postdocs and software engineers. And so please come and find me or reach out afterwards with your CV if you'd like to chat more. Thank you for your time, and thanks to all of my collaborators across the many years.
Yeah, and we definitely have time for questions. This microphone runs.
Very cool talk, thank you. Two questions. One is this ZK-KLF relationship is really interesting. I was wondering how easy it is to destroy like some other pairs. If I find some like overlapping binding site of different pairs and maybe look at the expression to see if the expression is anticorrelated or something, would it be sufficient to discover other pairs? It's a really cool thing.
Yeah, so there are two ways you can view this. The first is that solely from genome sequence, can you identify sites that have different, factors on top of each other? And you can actually discover this from PWMs themselves without even needing genome sequence. And so I had a short-lived project that got subsumed by my faculty search about trying to automatically identify these. In terms of incorporating RNA-seq, I think that's a really interesting idea, but I haven't thought too deeply about it.
And the second one is, can you maybe highlight the differences between like or de novo design? I'm a big fan of like learning by design idea. Design methods will teach us maybe more than, anyway, they will teach us more. Can you highlight the differences between like the de novo methods and the things that we can learn from them, and maybe the things that we can only learn from editing methods?
Yeah, so yesterday Johannes Linder gave a good talk about an alternate version of simultaneous work that arose about doing de novo discovery using gradients similar to Liddy, but in the de novo setting as opposed to the editing of it. And that's a method called fast-seq-prop. The idea there is that if you have a model and all you care about is designing something that achieves the target prediction, like maybe you're a pharmaceutical company, Genentech, and all you really care about is designing a drug that works, and you don't care so much about the underlying biology, that's where de novo design really shines. But my experience has been sometimes this doesn't work so well in practice because the machine learning models aren't perfect and they don't capture everything that's going on, say, in the genome. And so what I've found is that using a minimal set of edits in an initial template kind of provides an important regularization. That if you start at a genome sequence that's almost there and you just poke it in the right direction, that seems like a much stronger way of validating something than just completely designing something from scratch.
Hey Jacob, the machine learning models are not perfect, but the talk is pretty much perfect. I have a question about so you were trying, in the DD, you were trying to design DNA sequences and combining the original sequence with the DD weight matrix.
Yeah.
And how did you get can you describe what's the process to get the DD weight matrix?
Yeah, so this is a somewhat technical question, but the idea here, I'm going to just use this slide. The idea here is that this underlying weight matrix here can be interpreted as logic over the DNA alphabet. And so what you're doing is that you are basically you're learning how to alter the initial sequence in order to sample characters. And so because this can be interpreted as logic, it can be directly updated by the gradients from these machine learning models. That you can just directly learn that weight matrix. And when the weight matrix is near 0, it means keep the initial sequence. And when it's not near 0, it's saying either be this nucleotide or don't be this nucleotide.
Does it have anything to do with the attribution matrix?
Only in the sense that both methods are gradient based. It can be viewed similarly, but it's not, like I mentioned in the talk, when you run the difference between the true predictions and the desired predictions back, you're not highlighting the things that are driving model predictions. You're highlighting what needs to change in order to achieve the desired goal. So I wouldn't directly interpret the linear weight matrix as attributions.
wanted to ask a little bit about the cell representation. And so, I'll just paint a picture of what we would want to do at Genentech, right? So we want to think about T cells.
Yeah.
Right? In a particular case, but we wouldn't want to just think about T cells. We want to be thinking about T cells that are adjacent, T helper cells that are adjacent to places where there's T cell exhaustion, things that are interfering. And we further want to look at the patients that didn't respond and figure out what are the additional, changes to this sort of cell product that can avoid that. So we really want to look at it very much, or a very cell cross environment kind of thing. And so what are your thoughts for sort of future work to include strange environments, or, what might be for the data set kind of an edge environment for that cell? Or how much do you think you can push that cell representation to include stuff like that?
Yeah, so I mean, I think that when you say environment, do you mean spatial environment or just the setting that the experiment is done in?
But, factors, what happened to it, there might be typical, but the things that the cell's program is reacting to could also be other cells.
Yeah.
Broadly speaking.
So I think the gist is that algorithmically, this cell representation can be incredibly flexible. If you have gene expression values from a multi-omic experiment, but you also have some sort of like categorical representations from each cell that are derived somehow, you can absolutely put that in the cell representation, and this sort of thing will work. The challenge is going to be making sure that the data is high quality. One of the things that I've experienced with multi-omic data and trying to apply this to that is that the ataxic component of multi-omic data is usually quite poor. And so dragon fruit has worked much better when you just apply it directly to only single cell ataxic data, which is going to be more challenging for the setting that you describe.
Questions in the back?
Great talk. I really like the use of cell type representation and doing this hypernetwork style, because this led to interplay between solid states. So I was wondering if you tested cell type generalization, like during testing, or like during training, like you held out certain cell types, ideally even from embedding creation, and then so if the model does good on those cell types.
Yeah, this is something which conceptually I think is interesting, potentially even being able to forecast what's going to be happening next. It's not something that I have looked into because I have found that the best or the most robust uses of machine learning have been trying to explain experimental readouts as opposed to trying to generalize to new experiments or like future time points. And so it hasn't been as interesting to me personally to hold out a bunch of cells and see if you can make accurate predictions there, because my goal is to explain in each cell why you're observing that readout. But it's certainly an interesting area of future research.
I love this hypothesis you have about there being all sorts of like near binding sites all over the genome. And people can have questions about genome evolvability, would like love to know, like, are there regions that are like 1 or 2 edits away from being transcription start site, or even asking questions about like endogenous retroviruses? Are they somehow close to enhancers for specific cell types? Do you have any plans for generating that type of data genome-wide?
I don't have any immediate plans for generating that type of data. I'm entirely a computational person, and so I have to go around begging people to.
No, I mean with your methods with Ladidi, just run Ladidi genome-wide and just like ask how many edits away you are from like a useful enhancer or a transcription start.
That's a great question. One of the areas that I'm going to be moving into is this idea that I'm calling discovery by design, kind of as an alternate way of using machine learning models to attributions. Like attributions can explain if it's a peak, why is it a peak? But I think that design methods can be used for discovery in exactly the way that you talk about. You say like, I want to take this region and I want to make it more accessible. What would I need to change? You can get rid of repressors or something. You can look at retroviral integrations like you talk about. I think it's a big open area. And it's definitely something that I'm looking into. Maybe not that specific question, but the idea of discovery by design.
Cool, thanks.
Maybe the last one here in the front. Alex, one last one in the front.
Really great talk. So I want to ask, so these attribution methods, they basically, they allow you to look into the deep learning model and ask why you think those enhancers are being turned on, but that is based on the model's kind of oracle predictions, which has some error, some uncertainty. There's probably some spurious things in there. Have you thought of or been part of efforts to then take those attributions and maybe design something like a combinatorial screen to see, like, in an experiment, do those attributions replicate, and then you could possibly take the results of that screen and kind of use it for fine tuning it better. Yeah, like have that or have efforts like that move forward at all.
Yeah, there have absolutely been efforts of trying to use these attributions and see where they are successful and where they fail. I think that Onshore has quite a few papers at this point about using that sort of thing in various settings, not just in the, I think mostly this focuses on things like variant effect prediction. One way you could do variant effect prediction is by just putting in the variant and seeing what the change in model predictions are. The other thing that you could do is you could look at the attributions and see whether or not it's highlighting a, an activator or a oppressor and that sort of thing. And so I have not personally been involved in those sorts of efforts, but I know that many of them have happened.
Thank you. All right. Great, let's thank Jacob again. Noah, what is it? Are you on the Zoom already? Okay. Did you get an invite? You should, hopefully you got an email. It should let you in. The one tricky thing is it needs to be the same email address that you log into Zoom with. The NYGC guest should just let you on also. Sorry.
It's a little bit more spotty with Ubuntu.
Yeah. Yeah, if you
check them out.
sure. You can also upload the slides to the Dropbox that I send if that's easier. Or tethering, yeah.
Yeah, here what I'll do is I'll just turn
My postdoc also has this many tabs open. So
wow, this is only one window.
yeah, I'm sure.
Okay, let's see.
What am I looking for?
It'll be from Zoom. But it will be in whichever email. Yeah. Because that's the whatever email I had associated with data. Do you have the slides also?
We're co-presenting.
Right. Okay, so yeah.
Let's see if I can clear this. There we go. There we go. Okay.
Good. Yes. Yeah, so okay, maybe you didn't. Can you let's go to that. So if we go to whichever it was, Zoom accounts. Okay, so we would have gone to that email address.
Yes.
But that didn't go. You don't have anything from Zoom. It might not say MLCB. It might just actually I would have thought it would say MLCB.
Join webinar.
Okay, perfect. I think you do need to click that way, right? Oh, but you don't have Zoom on here. Oh.
You want to send me the link? I have Zoom.
Oh yeah.
Okay. Sorry.
The easiest honestly might be, well, you can try and do, yeah, I mean, you can try, yeah, since you're doing this, you can try and do this. I think it might be easier to put the slides onto mine also.
Okay, what?
So that's the email that you found that was from me. Oh, I think it's better to do it by Dropbox. Oh, actually, yeah, I think it's better to do it by Dropbox. So the email that you just found from me. So if you search for MLCP again. Yeah, so if you go to that Dropbox link, I think this will give you a way to upload. Yeah, so if you can extract the files into there.
It's a Google Slides presentation.
Oh,
It might mess up the format.
Yeah,
Right?
it might,
Maybe we
but-
can just send you the link.
Yeah.
Just not
Yeah, yeah.
share it.
Yeah, I'll
Yeah.
Well, this is, I should send you the Zoom link, Adam. Or just send me the Zoom link, either one.
That doesn't work because you're not, well, we can, but I have to add you as a panelist.
Okay, so send it in the presentation so you get some. Just make sure you can view it, yeah. Anyone, just, yeah. Yeah. Okay. What is the design? I'll just that. Okay. Just.
And then I think set showing first, I think.
We can just do the full slide.
Yeah, no, yeah, that's the plan. Like, so it's yours. I don't know Google Slides well enough to know, but you can
Yeah, I
make
can.
it go for 2 minutes.
I can do that.
I understand.
Okay.
You can hide this if you. All right.
Okay. Thank you. Perfect. Okay.
Close your laptop.
Yeah. Do we need to hide this? How do I hide this? Okay. Perfect. All right, cool.
Okay, I think we're ready to go.
Cool. So good morning, everyone. Very excited to be here. Thanks to all of the organizers. So I'm Noah. This is my co-author Adam, and today we're excited to present to you Structure Flow, a simulation-free structure learning approach for stochastic dynamics. Okay. So on the left, we see this graphical system, and we consider this a gene regulatory network, and that dictates these observed measurements, which are these noisy snapshot data taken independently at different times. And now it's great if we can model the dynamic evolution of these cells over time, but it's even better if we can learn to interpret and find the underlying dynamics, to decipher these underlying data generative processes. And so now, if we can do this together, we get closer to being able to interpret our predictions and reason about our models in an interpretable way, which allows us to reason about cell fate, predict responses to disease, predict perturbational responses, and facilitate things like experimental design. And now that I've kind of laid the tracks for what we want to talk about, it's good to delve into the two broad classes that we have. The first one is trajectory inference, which shows us how to model populations and how they evolve over time. The second is structure learning for dynamics, which poses the problem as a network reconstruction problem, where it abstractly describes these systems as sets of directed relationships. And we want to be able to do this jointly. So as I alluded to earlier, this joint task would give us more interpretable models, and this would allow us to generalize across conditions. For example, we could generalize to unseen perturbations, where we can use the structure as an inductive bias for out of distribution conditions. And we can also facilitate these downstream applications like experimental design, where we can learn to predict perturbations that maximally reduce edge flow or flow uncertainty. And very few methods have done this. There are some existing methods that address these individual tasks, but the ones that do this joint learning, like reference fitting, make some strong assumptions. And how can we address both these problems jointly? Well, again, our problem setup is that we have these noisy, unpaired time series data from single-cell RNA-seq. And we have multiple conditions. We have the observational conditions and we have these interventional conditions, which are the CRISPR gene knockouts. And for each time condition pair, we have an empirical marginal, PT, and this PT denotes the marginal distribution of our snapshots from different times, 0 to t, under specific conditions or knockouts, C. And there are a lot of challenges for addressing these specific problems and modeling these underlying dynamics and inferring the network structure of these systems. One of the first problems is that we have these partial observations or unpaired measurements. We can solve this through entropic optimal transport. The second problem is that these systems are intrinsically stochastic, and we can approximate this as a Schrodinger bridge. And the idea behind this is that the Schrodinger bridge gives us the most likely pairing between two processes, Q0 and Q1. And formally, this looks like the KL divergence between one process P and a reference process Q. So we kind of want to stay as close as possible to the reference process Q. And the third thing is these high dimensional distributions. And one of the ways we can solve this is through simulation-free score and flow matching, which was released in 2024 by Tong et al. And the idea behind this is that SF2M infers these dynamics from noisy snapshots by learning a population flow. And first, we kind of need to discuss the tools. One of these is this state SDE. So it models our cells or states under some stochastic dynamics. The problem is that we have these snapshot data that measure distributions instead of specific cells. And so now we need to use the Fokker-Planck equation. And this equation gives us a probability density function, and it shows us how it evolves under these stochastic dynamics. But conveniently, we can reformulate this into a probability flow ODE, where now we have this deterministic velocity that has the same marginals PT as the SDE. And Tong et al. Use this to approximate the probability flow ODE, and they find that there's a closed form expression from these Brownian bridges conditioned on endpoints x0 and x1 when we consider the reference process to be Brownian motion. And the second thing they do here is now they can define a neural field that approximates this closed form solution. And they do this in a simulation-free way. And what I mean by this is that they can sample batches of time, and then instead of simulating, they formulate this as a regression problem where they can regress over the vector field and this score log PT. And problem with this is that Tong et al. Doesn't exactly address this joint learning task, and it doesn't model interventions. So the first thing we do is we reparameterize so we can take an autonomous vector field. By that, I mean that our vector field is time invariant. And this drastically reduces the hypothesis space. So instead of just depending on time, now it's just state dependent. And we can model these structural dynamics with something called a neural graphical model. And you see in the middle this theta AJ. This is our adjacency matrix, and it's supposed to be sparse. So we use group lasso and this proximal gradient that gives us this soft thresholding. So now the results are some adjacency matrix where the genes I impact genes J only if the impact is significant. And the second thing we do is we try to model these interventions. So we mask our graph layer. We mask this adjacency matrix by M, where M says if we have some knocked out gene J, we zero out the entire row. Otherwise, we keep it. And the next thing we do is we also learn a conditional score. And this score is conditioned on the specific gene that we're knocking out. So we do this one-hot encoding where if the gene is knocked out, we give that information to the score, and otherwise we leave it alone. And how well does this work?
Okay, so I'm going to talk about some of the results of structured flow and how we evaluated it. So the first kind of experiment that we run on structured flow and its baselines is looking at how well it scales to higher dimensions. So to do this, what we do is we randomly generate some graph structures, like the matrix, like the graph structure on the screen, which we represent as a matrix. And then we simulate, we create simulated data with the SDE equation below So that's linear data, as it's just the matrix times the time point. And it's stochastic data, so it's noisy, which is what structure flow learns. We do some additional stuff. We ablate over 3 levels of graph sparsity. So the graph would be 0% sparse if every single node in the graph was connected to every other node. We measure the performance of structure learning task on the recovered AROC. So each method recovers a matrix A, which is the graph structure, and then we compare that using AROC to the true graph structure to see how well it recovered the underlying dynamics. We have 2 baselines, reference fitting and NGM node, which I'll explain in a second. So the first thing I want to highlight is that structure flow scales better to larger systems. So here what we have is as the size of the system increases, so in cells, that would be the number of genes, the dimensionality. Even as we get to 500 dimensions, structure flow generally does better than the NGM node and reference fitting baselines. As you get to about 500 dimensions, all the methods kind of collapse to essentially random, which random would be an AROC of 0.5. Structure flow has a particularly good performance on a 5% sparsity setting. So where only 1 in 20 nodes are connected. Or there's 1 in 20 possible edges. And then structure flow can also be trained pretty efficiently. We highlight this graph because NGM node is a not simulation-free approach, which means that to get a loss during training, you need to simulate with the model forward as an ODE, and then get a result, get a sample, and then calculate the loss from that. And then reference fitting, although substantially faster than structure flow, is a simpler method that it makes very strong assumptions about the linearity, which structure flow doesn't make. So we have a good balance of performance and efficiency. The second kind of experiment we do is having these smaller curated synthetic systems that each can kind of represent a common gene regulatory mechanic. So for example, we have a trifurcating system or a bifurcating system, a cyclical system or a swirl system. And we have these handcrafted systems, which are the static networks below. And then from that, we use a library called Boolean ODE, which generates simulated noisy data from underlying Boolean graphs. And we train the models on this simulated data and see how well it can recover the underlying graphs. So on the first task of structure learning, and on this task, we do the both joint tasks. So on the first side of the coin is structure learning. So here you can see the true graph for the trifurcating system on top, and then all the graphs of all the methods on the bottom. We're highlighting structure flow and reference fitting as the 2 top performing methods. You can see reference fitting and structure flow do a really good job of getting the underlying graph. A lot of the baselines don't, but they do better on other systems, some of the baselines. They're particularly bad here. And then in terms of numbers, like the AROC scores, we're highlighting the trifurcating system because there's so much data. It's just easier to focus on one. Both structure flow and reference fitting get really close to a 1.0 AROC score. And structure flow does really well on all the other systems as well. We also highlight which models cannot model knockouts, they have crosses along them. And we also want to highlight NGM SF2M, which you can see its performance. It's a little hard to see on the screen there, but it's the light blue and it's the rightmost bar for each system. And you can see the big difference between it and structure flow, really showing our methodological contributions. And then on the other side of the coin is the trajectory inference task, right? So here during training, we leave a time point out of the model, one of the time point data marginals. So it doesn't see the data at that time point, but it sees the data at all the other time points. And then we see if we simulate the data from the start, so t equals 0, and we get the model to simulate the data, how well does it reconstruct the data at the time point? Here we have t equals 3 as the time point, but when it's never actually seen that data. So in red, so the lighter gray is the start time point, time 0, kind of at the bottom, and then it goes in a circle. This is in PCA space. It goes in a circle, the data gets darker and darker over time, and then red is the t=3 time point. And then the blue crosses are structure flows predictions on the left, and then reference fitting on the right. And we do this for the observational data, so that's no knockouts, and then we highlight an example, one of the knockout conditions. And you can see here structure flow does pretty well compared to reference fitting. The blue crosses generally seem to get the distribution of the data a lot better than reference fitting does. And then in terms of numbers, again, we'll just highlight the trifurcating system, but you could look at all of them. Structure flow does better than reference fitting on the Wasserstein method. And I think it does better than reference fitting on essentially every single system for Wasserstein distance. And then it's usually pretty competitive on MMD. The SF2M model, we'll note, is essentially, it cannot recover the joint task. It cannot do GRN inference. It's just for trajectory inference. So it'll usually tend to do slightly better. It's a bit more expressive, but yeah, it can't do the joint task. So it's, yeah, we just use it as a baseline. The final experiment we do is we try and look for real data and evaluate the methods on that. So this data set is a multi-marginal data set. It has 4 time points. It's of induced pluripotent stem cells, and they use CRISPR interventions to create knockout conditions to make knockout data for 23 transcription factors. There's the data is a lot higher dimension than the synthetic small curated systems we were using. This is about 100 dimensions. It's from a study called RENG, and they put forth their own machine learning method for solving this. The downside is the ground truth network is a pretty small subset. I think it's 18 by 18. And it's really just an experimental estimate, right, from a biology experiment. So we don't know how accurate it is, but this is the data that we know of, and so we wanted to try it out. And you can see on the gene regulatory inference task, on the structure learning task, structure flow and RENG and reference fitting, they all do approximately the same. Structure flow has a huge variation among seeds. Sometimes it does very well, sometimes it does poorly. Ratio just means it's the number of times that the model beats random. And then structure flow also does do better than reference fitting on the trajectory inference task here as well. Yeah, so the real data is a little bit tricky. It's the one data set we know of, and we don't know how accurate the results are, but we wanted to try that out. So in conclusion, I think based on what we've shown, you could say it's still a pretty hard unsolved problem. I think we've shown that the joint using flow and score matching is a pretty good and efficient way of jointly inferring the network structure and the dynamics. It's all in a single model with a single end-to-end workflow, training workflow. But there's still a lot of things that we want to work on and move forward on. We don't know if the model is really learning true causal relationships. And we don't know, we don't have a really good way of systematically selecting good hyperparameters. Yeah, so thank you so much for coming to our talk. We want to acknowledge all our institutes that all our collaborators work at. And thank you so much. Any questions?
Yeah, no, really nice work. The thing I was going to ask about was that not? So was about sort of with these experiments, one of the limitations is because they're expensive. That we have these limitations on you don't get that many time points, or you don't get that many perturbations, or you don't get that many tissue contacts. So do you have a sense from the work you've done on this of like what would be the most useful things? Is it more time points that makes more of a difference? Like if you vary that in simulation, like obviously 1 time point you can't do anything, right? Mm-hmm. Stuff. 2 time points, it still feels very tough, but like at some point it's going to stop helping you that much. And then on the other angle, I guess it's that money you could have been spending on more time, you could spend on more time points, you could spend on more perturbations. It's like curious what your thoughts are on what makes these sorts of methods work best.
You want to answer?
Yeah, that's a good question. And I think that one of the problems we were facing is, I agree, we had time points. It might have been better if we had more time points. And the other thing is that for each of these conditions, each of these perturbations, we had a very small amount of cells. So if we spread it across more time points, then we might have 100 cells per bin, and that doesn't give us a large level of interpretability and predictive power. So I think kind of both would be good.
Yeah, and I think another thing, right, is like this is kind of early work into including perturbations in the models, right? This is kind of a new thing, and there's very few methods that do this. So it's definitely something we're trying to explore, right? Is how much can knockouts add to the expressiveness or the understanding of the model, for sure. Yeah.
Great, thank you. One thing I was interested in is when you think about this kind of molecular data, you usually want to think about a certain number of genes, and so you've shown some nice scaling performance with like hundreds of features, I guess. But even some people would argue, hundreds isn't enough. If I want to really be able to interpret that kind of pathway fit into that one and fit into that one, then I kind of need to have like a critical number of genes for like grouping them together and really teasing out the biology of like what's happening. And so yeah, we thought about strategies for like going into like larger amount of genes to put into the model.
Mm-hmm.
Do you think it's just a scalability problem? Do you think it's also a modeling problem? And you kind of circumvented that in the real data by going with like 18, I guess, or like the very few genes that you were looking into.
Mm-hmm.
But for working with the real data and making interpretations, I think it's a little bit limited.
Yeah, so I mean the 18 by 18 matrix, right, that was the from the study, right? That's the only part of the GRN they had experimented out. So I guess one of the things, right, is it's very data limited, right? For real data, right? I think in terms of the scaling experiment, right, we just went up to 500 dimensions to show the scaling factor. But that's kind of part of our approach is right, getting rid of this simulation, the simulation aspect, right, that NGM node does. Ours is simulation free, and so you could definitely scale this up to thousands of genes. These training times are relatively fast, right? They're like, we're training these models in a few minutes. You could absolutely scale to thousands of genes, but I think we are more limited by the data, right? And the real experiment data set kind of shows that. Right there, it's only over 100 genes, and the actual GRN is only over 18 by 18 genes. So,
yeah, I agree with that. And just to add on, it's data limitation, but I also think one of the bottlenecks is the NGM architecture we use. And the reason I think this is a limitation is because we have this FC1 fully connected layer that's nonlinear, but that's the only layer. So we can't model higher order interactions. We can't model other interactions that may have an effect. We can't measure superlinearity. We can't measure a lot of these things. So it could be a limitation in architecture as well. And if we can figure out a better way to scale this up where we have a more complex way of measuring the graph structure, I think that's also something we could try to do.
I think in the interest of recovering some of the time we lost for stand up, we'll go ahead and end it there. Thank the speakers again.
No, thank you. Great, thanks. You uploaded the slides. Yeah. Great. Are you seeing them?
One on four.
It just worked. I think we're still sharing. The one thing, I'm going to stop the share and start again. I realize it's a little bit cut off.
Okay.
Okay, you're all good.
All right, perfect. Hello, everyone. That's my good, the microphone? Oh, okay. Hello, everyone. My name is João. I'm currently a first-year PhD student at the Computational lab at Yale. And I'll talk to you guys a little bit about STAGED. So the name is a mouthful, so it's a multi-agent neural network for learning cellular interaction dynamics. But I think it's good that I came after Jacob, because what we're trying to do is that we're trying to use deep neural networks to explain the biology. So not only just perform specific tasks, we're trying to understand what is the underlying biology there. So it's important first. to give just some background on transcriptomics data. Like I think now everyone is familiar, and I think it's good that I'm in this session as well. So our idea is that, okay, we have this system, this biological system that has some latent structure. So let's say stem cells evolving. So it's a branching problem where the measurements we actually have is this cell by gene matrix. So we have this cell by gene matrix, and what we usually do is that we just project this into some another dimension. People usually use UMAP, but we use Fate also because we developed it, also because it keeps this structure. So Fate kind of recovers this tree structure, and in a way, using some diffusion processes, that we have this continuum mapping of the space. So we can actually now use this new space to infer our trajectories. And that's what MioFlow did, and that was another paper. What they do is, okay, I can see this structure, this tree structure. How are the genes evolving inside that structure? So we can infer multiple trajectories there. And from there, we can go back into our gene space. So you infer the trajectories on your latent space. Then you go back to gene space, and now you have each gene evolving inside each trajectory. So this enables us to visualize how each gene is being activated during the entire trajectory or the continuous trajectory. So the interesting part, well, this by itself is extremely interesting because you can find which genes are being knocked down or going up in each specific trajectory. But if you focus on the heat map, this heat map is actually hiding that it actually learns the internal gene regulatory network. So if I go here to like the initial step, the first block of genes, this could be like our initial node A of genes, and they're influencing the subsequent genes on this heat map. But you also have those like nonlinear relations where, for example, this group of genes here can be also responsible for some other node. So you can use something like Granger causality and infer these gene regulatory networks from this heat map. But as you guys saw at the start, I was talking about spatial data, and I've just been talking about transcriptomics, which is just to give some background on this idea of inferring those gene regulatory networks. An important step is also the fact that like these gene regulatory networks, they're not static for each cell, especially in like a stem cell that is evolving. The gene regulatory network is changing through time. So we could just kind of like bin this heat map and try to infer how those gene regulatory networks are changing in the trajectory. So we're going from that stem cell that has a fixed GRN, and now we're evolving to different new cell types, but we're seeing how the GRNs are evolving. This was what Rutini was doing. So now finally, we're going to spatial data, which is, okay, all of this works if you have this single cell data. But now we have this extra information, which is we know the exact space, so we can now try to model the microenvironment. And why this is important, I'd like to highlight like this. This is like the classic image from Xenium. But if you see cells in block A, there are those green cell types. And you can also see in B, you have some green cell types as well. The difference between these two cell types on this transcriptome level could be almost none. And that's why they're mapped as the same color. But their microenvironment is completely different. So like cells in the microenvironment A, they're neighbored by green cells as well. And on the microenvironment B, by those orange cells. So how can we try to plug this information in our model and try to find those intercellular connections? So the idea, and a lot of people in spatial transcriptomics do that, we build this kind of hierarchical graph where we have this first level graph, which is your cell and your neighbors connected. And then we plug inside each cell its internal GRN. And then we can just try to infer edges between those two GRNs. So we can find literally like gene level connections between those two. And if even more than that, you have this time evolving spatial transcriptomics data. So you have multiple slices through time. Now you can infer how those cellular interactions are evolving through time as well. So how is Stage actually trying to tackle this problem? So again, I always go back here, just sometimes it can be too quick. The whole idea is that we have this initial level of like cell-cell communication, so cells in the space, and the second level, which is the internal GRN. And we're doing some kind of agent-based learning to infer those. So this is a pretty high-level architecture. How would you do that? Basically, we create the spatial graph, you just connect to your neighbors. Then we plug the cell-cell GRN, so each internal cell receives its GRN. We press this to a graph attention network, and we try to learn what is the gene expression on the next time point. So we get this like time evolution from the previous methods I showed, so mu flow, and then we try to predict the next time point, basically. And if you learn this well, you can then now look at your graph attention networks and see which edges it's attending to. But of course, there's multiple things, so we expand this a little bit a little bit further. So the biological prior the first one we do here is ligand receptor pairs. So we know that some genes, they're ligand and receptors. So we don't want to connect my entire GRN to my neighbor's entire GRN if I have the information that, for example, gene A connects only to gene B. So only connect cells that we have some biological information that they should be talking. This is like different for each data set and for the problem. We usually work with a biologist that kind of knows like, okay, this gene usually interacts with that one, so we can start making those connections. Then we spend a little bit this like idea of just an MLP doing basically predicting the next time point, and we plug a neural ODE here. And the reason is because on those trajectories, the genes are not, well, first, just using MLP for like dynamic inference is not good enough. So the idea here is also that there's some kind of continuity to that gene. And that's why those trends, like the gene wouldn't go from like super activated to 0 and then back to super activated. So we're using a neural ODE to make everything smoother and also get better dynamic inference. And then finally, the second biological prior is the idea of agent-based learning. If you simply take the entire space and plug into a graph attention network, this would be a huge graph, extremely hard to interpret. All the attention will be all around. So what we do is that we hope that each graph attention network will basically model your cell type. So what we do is we instantiate one graph attention network per cell type. So hopefully if we go back in that example where we have the green cell and its neighbors green, all of those are being modeled by the same graph attention network, but then the orange cell is modeling its neighbors. So we have one for each cell type, and now this will enable us to actually look into those graph attention networks and recover the spatial temporal regulatory dynamics for that specific cell type at a specific time point. So again, just basically reiterating everything is we have this spatial graph, we model each cell GRN, then we pass through those graph attention networks and try to predict the next time point. So the task itself here is not as important, it's just a way for the model to learn how those genes are being, basically how this signal is passing on your graph. The actual final output we want is to find those connections between cell types. And they are also like biased towards biology because we forced it to have those ligand receptor pairs. So this is the entire idea. Now we have some preliminary results. I just wanted to show. So first, the important thing is like, can you actually do the task? Can you predict the dynamics? So this is just for some random simulated data. We just simulated like oscillatory dynamics just to get some complicated. Usually genes don't perform like that, at least in our experience, but we just do that to see, okay, this neural ODE is actually capturing the movement, so it's good enough. And then once you have that settled up, you can go back to the graph and try to understand what is going on. I think this figure needs a lot of improvement, but the idea is that we did some like circular GRMs. Turns out it's really hard to simulate GRMs, and I think the last talk gave me a good idea of like a tool I could use. But we just put a gene, basically each gene there is sending a signal to the next one. So one is activating, the other one is activating after that. So I have this cascade of genes activating. We learn the dynamics, and then we try to visualize the attention. So we're focusing on gene 1 here, and we can see, for example, in evolving time points, like time point 5, time point 6, time point 7, we can see the attention going towards gene 1 and going away. So hopefully that's what we obtain when we go to like true biological data. In the end, we did apply this to some real data. So there's some data set on Alzheimer's, microglia evolution in Alzheimer's disease. The top one, like the panel A, is just how we're getting the gene trends. So the gene trends is like, it's noisy, but it's kind of following the trend, which is the most important part. Like you don't need to actually perfectly match it, but if you're following the trend, then the attention should be good enough. And then panel B is kind of trying to showcase for a specific gene, how is the attention going there. But this still needs a lot of improvement, and I'm happy if anyone has a good idea of like a better data set or a better way to model those as well. And then some next steps is like the first one is actually trying to use those simulated GRMs. As I mentioned, turns out this is an extremely hard problem. So we're trying to use GillSpy to module all those GRMs and trying to get a reference frame so I can show you guys like, oh, this is an actual network of an underlying GRM, and this is how our network is performing. GillSpy works well if you're modeling one GRM inside one cell. When you try to make them communicate, it becomes a little bit of a trickier problem. And then, so one of our ideas is to use like small simulation. There's a whole like PhD on just modeling three genes GRNs. So we can try to use that to kind of validate our model as well. Of course, improve our benchmarking. There's a lot of talks about like applying multi-agent RL instead of the graph attention network. So we're thinking about that. And finally, we are collaborating with some people at University of South Wales in Australia to basically try to apply this to real-world cancer data where you're having metastases and try to have some better results. So yeah, so I just want to acknowledge my other co-authors, Scott and Shinji, and I'm happy to take any questions you guys might have.
Questions? Nice work. So the thing I was missing was, are you thinking about data settings where you would have it's the same cells across different time points, like you have that correspondence already somehow? Because I guess a lot of these things obviously are just destructive, right? So we don't get that, but I guess we're asking why in things where you do still have that information.
Yeah, no, you don't have that. You infer that using, like, for example, any trajectory inference you have before. So you're gonna we use the optimal transport to map those cells in some sort of way. It's not a one-to-one.
Yeah.
It's more like a kind of like a density based.
Yeah, okay. And then so then there's that, so the spatial graph is also then changing across.
Yeah, exactly. But that like, it's actually what is good to model like those hierarchical graphs is that you don't care about the actual position, right? You just care about your neighbors. So basically, if I have this cell which is evolving and I know its neighborhood,
yeah,
I don't care about its actual position.
Yeah, sure.
But
that neighborhood is going to change. My neighboring shop.
Yeah, your neighboring is changing at every time point. Yeah, every time point you have a measurement for. All right.
I guess I have one quick question, which is, are these GRNs that you're learning, are they stable? I mean, like, you have a small number of time points, so I can imagine you learn a system that, like, in 3 steps looks okay, but if you were to actually run this out, right, it just kind of diverges and goes crazy.
Yeah, I think that's again, so one of the advantage of like those trajectory inference in a continuous state is that you can kind of create fake data with like the fact that it's continuous. But again, like, of course, it's not like, it doesn't match the reality as well as you would like. And so yeah, that's definitely a problem. It does stabilize, like the training stabilizes, but it could be just like kind of like an overfitting artifact. But when you're simulating that's really hard. Like the simulation is actually the hardest part. So I saw in the last talk, they talked about the Boo ODE, I think, but yeah, exactly. So we're still trying to kind of validate this, but I think that's the
Okay.
No, everyone. Yeah, so this graph here is like, there was a whole PhD thesis of someone trying to model those three. And there's like if I'm not wrong, like the black ones is like, it just never converges. The red ones is like converges, everything is destroyed. Blue ones is like, okay, you have actually one gene that stays stabilized. And so it's really unstable. Yeah.
All right. Thank you, speaker again.
Thank you very much.
Oh yeah. Do you want to try and do it off yours? So oh yeah, you're not in here, huh? I wonder why. Okay, I'm not sure why that happened. Oh, that's why. Okay. Actually, if you just the easiest would be if you put in your name and then we do also have your slides. We can put them back up here. Yeah, let's do save, and then this doesn't work, which is great. I don't know why it's crazy to me that Zoom still has these issues. Sorry, one more time. Have to refresh it, otherwise it doesn't work. Okay, you should have an email now. Okay, it should come through from Zoom. I think if not, we should just do. You uploaded your slides, we have this time.
Yeah, I'm ideally I'll keep the notes
Oh yeah, yeah.
and also make some changes.
Okay, there you go. Let's have you join.
Do I keep the audio?
No, the audio will go through mine. Yeah, so if you wanted to, yeah, I think you have to extend it if you want the to be able to do presenter notes. Yeah, and then you drag the one, drag the thing you want to present, I think, into there, and then you just need to share to Zoom. Yeah, I think that is all good.
Right. Thank you. Hi everyone, my name is Eljan, and today I'm excited to share some of our work on benchmarking foundation models for perturbation response modeling. So first of all, why do we care about perturbation modeling? Well, the future of therapeutics isn't about single drugs hitting single targets, but it's about these complex combinatorial interventions. And we'll need to hit multiple targets at the same time to get real synergistic effects. But the challenge is that manually testing every possible combination is obviously impossible. The search space is just too big. So the only realistic way of forward is through accurate prediction perturbation models to navigate this massive search space and find those promising high-order interventions. But before we get there, we need to solve the simpler problem, which is, can we actually build perturbation models that can generalize to single unseen perturbations? And in recent years, we've seen the rise of these biological foundation models, which are trained on massive data sets. And with the right objectives, they can capture causal links between the different genes. It's a promising direction. However, we've seen several works in the last few years which show that these foundation models actually often fail to beat simpler baselines like PCA or just linear models. And the question is why? Is it because the data is limiting for these models? Is it the downstream predictors are not the right ones? Is it the fine tuning, or are the models themselves just not good enough? And so to shed some light on this, we decided to benchmark a wide range of gene embeddings from different foundation models. And we designed the benchmark so that we're comparing the embeddings themselves, not confounding them with the choice of a predictor. And since we don't know what the right modality is for this task, and we actually don't even know what the right metric is to evaluate perturbation prediction, we pulled in embeddings from transcriptomics, proteomics, and other sources. And we also looked at both simpler metrics like L2 correlation, but also at more like some more advanced biological informed scoring schemes. And so there's two main tasks we focus on. The first is the standard gene expression prediction, for which we operate in pseudobulk space. So for each perturbation P, we average the expression of perturbed cells, and then we subtract from that the average expression of control cells. So that gives us this perturbation delta. And now, given the embedding for the perturbed gene, the goal is to predict exactly what this delta vector is. To do it, we use a k-nearest neighbors regressor, and we deliberately chose this predictor to be simple and easy to tune, so avoiding deep nets or other complex models. But interestingly, we found that even if you use other linear models like Lasso or Rich, you get very, very similar performance in terms of the ranking, at least. And so we just decided to proceed with KNNs because it's simple and straightforward. And the second task is goal term prediction, which gave us a more functional view of this task. We performed differential expression analysis followed by gene set enrichment analysis to actually determine the set of go terms for each perturbation, which are enriched in perturbed cells when compared to controls. And then we build this indicator matrix of perturbation term relationship. So there's a 1 if that term was enriched in that perturbation and a 0 otherwise. And now the task is to predict exactly these terms given the embeddings. And so for this task, we used a multi-label logistic regression where we weigh the classes to account for any underrepresented terms. And the setup lets us test not just whether embedding them can predict counts, but also whether they can tell us in the language of biology itself what the perturbation is actually doing. And so we evaluated foundation models from 5 different classes, along with a comprehensive set of simple baselines. And I will focus on 2 of these baselines. So the first one is the idealized baseline, which acts as a positive control baseline. And so here we give the model access to both train and test perturbations, and then feed PCA on this pseudobulk matrix, right? So the only information lost here is whatever you lose due to compression through PCA. And of course, this setup is unrealistic, but it's still useful because it shows us what's achievable in principle. So think of this as like an existence theorem in math. Like it tells us that such embeddings can exist and this level of performance is possible. And the second one is the random baseline, where the embeddings are just drawn at random from a normal distribution, and therefore contain absolutely no information about the different relationships between the perturbations. And so we do expect all the other embeddings to lie somewhere in between these 2 extremes, right? So the idealized baseline at the top and the random at the bottom. And speaking of embeddings, we consider several expression-based foundation models. We look at a few protein-based models and DNA models, like including models from the embeddings from the ADO family, SCGMPT, Sprint, DSM2, and so on. And also we look at 2 types of embeddings which are derived from prior knowledge, which comes either in the form of a string network or textual information. So for the network embeddings, which are based on string, we basically fit different types of graph neural networks, which are trained using contrastive losses to predict whether a link exists between 2 genes or not. And these are just different architectures, but the overall what they're trying to capture is something about the connectivity. We also consider several variants of GPT, which is basically just getting ChatGPT embeddings of different textual input, which in our case is either NCBI descriptions of genes or proteins, or is just the gene ontology terms from either biological processes, cellular component, or molecular function. And so overall, these models just capture a wide spectrum of modalities. And for the benchmark, we relied on 4 widely used cell lines in perturbation modeling. There's 2 data sets from Reblogel et al. And 2 from Natig et al. And from these, we focus on essential genes, which are basically genes which are important for cell function. And after filtering some of them, we retain around 15 to 1700 perturbations per data set. Of these 4 cell lines, 3 of them are cancerous. The only exception is RPE1, which is a non-cancerous reference. So before I dive into the results, one important point I want to emphasize is that cross-validation is absolutely essential. We observe very large variability between the different splits, as you can see from the plots. And reporting only a single split can be highly misleading. I learned that the hard way. I spent quite some time showing results on one split and thinking that one of these models was better than the rest, but that was actually not the case once we switched to cross-validation. And so to avoid that, we actually used a nested cross-validation scheme here. So we first tuned the hyperparameters of our models, which in this case is the KNN, using a inner train validation split. And then once we find the best hyperparameters, we refit the model on that full inner fold, and finally evaluate on a separate held out test data set. And so this process is repeated independently for each split and each embedding. Now for the expression-based models, we generate embeddings by first passing control cells through the model, which gives us a tensor of shape, typically of shape n cells by n genes by some embedding dimension. And then to get the final gene embeddings, we just average along the cells dimension. Some models here, like GeneFormer, for instance, work a bit differently because the gene set is variable. And so in these cases, the set of genes included for each cell depends on criteria like the most expressed genes or the highest variable genes And so as a result, the gene embeddings which are built on top of, by pulling the cell embeddings from these models, actually have variable number of cells as well. So we only pull the cells where the gene actually appears. So there's some variation there. So in these plots that I'm showing here, the black and red vertical lines, which I'm realizing are a bit hard to see, are basically representing the idealized and the random baseline. And as expected, most methods fall somewhere in between these two when we measure the L2 error. What's more interesting, though, is that we actually do see clear differences between embeddings, something that doesn't fully align with prior studies. And one reason for this could be the fact that many of these prior works actually study data sets which have a very small number of perturbations. Like, for example, there's a very popular Norman data set which everybody uses, which contains less than 200 perturbations. And so treating any kind of machine learning model on 200 samples is likely not going to generalize well. But in this case, we have around 1,500 plus perturbations per cell line, which is a much more diverse set of perturbations to work with. And we also see that most embeddings actually do outperform the simpler PCA baseline here. So this is a fair PCA, I should say, which is actually fitted on control cells only. So we fit this on the gene by control matrix, actually. And then we only take the embeddings for the perturbations in our data. Another point to highlight is that the results are fairly consistent across all the 4 cell lines. The relative ranking stays the same. And finally, notice the gap between the top performing embeddings and the idealized baseline. So it's like almost halfway from the random to idealized. So which suggests that there's probably still some room for improvement, although again, I should say that this idealized baseline is very unrealistic. So it's unclear if we can get close to it or not. Finally, the performance of the no change baseline, which is just predicting the expression of control. So it's saying that perturbation has no effect. So the gap between no change and random is actually much larger for RPE1, which is this non-cancerous cell line. And so what this suggests is that maybe for the cancer cell line, the knockouts typically don't change the cell very much because there's so much going on in this cancer cell line that just knocking out one gene doesn't do anything. But the effect is much, much stronger when you look at the non-cancerous cell line. So just one interesting observation. And so just to see what methods actually perform best, like we condense this result into this single summary plot, which is basically plotting the rank of each embedding across the 4 cell lines for each split. And so it's a little bit easier to see which embedding is consistent on best here. The clear winners are knowledge-based and network embeddings. So the top 2 performing methods are actually the GNNs that we trained on string, WaveGC and MBFNet. And close behind that, we see several variants of GMBT with the NCBI description performing best. Interestingly, though, text embeddings from Go cellular component outperform Go biological process, which suggests that mRNA or protein localization might be quite important for this task. Also, some expression-based FMs, like I said, surpass PCA, including SHGMBT, the ADOCell family, and scPrint. And for ADOCell, we also see a small but consistent improvement as you increase model size, which hints that scaling might be a viable path for improvement here. Finally, among protein embeddings, those from space, which is based on string, actually perform best. And I think they use, basically it's just string sequences, embeddings derived from one of the ProTrans transformer backbones. Now for the function prediction task, the ranking at the top looks very similar. So knowledge-driven and network embeddings once again perform best for this task. However, a key difference here is that the protein embeddings now outperform the expression-based models. And so in particular, this space sequence and also ADO protein IF actually show much stronger performance compared to the count prediction task. But still, none of the methods here comes close to the idealized baseline, which achieves nearly double the F1 macro score. Also worth noting that some of these models were trained using Go sets as inputs, so the comparison is not completely fair. Like for example, genotype B is trained using the actual Go graph as input, but still the network-based embeddings still perform best even there. So it's interesting to see how the rankings shift when you move from predicting counts to predicting more biological, more functional outcomes. That said, there's a few limitations to keep in mind. First, we only used one type of predictor, like I mentioned, and these were deliberately simple. It's possible that the real strength of some of these embeddings only shows up when paired with more powerful nonlinear models. Second, we only focused on predicting pseudopole vectors. Perhaps cell-to-cell variation also carries important signals, either by influencing optimization dynamics or by helping the model capture correlations and cellular context that gets lost when you pseudopole the cells. Finally, we did not fine-tune these models, which could well change the results significantly. However, I should say that it's also important to see what these models are learning without fine-tuning, which might hint what architectures or objectives are inherently better than others for this task. So all of these are interesting future directions, but I also want to mention two other exciting directions that we're currently exploring. First, we've seen that many embeddings are actually complementary to each other. And so different modalities perform better for different subsets of perturbations. And which raises the questions, well, what happens if you actually combine them? For instance, by jointly fine-tuning across modalities. And I'm not showing any results here, but basically this like results on the joint fine-tuning. But what I'm showing here is the performance of these different embeddings on all our perturbations for the K562 cell line. And we do see that for some of them, there's groups where some perturbations perform poorly. So blue here means better and red means worse. And there's other embeddings, like for example, the ADO and SGMT models, where those same embeddings, where they perform best on those same embeddings, but worse on the others. So combining them might bring some synergistic effects. Finally, a natural next step is to train a single model across different cell lines. If these are truly foundational models, then exposing them to more diverse data should only make them stronger. So to conclude, we presented a systematic band model study of perturbation embeddings with the focus on those derived from foundation models, and we evaluated their predictive utility separately from downstream predictors. So we included more than 20 embeddings from different modalities and different representation learning strategies. And these were evaluated using either L2 into the expression space or using this novel biologically interpreted formulation of perturbation response. And the results strongly should suggest that knowledge-based and network embeddings are currently very, very hard to beat. So with that, I thank you all for listening, and I'll take any questions.
Thank you. Thank you for the talk. I very much I was pleased to see that the embedding seems to be one of the best or like most important thing for comparing those different methods, and I very much agree with that. What I was curious about is one takeaway I got from some of your plots is that basically most methods still improve over random or over no change baselines. What I wanted to ask about the cross-validation, is it IID cross-validation that you conducted?
Yes.
Okay. So that brings my next question. I think it's actually I would be a bit critical, and I would think about the way that we would apply those algorithms. Would be I have my perturb-seq data, and I selected a pool of perturbations from some existing knowledge, so it's biased towards something. And in your case, it's still biased because it's essential genes, for example. So that target some of the same machinery, and they have like a lot of similar effects. And then I want to apply them on the whole, like let's say genome, or like, and then I have some kind of shift. And so maybe, and those numbers the number that you presented could become much worse when you start doing biologically informed split. So I wanted to know, maybe this is something you've already done, and or maybe it's in the manuscript, but like, do you still get the same conclusion that most methods are informative, or do you see that some embeddings are completely unuseful and some of them still are the results more nuanced, and how do they change, you think, if you would use a different cross-validation method?
Yeah, so we do have plans to run those more biologically informed splits, but the goal here was to just look at the relative ranking of them, right? So if the split is ID, it's also ID for the random baseline. also ID for the baselines, right?
Do you think the ranking could change if it changes this?
It's good, it's good. So that's it's left for future work.
If that was.
Great talk. I have a question in terms of the data and data distribution part. So I really like the part you mentioning, you did a lot of cross validation and the model has a variance, right? In terms of performance. And I think those variants kind of indicate, the training data and the held data, they have some distribution shift. So I'm working on a similar problem in the space of T-cell receptor. And, I think the hard part is, how, if we train a model on if we have the learned model on this training data set, and we have hopefully a brand new testing data set, and the model have never been seen before. So in that case, how can we make sure, what model to select or what kind of model we should use during the training to ensure that, the model will perform well on the unseen out distribution data set?
So in that case, do you mean unseen cell lines, or like what is this new data set? Is that unseen perturbations or unseen cell lines?
I guess it could be either. I don't know much about the cell line, but for my domain, I work in the T-cell receptor and the novel peptides. So for me, it's like a make prediction, predict the binding affinity for the novel peptides. And those novel peptides, the trained model has never seen before, so they probably know nothing about those data set, and the novel epitope are considered as out of distribution data set.
Yeah, I mean, it's a problem in machine learning in general, right? Like, how do you generalize to this?
Right.
New data sets.
How do you select the best model?
Exactly. Yeah. So that was not the focus of this work, but like for instance, the ARC challenge, I think, is doing something similar now, where you have a completely new cell line, like this stem cells, and you're trying to see how well can you generalize to that unseen cell line. Yeah. So it's a good direction for future work. But that was not the goal for this mission, for this benchmark here.
Great question.
No, it's the last question.
I have to say, yeah, great work and really valuable benchmark. I was wondering, so you kind of motivate this by basically methods to apply foundation models, but for the knowledge-based and network-based, maybe more specifically, do you think this could be used to make statements about how well ontologies or like knowledge graphs can be applied to certain biological systems, like taking a statement like the reactome database captures cancer cell line dynamics well, but maybe neural cell lines are much worse by applying it there. Do you think like that is those types of statements would be valid based on this type of analysis?
Yeah, absolutely. I mean, as well, we see that GPT also performs quite well, and that is just textual information, right? So what is it actually giving us? We think it's some sort of soft interaction. So still, obviously, ChatGPT wasn't trained with gene expression data, just trained on all the publications out there. And the best thing we can learn from that is some form of interaction, like what gene affects what gene. And so I think it definitely makes the point that this might be important for patient modeling.
Let's thank our speaker again.
Yeah, we have a break now. We're a little bit behind, but we'll restart at 5 to noon. So in 25 minutes.
For the
Yes, I guess I
do it?
mean it's all on my laptop,
Yeah.
so it's kind of
Makes
easy.
sense.
I have all but one of them, so
Yeah.
it's pretty close. So
Sounds good.
yeah.
Questions.
Yeah, no, it's good. I'm almost always happy to ask. The one time you can't ask questions is when it's your own student. And you're like, okay, so,
Great. Somebody
yeah. How's everything going? Settling in?
Good, yeah.
Yeah. Do you have rotation students already, or is that yeah, nice.
Oh, yeah.
Nice.
Oh,
Oh yeah, nice.
Nice that you stole my.
Oh, yeah. You still missed them.
Well, you got
Hey,
the crazy.
how's it going?
criteria. Thanks for the weird, like,
Yeah. Oh yeah. No worries. Yeah, it's been a busy time. I don't know, a lot going on. So. Oh hey, you're next, right? Yeah, I have your slides. I think right. You uploaded them. So yeah, so it's all I did. The only thing that's difficult is the presenter view. So like, if you're okay without presenter view, that's easier. We can. Yeah. Oh, yeah. You don't have it. Yeah. Yeah. It's yeah, it's all a little bit like more tricky than I feel like it should be. It's like this connects through HDMI, but that connects through Zoom, and so it's like there's this. There's like what's on here, what's on the external monitor, and what's going to Zoom, which are all like separate things. So it's like combinatorial, like and then when stuff just it actually doesn't work. So
I think PDF. Okay, so if you have the
yeah, that's the safest. PDF's the one thing that's like it's the safer thing. I usually
It's nothing fancy.
I do sometimes remember to do that. Well, it's like if you have something like Jacob's thing where there's like animation, crazy animations, then I think in theory you can have that stuff embedded in a PDF actually, but like it would, that is feels wrong somehow. Yeah. I did. My faculty talks are all Beamer, so yeah. But in Keynote usually these days. Actually, no, teach PowerPoint. Okay, great. Yeah, I have your. I should have told her. I got. Yeah, and in retrospect, I looked at it like.
Yeah, here's my first steps, and then I'll wrap it up.
Yeah, maybe that's what I should actually do now. Is be doing this. I should be checking like. Yeah, great. Well, thanks for being on top of it.
Yes, I did. Two months ago.
Wow,
No,
you're already
yeah.
back.
Good timing. Start the faculty position, all the energy.
Maybe like 2 weeks early, I was like, yes.
Oh yeah.
Wow. It's okay, yeah, but it's pretty busy. Yeah.
How's the sleep?
Okay as well. Yeah, not too much, but okay.
Where did you end up settling?
Housing, yes, NYU was housing by Washington Square Park. It's a 10-minute walk from here, so that's why I was very happy that this meeting was happening here. I was like, I don't need to travel, I can just go.
I stayed with friends at NYU last night, so yeah, it's awesome.
Is this always going to be in New York, or like
we haven't talked about. I mean, it's been in University of Washington the last few years. They were more supportive on the. No, I shouldn't. I guess. I mean, it's really not.
No, no,
It's not cutting off that much. There's nothing on them.
It's to be. It's been annoying for a bunch of
Yeah.
folks,
Yes.
Right. Okay. She's much better at shouting than I am.
Louder.
I tried. It was quite ineffective. It cost me harder they have like a gong.
Yeah. Oh, that's
yeah.
useful.
They should actually have that here, like, because it's always an issue.
Like some loudspeakers have a little alarm thing.
Actually, that's true. I guess the sound does go out there, so I guess that's a good point. Actually, they do hear it out there, so that's a good thing for it. Okay, Freeland's here. That's a sign that no, you don't need to be here. But that's a sign that we can stop. I think we should get going. Yeah. So, Luke Puzzle, thank you. Take it away.
Okay. So hi everybody, I'm Luc. I'm currently doing a postdoc in Paris at Sorbonne Université with Laurent Jacob. And today's just presenting more of a work in progress on a method we've been working for deep end-to-end likelihood-free phylogenetic inference. Okay, so the goal of phylogenetic inference is to describe the shared evolutionary history of sequences in a multiple sequence alignment. And the best way we found how to do that is with a tree that describes this history. And the tree is just a topology with a set of branch lengths. Now, traditionally, what you do to find these trees is use maximum likelihood-based tree reconstruction methods, at least for point estimation. And the way this works is you have some probabilistic model that describes how sequences evolve along the branches of a tree. And then you try out a bunch of different tree topologies and compute the likelihood, so the probability of seeing the alignment given the tree and the parameters of your model. And so you explore some part of the tree space and find the tree that optimizes or maximizes this likelihood. Now, that's kind of the state-of-the-art method. It's they're accurate, but they're quite slow because the tree space is absolutely enormous. And also just computing the likelihood for a given tree can be quite computationally intensive. So that's a drawback, but another perhaps more important drawback is that this limits us to using models where we can actually compute or at least approximate the likelihood. And that's not possible for a bunch of maybe more realistic or complex evolutionary models that might be interesting to use. So that's kind of the motivation behind likelihood-free inference or simulation-based inference, where it comes from the observation that, as I just said, going from the alignment to infer the best tree is hard through this model. However, going the other way around, so having a tree and simulating sequences along this tree according to the parameters of the evolutionary model is actually quite easy. So what you can do is you can simulate a bunch of these tree and alignment pairs that follow the model that you're working under. And then you try and learn the hard part and learn the mapping from the multiple sequence alignment onto the tree. Our first approach at this is called Phyloformer, and we published this last year. And we've taken a slightly simpler approach where we don't directly predict the tree, but instead use the phylogenetic distance matrix. So the sum of the branch lengths along the path between two tips as kind of a proxy for our tree. The way this works is we can embed our multiple sequence alignment into kind of a higher embedding space, and then create sequence pair embeddings because we're interested in pairwise distances. So we want to work in sequence pairs space. The way we do this is just average the two embeddings of the two sequence embeddings for each pair to get this pair embedding representation. Then we run it through actual attention stack. So that's the same as or very similar to MSA transformer, except that it's acting on sequence pairs instead of single sequences. So we do this for 6 blocks, and then we get hopefully a meaningful, informative representation of these sequence pairs. Then we can just project it back into a single dimensional embedding, take the average over the sequence length, and what you're left with is one single value per sequence pair. And with an L1 loss, we can optimize the model so that this corresponds to the true lower triangle of the true distance matrix. Now, and then at inference time, you just predict the distance matrix and feed it to whatever distance-based tree reconstruction algorithm like neighbor joining, or in our case, we're using FastME to recover the tree. Now this works pretty well. So filoformer here is in red. And if we look at this plot, this is the Kuhner-Felsenstein distance, which measures the distance between trees. And it takes into account both the topology and the branch length. And we can see here that we're in the kind of the same performance range as the maximum likelihood methods, which was a really good result for us. And since we run on a GPU, it's much faster. However, it's not all rosy because, well, it is a transformer-based method, so it's by far the most memory-intensive method. And also, a thing that was not necessarily puzzling us, but bothering us, and which is the motivation for this work, is if we look at the purely topological distance metric, which is Robinson-Foulds distance, you can see that where before we were in the same performance range as maximum likelihood, here there's a gap when you just look at topology. And we think that this comes from the fact that we're not actually predicting the tree, but we're predicting the distance matrix. In theory, that's an equivalent task because if you have the true distance matrix, there's a theoretical guarantees that you can recover the tree. In practice, it looks like that's not really the case in our scenario. So that's the motivation behind this project, which is, okay, how do we do actual end-to-end phylogenetic inference and actually predict trees with our network? Our first approach was to try and build a differentiable, end-to-end differentiable version of neighbor joining. It kind of worked, but we weren't really satisfied with it. So we tried to go a different route, and we're tackling this problem now from the approach of neural posterior estimation. So the way this works is you still have your probabilistic model, right? Your evolutionary model that describes the way sequences evolve along the tree, and some priors. And these priors just correspond to the settings on your simulators, basically. And the task we want to do is to estimate the posterior probability for our trees, so the probability of the tree given the alignment. The way we do this is that we can build a family of distributions which are parameterized by our neural network, which somewhat uninspiringly we're calling filoformer 2. And then we train our neural network so that this this distribution cube psi approximates the posterior by minimizing the KL divergence. Now in practice, you don't have access to this true posterior, so what you do is you just maximize the log approximate probability by sampling from our simulators a bunch of times. So the way this works, the way this looks like in our method is that we have here filoformer 2, which is composed of two modules. An embedding module, which we're calling EvoPF, which is going to take simulated alignment and produce kind of a more informative representation or embedding of this multiple sequence alignment. And then a module, which we call BisenJ, which is going to take as input the real tree and the embedded version of the MSA, and compute the probability of this real tree given the embedded MSA. And then since we rather minimize than maximize something, we just use the negative log cube psi as our loss to compute to try and find our optimal parameters. And then at inference time, the way we've designed our BisenJ module, we can just kind of flip a switch to go from probability estimation mode to sampling mode, and we can actually sample trees from the learned distribution. Since this is still a work in progress, and all the results I'm going to show you, it's all point estimation, and we're not actually comparing distributions. But this is something that our method can do, and that we're working on to see how well it does it. But not in this talk. Okay, so first, the EvoPF module, which is going to give us the embedding of the multiple sequence alignment. And it's basically an adaptation of EvoFormer, because the two tasks that EvoFormer and EvoPF are trying to solve are kind of transpositions of each other. We both have an MSA, and where EvoFormer is going to try and represent relationships between sites, we just kind of take the transpose and we want representations between sequences. So it's kind of generally the same architecture. You have a MSA representation and a pair representation that communicate with each other, and there's actual attention and a bunch of that. And at the end, what this gives you is MSA embedding that hopefully includes pair information in there. So what's nice about this is that it's more expressive than just a straight up MSA transformer because you have this pair representation. But it's also more lightweight than fileformer 1 because the pair representation is not tied to the sequence length. So we have this this way of embedding the multiple sequence alignment and extract pairwise information, hopefully. So now how do we actually compute the probability of the tree given this MSA representation? In order to explain how we're doing that, it's important to realize that a tree can be described as just a series of pairwise merges. And if you have an order on this merges, you have a unique way of describing the topology of your tree. And here, the order we're using is to just merge the shortest cherry that is available to be merged. So in this case, we start by merging this one because it's shorter than this cherry here. And then we merge this cherry, which has become available because this has been merged before. And then we merge this third one and then the fourth one. And so this ordered succession of merges, 1, 2, 3, 4, gives us a unique representation of our tree topology. And if we tie to these objects the branch lengths, we have a full representation, a description of our tree. So how do we use that to compute the posterior probability of the tree? Well, we kind of factorize the tree probability as the product of successive conditional merge probabilities. So at each, you apply each merge of the tree in succession, and the final tree probability is just the product of, at merge K, the topological probability of applying the merge of the real tree given all the previous mergers, and then the branch length component, which is what's the probability of seeing those branch lengths at that merge given all the previous merges. And so in practice, how is this implemented? So it's iterative approach where we apply each merge successively in sequence. And we start from these clade embeddings, which is just the average over the sequence length of the MSA embedding. The first thing we do is to compute a pair score, basically just a single value per clade pairs with bilinear form. And then we can run this through a softman to get a probability distribution. And then if we're here at merge I don't know if you can see, let's say we're merging human and dolphin, we can just go read the probability of that merge in our distribution. And that's going to be our topological component of this conditional merge probability. Now, since this is iterative, we want to apply the merge to our clade representation so that we can have this updated version of the clade representation for the next merge. And the way we do this is just with another bilinear form. We compute a parent embedding from the two child embeddings and just replace these two embeddings in the clade representation and shrink it by 1 to replace it with the parent embedding. And this parent embedding is also going to help us compute the branch length probabilities with yet another bilinear form. We just take one of the child embeddings and the parent embedding to compute the average of a normal distribution, which is going to give us the probability of one of the real branch lengths at this merge. And so what we get here, we have the topological component of our merge probability and the branch length component of our merge probability. And if we just keep doing this until the topology is fully resolved, until we've reached the end of our list of merges, we have the whole tree probability. And what's nice about doing it this way is that at inference time, we can kind of do the same thing, but here, instead of looking up what the probability of the real tree merge is, we can just sample from the distribution to sample a merge. And so we can keep doing this until we have a fully resolved topology, and we've just sampled a tree topology. Likewise, with the branch lengths, we predict means for normal, and then you can just sample branch lengths from those distributions. Okay, so I guess the question is, does it work? And in order to explore this, this is kind of very preliminary results, work in progress. we decided to test our model on kind of the, in retrospect, I guess is the hardest test case for us because it's a very simple evolutionary model, LG. This is the same test case as the Filoformer 1 paper. And this is kind of the best scenario for the maximum likelihood methods because it's a simple model, and it's kind of the worst scenario for us because we don't expect to be maximum likelihood. The model was trained with just topology, we kind of forget the branch lengths for now, on trees of size 50, and it's tested on a separate test set with a bunch of different tree sizes. And what we can see here is that on the size of the tree that it learned, it's actually really good. It's better than maximum likelihood, which is, we didn't quite expect that, but it's a nice surprise. And we think it's because it's able to learn from the prior distributions, of which maximum likelihood can't. But as soon as you kind of get away from this region where you train, there's kind of severe size overfitting to the tree size. However, if you do fine tuning on a data set with like different tree sizes, you can kind of mitigate this effect. And here we fine tune on trees of size 10 to 100. And so on all this kind of section here, we're doing better than maximum likelihood, which is we like that result. But overall, no, the cool thing is that we're doing much better than Filoformer, just in general, on topology, and so that's cool. But the other cool part is that it's even faster than the Filoformer because it's run entirely on the GPU. We do the tree inference on the GPU, whereas Filoformer had a CPU step where you had to actually infer the tree from the distance matrix. And it's much more scalable than Filoformer because we're able to infer bigger trees. But we have twice as many attention blocks, and the embedding dimension is like 4 times higher than Filoformer 1. So still, you know, a lot more memory intensive than some of the maximum likelihood methods, but much better than Filoformer. However, there's still some kind of short-term challenges that we'd like to resolve, hopefully quickly. And one of the things that we're really trying to do, which is proving to be more challenging than we thought, is to learn jointly the topology and the branch lengths. And we're not quite there yet. We're almost there, but it's not working sufficiently well enough for me to put results on these slides. And another thing that we'd really like to do is to try maybe and compare how good our distributions are and not just point estimations. And to do that, we need to compare maybe to like MCMC tools, which means we probably had to revise our priors so they work with MCMC. But overall, I mean, we're pretty happy with what we managed to do. Topologically, beating maximum likelihood methods on this kind of super simple LG model is, we're quite happy and proud of that result. Still while being more scalable than 541. And there still needs to be a little work, but we feel like we're pretty close to having an actual end-to-end phylogenetic inference tool with deep learning, which is cool. And so then kind of further timescale, what to do next once we've tackled these short-term challenges is to look at really where we think it could shine the most is kind of more complex biologically realistic models. Like, for example, can we do phylogenetic inference under a POTS model, sorry, or models, other models that model epistasis or selection forces, whatever. All of these are probably more biologically interesting than these really simple site-independent likelihood models. And so we think that could be really cool to try. And we did try some of these with Filoformer 1 and it worked. So we're pretty confident that we'll manage to make it work with this architecture. So thanks to all these people from the lab. Thanks to all of you guys for listening to me. And thank you again to the organizers for the conference. Yeah.
Yeah, we can do a couple of questions. Especially if I can figure out. Yeah. Oh, it's actually on.
Hi, nice talk.
Hi, thank you.
So one of the, as you say, tree space is really big. And neural networks don't generalize well outside of their training set size, unless you have right enough bias and scenario.
Yeah.
That.
So the prior is really important for your simulations.
Yes.
And so how do you choose that? And what happens if you switch, use like a slightly different evolutionary model for your prior?
So, well, there's two really levers that we need to like control. Well, is the tree topology distributions, and then the actual evolutionary model, which is going to give us the multiple sequence alignment. For the tree topology, we are trying to simulate trees that fit empirical databases of phylogenetic trees. There's a Treebase, I think it's called. And so we're using birth depth models to simulate trees and then rescaling the branches so they're not ultrametric. And then trying to control the diameter and other metrics so that it kind of fits into the distribution of trees we can see in Treebase. And then for evolutionary models, I mean, that's kind of you have to, this is still model based, right? It's not model free, so you have to pick a model or a selection of models. But that's also true of any likelihood method, right? Yeah, you have to also pick the model, right? So, but we did spend a lot of time trying to fit, like having a good tree topology prior, which was, yeah, not necessarily very easy, but yeah.
Thanks a lot.
Thank you.
I wanted to, can we go over the technical part when you explain how you fit the model? I was kind of
this,
or even after, I guess, or sorry, the elbow that you presented, the Kuba divergence.
Oh, this, yeah.
Yes, I was so in here. Basically, do you assume that your simulator is kind of differentiable and basically you sample from the simulator? Or I guess no, the simulator is fixed.
Yeah.
And you want to just learn the variational distribution.
Yeah.
But when you see, so what I was confused by is if you try to minimize the KL above, you would get an expectation over the posterior.
Yeah.
But here you have an expectation over the simulator. So do you minimize the other KL or do you?
To be honest, this kind of diversion comes from variational inference, I think. I'm actually not quite sure how it works out. I just know that people do it this way.
Okay, yeah.
For simulation-based inference, but you definitely don't need your simulators to be differentiable.
Okay.
Yeah.
Then maybe the reverse KL. Yeah. Thanks.
I have one question, which is I think related a little bit to Quade's question, which is, so you showed this one way around, that is, so if you train on smaller trees, you simulate smaller trees, and then you try to generalize the larger trees, it struggles.
Yeah.
Maybe not surprising. The other way around, like if you train on big trees, does that understand how to do little?
So we haven't tried that yet. Because it's still a fairly big model.
Yeah.
I
I
mean,
guess actually looking at this, like it does worse at 20. Is that, can I?
Yeah. I mean, I think
it's doing worse at 25.
it's just really fitting to whatever the size that you give it is.
There's some cool stuff on the, not quite chain of thought, but like varying inference time compute, these people nets where you can like adaptively apply the layers that they have a different number of times.
Okay.
To adapt to different, they've done this for like solving mazes.
Right.
I'm thinking that would be kind of analogous to this, where if you have a neural net that can adapt how much compute it does at inference time.
Yeah.
I don't know how it would fit into your framework entirely, but.
I mean, basically, I mean, since we're doing this iterative merge thing, there is a variable number of, there's a loop in there, right?
Yeah. All right, thank Luke again, and we have spotlights next, so if your spotlight presented, please be sort of available.
All right.
Thanks. Joe, you're up first, right?
Yeah, I'm usually not very good at timing my presentation, so this is my early apology for people looking for lunch breaks. This is just kidding. I'm not actually going to run over them. Okay, hello everybody. My name is Xiaoyu Su. I'm a PhD student at Columbia and also at Genome Center with David. So yesterday we had a very inspiring talk, keynotes actually from Barbara, talking about how you can learn rich rich information from existing spatial live recording. And now I'm going to share with you our own story of like gold mining from existing data. So the first component in this story is the gold detector. So here we want to solve the task of pattern detection, and which we want to separate, discover meaningful, interesting structures from, let's say, noise. So because we have tens of thousands of genes to screen, people usually rely on some geostatistics like the Moran's I index. Those statistics are great, but they are not perfect. For example, they can achieve perfect performance in simulations with artificial data. They can have very strong powers on some very strong data, but completely fail upon a little perturbation. So actually, we proved that theoretically for this particular method, the space of the invisible patterns to SparkX is much larger than the pattern you can detect. So what's the problem? Oh, this is a joke. So if you're not looking your slides gene by gene, there might be the case that you have a secret Mona Lisa hiding and smiling at you without being noticed. Okay, so what's the issue? The issue turns out to be that we do not know what are the patterns that are presented in the data, but we sort of want to detect all of them. At some point, we even want to detect the noise. I mean, if you keep observing the same chaotic behaviors in the disease, it means that the noise itself is a feature or a pattern that you should care about. So our solution to this is instead of shrinking the space of the spatial patterns, we would better rearrange it through like rich theories in a graph spectrum theory and also the non-parametric kernel-based tests. Happy to discuss detail at the poster section. So now I can talk to you about the real goal that we were able to find from this analysis. So there are a lot of talks on gene expression, but this is not the end of the story. Instead, it's only the beginning. So the function of the gene is largely determined on how you actually process it after it's being transcribed. So you can have one gene simultaneously being a tumor suppressor and also be an oncogene. So to model this complex multivariate selection of the isoform is very difficult. It's equivalent saying of now you're instead of detecting single frame patterns, you're detecting a multi-frame dynamics like a little gyps hidden inside the data, which we managed to solve. We apply our methods to real spatial transcriptomics data and surprisingly found out that there is enough information to get new insights. For example, this is with standard 10x Visium short read data. We show that you can have transcript level information from it. And this is a Xenium data, although they did not market it for this, but they do have multiple exon probes, allows you to separate different isoforms of the same genes. So I want to end my talk by showing you one of the little molecular Mona Lisas that we were able to find from this data set. So this is a gene in the mouse brain, and it had these two separate, exome usage, clearly separating the glial cell versus the neuron cells. Well, apologies for the, if you're interested, we can talk more at the poster section, and this is the QR code for our preprint. Thank you.
Oh, yes. Yeah, go ahead.
Hi everyone. So I'm Tianyu, a PhD candidate from Yale. So since Mishan has some personal issues, I can't come here in person. I will present this work on behalf of him. Our topic is towards building a generative pre-trained transformer for learning the cellular spatial context. So we are also working on spatial transforming part. The key idea contribution here is we notice that the current methods have some problems in tokenization, which means in our method, we actually understand the spatial cell context is very crucial. And we create a new method to encode the spatial information in the tokenization stage. And you can see that in this figure, we actually mixed the spatial position tokens and the gene expression tokens and pass it to the transformer backbone. And our input here contain both the cell's neighborhoods information and also their target cell location. Output will be the target cell gene expression profile. And this figure will show the structure in a better approach. And our key contributions can be decomposed into three components. The first one is like a spatial serialization, so which means that we pass the gene expression tokens with a mixture of spatial information and also spatial information together to the transformer architecture. And the second one is self-tokenization. We mentioned that we have encoded the expression profile here. And the last stage about their transformer block, and we'll reconstruct gene expressions based on autoregressive approach. Our method can facilitate various downstream applications. They are quite novel, like the generated unseen cells in the spatial context, and also the spatial classing, as well as spatial annotation. In this part, I will talk about the generation problems or generation tasks. You can see that we can actually generate unseen gene expressions by promoting the input cells. And you can see that the ground truth information and our generated information are quite close. And we also incorporate comprehensive benchmarking with Gauss processing on the MLPs. We can see our metrics show better results. The second advantage here is about spatial perturbations, which means that we can change about their spatial information and see how the gene expression will be affected. And you can see that we do observe, like after having the in silico perturbation, the gene expression Pearson here is higher than the control group. And also we have some examples here, like after having the in silico perturbations, we do see like there exists a very obvious gene expression changes. And if you have any questions, you can contact me, Sheng. Thank you.
Okay, I think you're good.
Hello everyone, my name is Kevin Lam and I'm a statistics PhD student at the University of British Columbia. And I'm really excited to present to you the CNSBM, which stands for the Copy Number Stochastic Block Model. And the CNSBM is a Bayesian statistical model catered towards the use on copy number variation. And the overall goal of the model is to be able to disentangle a set of copy number profiles into primary variation and residual variation. So here, when I talk about primary variation, I refer to more large-scale recurrent alterations, whereas when I talk about residual variation, this corresponds to finer sample-specific deviations. And you can think of that as, for example, chromosomal instabilities, and that might carry biological meaning and would otherwise not be captured within the primary variation. So towards this end, we developed the CNSBM model, which essentially models categorical copy number states as categories, and we use variational inference to jointly cluster the samples and the genomic bins. So in terms of outputs, you would receive cluster labels on the rows and cluster labels on the columns. So we applied the CNSBM model on a small low-grade glioma data set from the Cancer Genome Atlas project. So that contained 490 patients. And overall, we found that there were 6 primary clusters, which I've illustrated in the copy number profiles on the left. And as before, these clusters corresponded to arm or whole chromosome level alterations. And on the right-hand side, we have regrouped the samples in terms of their residual variation. And both the primary and residual clusters basically carried some biological meaning. So when we performed survival analysis on that data set, we found that primary clusters captured broad differences in survival probabilities, and residual clusters tended to refine these survival predictions. Yeah, that was a sneak peek of our work, and if you're interested, feel free to come join us at the poster session. Thanks.
Yes.
Hi everyone, my name is Shubham. I'm from Helmholtz Munich, and today I'll be talking how AI-based histopathology phenotyping reveals germline loci shaping breast cancer morphology. Just a brief background about what GWAS is. GWAS is a statistical study where you correlate SNPs and a phenotype of interest. And the strength of this association can be visualized by a Manhattan plot. Now, we have been doing GWAS for almost 20 years now, and we have identified thousands of loci. But we still lack understanding of biological process and pathways affected by these variants. Now, doing genetic analysis of intermediate phenotypes, such as gene expression, can help mitigate this by identifying tissue, cell types, and genes affected by disease loci. In our project, we use histology as an intermediate phenotype, and we ask the question, can histological images reveal how germline variants shape tumor morphology? Why do we want to use histology as an intermediate phenotype? Well, They're used in clinics for disease diagnosis, progression of disease, and genetic basis of cancer morphology remain largely unexplored. In our approach, we do this by doing GWAS of histology embedding derived from foundation model. And we extend this by doing GWAS of text reports embedding derived from foundation model. And we end up identifying 6 loci, 2 of which are co-localized across modalities. Then we perform downstream analysis for interpretation of this SNPs by visualizing the genetic effect of the SNPs in image space. We do pathway analysis, survival analysis, and we extract phenotype to link it to genotype and morphological changes. How we characterize the genetic effect on histology. Well, the GIF is not working here, but what you see is the cell proliferation from low stage. Okay, you see it. From low stage cancer grade to high stage cancer grade. This is the genetic effect from the loci in the previous, you see on chromosome 1. We do pathway analysis and survival analysis for the gene set linked with the SNP. If you're interested more, please come pass by my poster. Thank you.
Hide this for you.
Yeah.
Hi everyone, my name is Antoine. I am a postdoc at the Tenzi Lab at MSK, and we are working on this adaptive screening of organoids. And our goal is to find the best drug combination for a new patient. And we work under the constraint that we don't have genetic data for this patient because of time constraints. And we have very few rounds of iteration of the active learning loop. So we want to get as much information as possible with our first batch of experiments. And because we don't have much information for our new patient, like it's very difficult to find a good informative batch, and random is a pretty reasonable baseline in that case. But we want to do a little bit more better than random, so we propose this approach. Sorry. Okay, thank you. So this twofold approach where we use a pre-trained model trained on previous patients and previous studies of organoid drug screening. And we can, for each drug combination, we can have a IUC value for each of these patients, give us a representation. And in this representation, we use chemidoids to select a batch of combination. And then for the dose, we assign a dose to each of these drugs in this batch based on an importance sampling where we give more importance to those where the curvature of previous dose response is larger. And by doing these two techniques, we get significant improvements against the baseline, which is taking the batch at random. So if you want to know more about this approach, please check out my poster. Thank you.
I think I never got presentation 13. So are you Sergio or? Okay, great. I got yours in multiple formats, so that's great. Okay, that's the start.
Yeah. My body's still in California time, so good morning. So today I'm presenting the continued domain-specific pre-training of protein language models for PMAC binding prediction. Full mouth. I'm really interested in vaccine design, as we know that you can stimulate an immune response when provided a vaccine. You generate a population of immune cells that then, when coupled with a tumor cell, that presents a epitope to the surface of the cell by the MHC1, then we have a cell killing immune response. And this is really pivotal for the development of vaccines, understanding what neoantigens bind to the MHC to then have that immune response. But the questions remains, what new antigens should we target for this? What new antigens should we target in the vaccine? Well, in some large studies, we identify over 1 million new antigens. So experimentally, it's infeasible to test all of these. So we need accurate predictions. Again, we can narrow down this problem to the modeling the binding to the MEC class 1. And for this, we're going to be leveraging pre-trained protein language models. There's quite a few tips and tricks that we're going to also add to this. I didn't include them today, but please come check out the slide in my poster. But overall, there's quite a lot more problems that we're encountering with this type of modeling, where we have really sparse data. We have a very limited number of samples that we can train our models with. This is highly polymorphic MEC class 1 protein. So then we encounter that, and there's a lot of different ways we need to clean up this in order to make this more effective. So did this work? So yeah, compared to some other models that have been for a few years now, we are leveraging these pre-trained language models, and we are showing that this workflow is working, at least in our held-out test set, as well as some other benchmarks that we're evaluating this. So if you're interested into vaccine design and want to know more about the MEC, please come check me out at poster 14.
Okay, final spotlight. That's the first one, right?
No.
No, okay.
All right, hi everyone, my name is Sekum. I'm a PhD student in Craig Morris's lab at Memorial Sloan Kettering, and today I'm going to give you a brief preview into our work on mRNA bench. So as we are all familiar, alternative splicing can create multiple splice isoforms with distinct properties and functions, and self-supervised nucleotide foundation models have been shown to predict properties like these, but there are no existing mRNA-focused benchmarks. So we introduced mRNA Bench, which is the first mRNA-specific nucleotide foundation model benchmarking suite. We evaluated basically every existing pre-trained foundation model across DNA, non-coding RNA, and mRNA across a suite of tasks, including like variant effect prediction, half-life, localization, etc., for a total of 59 subtasks. In this benchmark, we categorize our tasks as either being local or global, where local sort of means that the readout or label that we're trying to predict arises from nucleotide level or motif level changes in our sequence, whereas global tasks sort of refer to maybe the sequence features that result in the label are scattered throughout across the sequence. And when we plot the performance across all the different models that we tested, where on the x-axis we have model size, on the y-axis we have average performance across our tasks, we see that the top performing models are EVO2 and Orthros, and the best performing models are ones that have seen mRNA sequence before. To look into this a little bit more, we wanted to understand what is like the performance difference between Orthros and EVO2. And what we found out is that Orthros basically does much better on global tasks versus local ones due to its contrastive learning pre-training objective, which has also been shown in the computer vision literature. So to solve this, we basically just naively added a local training objective called mass language modeling to train our Orthros model alongside contrastive learning. And we see that it basically outperforms all the other models at a fraction of the size. So what else did we do? We also also wanted to understand, why do DNA and non-coding RNA models maybe not perform as well in mRNA tasks? And so we also did some analysis through a compression lens, as well as evaluating the generalization capabilities of these models using some sort of compositional generalization task. So if any of this sounds interesting, please feel free to come talk to me at the poster session and check out our preprint. Thank you very much.
All right, thank you to all the Spotlight presenters for being efficient. We have lunch now, and we'll be restarting at 1:30. Yeah.
Can we go grab lunch? Yeah.
There is pizza. I mean, you're obviously very welcome to get a nicer
Tattooed. Yeah.
Like a nice Korean place. I don't know.
Yeah, I mean, that's a ton of places, so, yeah.
The big city, as I
Yeah.
understand it.
turnout.
Yeah. Wait, what is it this time? Just
Weekends.
no, nice. Yeah. Yesterday was do not suck.
Yeah, Goody Tama. Yes. Goody Tama is like the most depressed egg ever too. If you ever see this character, hilarious.
Okay.
Are we good? We're good.
Now we're ready to.
Yeah. All right, we're going to start the afternoon session now. I'm really excited to introduce Richard Bonneau. Rich is currently the Vice President of Machine Learning for Drug Discovery at Genentech, where he co-founded and co-leads Prescient Design, a molecular design accelerator, developing new methods that combine machine learning and molecular modeling to advance therapeutic discovery. He also had a long academic career as a professor of biology and computer science at NYU, where his lab has made important contributions across systems biology, regulatory genomics, and macular molecular modeling. Richard has earned his PhD at UW, where he made seminal contributions to Rosetta. He has not only pioneered methods for protein function prediction, but also for networks of genes and even social media. And many of his contributions remain foundational to the field today. He also has an incredibly dry and witty humor that I hope he will sprinkle throughout his talk today. No pressure.
Yeah. That might delay the first joke in the talk by a little bit. It's too much pressure. Well, first of all, thanks to everyone who's here, who's given their posters. Thanks to the organizers who did a fantastic enough job that I'm in so much trouble because I've skipped very little of this conference, and yeah, I'm in serious trouble. My inbox is filled with stinkograms now. Before I start, I'll tell you a little bit about how we set things up to do what we do at Genentech, and like Peter shared, we apply machine learning methods to drug discovery. And you can imagine doing that in a lot of ways. And one of the ways you can imagine doing that is the way that is really part of the media narrative most of the time when we talk about startups that are contributing to drug discovery. And that's you take a rate limiting step and you just really focus on that and do it well. And that's a great way to do science. But the problem with drug discovery is that there's so many steps and they're so integrated that even though I'm trying to think about molecules and rotating bonds and DNA and all that good stuff, DNA is also a molecule, I guess. We do actually have to think a lot about the engineering and the machine learning overarching meta layers, things like agents and LLMs. And so I'll talk mostly about molecules, but I'll show just a little bit about how we try to put these things together in an end-to-end system, because the drug discovery process does include a lot of surprises and a lot of things that are actually more about process engineering than about molecules. And the same was true the early days of scaling up genomics, right? So we're used to this. And so to that end, we've got tech dev that's really aimed at small and large molecule drug discovery. We put peptides in with small molecules because we modify the heck out of them at Roche. We put glues, molecular glues, prox agents, and a lot of other things in with small molecules. Large molecules, we include nucleic acids and other modified nucleic acids because they end up fitting that way. And so really, are you essentially a smile string or are you a sequence? That's the sort of division there. Frontiers research is led by Kyung-Young Cho, who is sort of famous for his work with Yashua Bengio on attention and some other things. But he leads a group that is really just allowed to go into deep space, which is a lot of fun. They do basic science, but they're always doing it with Roche data. And the idea that you have basic science really just contributing many, many tens of fold their salary back to the company, it actually really works wonderfully well. They publish, they're free. Nothing that they do is closed source. But the benefit of having these folks innovating within our efforts is just something we're reminded of every day. And then the LLM and engineering teams, when you try to scale these things to active drug discovery projects, how do you shelter your scientists from the praxis of deploying things into these tight deadline-driven projects? That's something that we have to think about, but it's something that we've been able to do really well.
And that effort's led by Henry Dwyer, who used to work at Lyft and Facebook and all these other places, but is famous for freeing Lyft to the Google Spatial API, which apparently was a big deal and involved a lot of people. And so now he's freeing us from the fact that we are really bad at writing code. So it works. Okay. Before I start, I'll just kind of talk also a little bit about, again, the overall context, which is that, right now, drug discovery is a process where there are lots of serial handoffs, right? The first stages are all wrapped up into the orange part at the top of this, and that is we do genomics, we look at human genetics, we examine what we know about a disease, and we try to find targets that, when perturbed, would allow for some mitigation of either the disease state or the prognosis or progression of the disease, right? And, in the beginning, of targeted therapies, as they're called, it was really about oncology. It was a lot of work was done with targeting specific things that mark a particular tumor and a bunch of other cells that you can live without. And that was targeted therapy. Sarah Mustafavi and Fabian are moving beyond that to think about synergy, to think about the fact that most drugs today that are exciting to me anyways are 3 and 4 valent. So there are actually many different pieces of perturbation all wrapped together. And they're wrapped together so we can control dose and location and things like that. But really thinking about synergies and thinking about target discovery, given how much more we know about biology and how to perturb and interrogate biology today, that's something that she's doing. And yeah, I think there are a lot of people from Genentech and from her group here, a couple posters, a couple nice talks from that side of things. That involves a lot of foundational models that surround DNA and regulation. And now the practical part of that, the part that relates into this talk that describes molecules, is that we envision collectively that when those targets are being perturbed and thought about and ranked and lined up as potential drug targets, the system is already queuing up the drug discovery efforts for all modalities, so that the ranks are perturbed not just by what's happening on the biology side, but also on how tractable, druggable, and what those drugs and modalities of drugs mean. And when I say modality, by the way, I mean what kind of drug, small molecule or large molecule, even though we typically reserve that word for data types. Here, it's a term that's commonly used in pharma to indicate what's the type of molecule that you're going to use as the basis for your therapy. I'll focus today on the bottom part here, which is our orchestration layers and also our drug discovery layers. And I'll focus mostly on just how we make the molecules, because those are things where we have a lot of ability to share that work. And we don't think that that's the competitive differentiator. We think that designing molecules is something that's rapidly evolving to be a commodity capability across the organization. And Genentech really supports a lot of openness in that area. So I can talk about that. All right, so drug discovery with a lens really strongly focused on the middle part. The center of this waffle cookie is how we actually make the molecules. And here I'm focused on a handful of things that are important. The most important thing that we found is that we have a lab in the loop. That is, we don't ever build methods that we expect to be 0 shot or 1 shot. We have a whole lot of really high throughput methods for drug discovery. All the costs are already spent. So we should think up methods that are compatible with active learning scenarios. And so I'll talk a little bit about what that means. And I think for people who have thought about model-driven experimental design, it'll be a review. You're going to be irritated. It's going to be a little too basic. But I just want to illustrate one key point there. Integration across timelines. So if you have noisy predictors of what came before, if you had wanted to try something differently, if you have noisy predictors of what happens when you go into the clinic, these are things that we don't develop in my unit, but we have to be aware of, because there's such a huge combinatorial space of molecules we can design that hit the basic affinity, expression, and biophysical parameters that we can actually select given those other oracles, even if they're noisy. And so this is a key way that we compress timelines and also increase the chance of success for some of these molecules to go through to a clinical trial. And we've got a lot of nice examples there, but I won't talk a lot about that today, because things going into the clinic are usually things that, if I talk about them, the Swiss assassins drop from the helicopter, kill me dead. It's the most, well, you know how this works. Yeah, we have a few people from Genentech who know this. Another thing, and this is the thing I'll mostly focus on here, a lot of what we do in pharma is still really, you know, columns of people working different departments. The handoffs are slides sometimes, and so we're fixing that. The data systems at Genentech are going through a full revolution. But another key thing is that we think of each molecule, cell-based therapy, small molecule, large molecule. We think of the different modalities as being entirely different departments. And I think that tends to be necessary given that chemistry to make small molecules is very different from the molecular biology that's optimal for finding antibodies. There's also a lot of differences in how you develop those molecules and what tends to go wrong. So in the lab, there's some need for these modalities to be different departments, but there doesn't need to be that big of a difference on the computation side. And so we found that it's really actually quite a battle to fix that on the computational side. Culturally, there's a lot of resistance. It's also not so easy algorithmically, but I'll show you one attempt there to make models that span all the different types of molecules. And we have foundation models that are really based on the sequence, structure, joint encoding, and we also have generative structure-based models, and I'll show you the latter today. Okay, so diving into this cross-modality thing, at Genentech University, we do a little bit of everything. And so, you know, the stock price is not our main objective. It's to make good scientists, and you know, Roche has to live with that, right? So, and if this is recorded, I'm toast, so I'll be applying for jobs, you know, later. But you know, so you know, we have peptides, we have antibodies, we have all these things, and every once in a while, yeah, that's right. I have a backup plan. Yeah, I could sleep on Dave Gresham's couch or something like that. Yeah. All right, so this sometimes bothers me because I think we're putting so much effort into these different modalities. What if we concentrated all of our data collection efforts, everything on just a smaller number of things, smaller number of ways of perturbing the cell? The problem is when you look at the big experiment that's happening, all the different pharma happening, and you just read out what's working, what's actually affecting the lives of the greatest number of patients, you see, hold on, I have this is a good one. You see, you know, peptides, small molecules, cell-based receptor. All right, that's not going to work. You see basically a little bit of every modality. And you also see that sometimes these modalities are different in interesting ways because the biology that you need to perturb really does need to be localized by a migrating cell, or it needs to be a really attuned adaptive receptor response that you can only get with a cell or with a particular kind of molecule. Antibodies are also huge monsters, giant pains in the butt, but they bind the complement. They initiate cell killing. They last in the bloodstream upwards of multiple months sometimes. So, and they recycle. They get past the blood-brain barrier sometimes better than small molecules if you have the right binding modalities. And so, you know, these different modalities actually have not just different histories of how we find them, but they have a lot of really strong structure function reasons for keeping them all around. In general, if I was giving this talk about 10 years ago, I would just look at what's been approved lately and what the blockbuster drugs were. And that picture wouldn't have been a little of everything. It would have been almost entirely antibody, right? So starting with Genentech, and then, you know, lots of other companies got involved. Human products that mimic sort of hormones and other growth factors and things like that, and antibodies. Essentially, biologics were all their, well, they were some of the main entries into the most important parts of target therapies for oncology and definitely for immunology. And that's less case so now. But this, for anyone who's interested in kind of the previous iteration of this work, I just want to leave this up here. So the first author here, in case you're in the back and it's too small, is Frey, F-R-E, F-R-E-Y. The last author is Gogorovich, and everyone knows how to spell that. So we were able to actually look at, in this particular paper, many different targets where the target was attrited not because the molecular design failed, but because the biology evolved. We had a competitive molecule that we licensed, or there was some other reason why that drug discovery project wasn't necessarily going to be super important commercially. And so we got approval for upwards of 6 or 7 projects. And so we can go through, and we do go through here, the full drug discovery process, including our oracles, our generative AI, and our active learning framework. So I encourage anyone who's interested in that to take a peek there. But, you know, ultimately the goal is to skip the loop and to design antibodies directly, right? To just pick an epitope, which we mostly are drawing as green little splotches on the surface of this protein, and directly design into that. And as someone who is trained in the structure, design, structure prediction world, this is very attractive to me. And it's attractive not just because it would be impressive to be able to just directly design something, but it's also important because it's more mechanistically focused. The minute you design something against given epitope, you also have that model of how it bound and the geometry with it bound. And as you increase that accuracy in coming up with binders, presumably you also increase the accuracy of the underlying structure model. And this is very important because binding is just the beginning of the structure-function relationship. When we're modulating function with antibodies, we're bringing receptors together. We're pushing them apart. The geometry and the epitope that you bind the target protein with is almost always very important. Our antibody engineering colleagues always tell us that we're in love with this particular epitope, even though it's the worst binder and the rest of them are much better. So being able to directly design to a given pocket, if you're a small molecule, or to a given epitope, if you're an antibody, is actually almost as important, or it's perhaps much more important than the degree to which you optimize just the rote number. And so we have efforts for protein de novo design, as do many other people, many other groups out there. We also have our own small molecule and peptide generative frameworks. Reach out to me if you're interested in those, but we also have all our publications up on the site. The goal here, though, is to still go after that transformative technology of being able to just directly design molecules into any surface or active region on a protein or structured nucleotide. The goal is to be able to design with one system that can learn from every molecular interface, irrespective of whether or not it's small molecule, large molecule, nucleic acid, protein, et cetera. And so to this end, we've been developing things that are based on atomic representations that are modality agnostic. And we'll talk a little bit, and I'll show some papers that show the machineries that we're using. There have been a few mentions of neural fields and other ways of taking explicit spatial or graph representations and condensing them into these representations called neural fields that are more memory efficient and have a lot of other advantages in terms of training, not being quite as brittle. That's what we're ultimately going to get to here is a format there. But essentially what we want is we want a channel per atom. We want the light gray box, or the light gray part of the box, the dots, light gray dots, to show the protein surface. We want some way of indicating the center of mass of the desired epitope. And then we want to design molecules de novo into that pocket. And the first paper that did this, this unconditioned generation into pockets, really just saying don't bump into the pocket, and be a synthesizable, reasonable extant or realizable small molecule, was Voxmol. And I think we have the author in the audience today. I think raise your hand if okay, there we go. Yeah, usually wearing yellow, but red shirt today. So this was a really nice piece of work where, essentially they came to me and said, okay, we're going to use all this stuff from Vision. We're going to just do atoms or atoms. And I really thought it was far too ambitious. And, something like 3 weeks or a month later, they had a really nice generative model. After that, it was quite difficult. And there was quite a bit of work to figure out how to condition on pockets and really get the scoring right. That led to the VoxBind paper. And now they've got a combined method that will, I think, really be quite something. And so these are the papers that'll get you the authors. And of course, it's the smallest font possible. Apologies. But FuncBind is what I'll talk to you about today. And so here we have both the generative model, conditioning on pockets, and then also the ability to represent this substantially more efficiently and scale to things like proteins. The key part of FuncBind with respect to using neural fields and other things like that, this is really what lets us design proteins. A lot of that early work was really well suited to small molecules, but not to larger small molecules, barely to peptides, and definitely not to proteins. And now this scales to any of the modalities. And again, the early work, we had this encoder-decoder pair where we voxelize the protein surface. We encode initially just as a self-supervised generative process, and then ultimately as something conditioned on the pocket, and we decode. And this process of noising this and denoising this lets you step through the space and successively, iteratively generate new composition of matter. The really amazing, in my opinion, the really amazing advancement here is that now this model is trained on proteins, small molecules, and peptides at the same time. And this method seems to work quite well. Now, we do hear this story a lot, that when you switch to a multimodal framework, you can now see a substantially larger data set. You still have to figure out you still have to test things to make sure that that's going to work, right? You know, if you have lots of data, because you now have multimodality, but there's very small intersections between the different modalities of data, you know, it's not a guarantee that this is going to work better. But I think the guys have shown that this is actually something that's working quite a bit better. And so here we have different encoder heads for different modalities, but a shared decoder head from the latent space. And again, an explicit representation of what you're trying to decode. So a sort of discrete representation that tells you what is the desired modality to spit out. A real important part of this, though, is a piece of fundamental work that's actually a little bit old now. And so Saeed Saremi, who's with us, is also affiliated with Berkeley. He enjoys teaching 300-person machine learning classes, and so he teamed up with Jen Listgarten to teach recently, and was, thanking her and me. Oh, thanks for letting me do this. I think Jen's response was anytime, So, but this is, Saeed worked in the sort of neural empirical Bayes field, and then also started to apply that to different ways in which we can do diffusion-like sampling. And he came up with his own version of this, which he calls walk-jump sampling. And again, for us, what we can do is we can, when we get into the encoded space, we can then noise and denoise that process to move around. And this is something that lets us, hop over local minima, make substantive changes, not get stuck too close to whatever iteration you're on. It has a lot of different practical functions that are very similar to what you do with diffusion or other kind of noising and denoising processes. But I think this is a lot less brittle, or I know it's a lot less brittle, has a lot fewer free parameters. Maybe it has 0 or 1, depending on whether you think the variance of the data is a free parameter. And it's been something that in our hands has worked for different modalities and lots of different generative AI settings. And so I would recommend anyone who's not familiar with walk-jump sampling and Saeed's work to check this out. And again, let's see if I can... because everyone else had movies, I put some in this morning. This is just sort of showing what the process looks like. And so you can see that the molecules are changing conformation, their orientation relative to the pocket, and their composition. And, you probably can recognize the pocket on the left. No, we've seen enough nuclear hormone receptors that we all, anyways. So this is a case where we've been able to show that this works for all modalities. For small molecules and large molecules, we've got experimental validation, and we also have some, continual experimental validation that's in progress. For peptides, we don't have as much experimental validation, but we do have some ways of benchmarking that are really exciting. This is something that is in the new paper that is really exciting to me, and I think exciting to everyone on the team. This is a case where, that shows that the peptide didn't just use known amino acids that you see in the training set. It actually thought up a new amino acid that made some good contacts. It looked like it improved the score. It is synthesizable, right? Any primary amine can be put into either a monomer or submonomer synthesis here. So it's realizable, it's synthesizable. The side chain's not too exotic. It's not, just like going aliphatic and going crazy on us. But it's new and it wasn't in the training set. And so it's able to dream up functionalities that really let us believe that the small molecule and the peptide side of this maybe has a future. All right, and so this is state of the art on pocket conditioned small molecule generation. And, for that particular task, there's a lot of good public benchmarks. There's a lot of benchmarks that have been published either by companies that sell this kind of software or by other academic groups. Epitope conditioned CDR redesign, we've had some really nice results, and we've been able to get our colleagues in antibody engineering to try this out, to test a lot of these designs for both active projects and also projects that that we can talk about and publish. And so, you know, typically we get binding rates that are above or at state of the art, but we also know that they're conditioned on the epitope of interest and not just binders to any part of the surface of the protein. So that's really exciting. We'll you know, stay tuned for some benchmarks and some other data products that are part of this paper, but the benchmarking on the peptides is still something that's being actively worked on. Although I haven't checked in on this project in a week, so who knows, right? All right, coming back to, you know, so this is how we generate the molecules, how we make binders. Again, that's just part of this serial process. If we think about now the other drug discovery properties, how long does it stay around in the body? Does it elicit an immune response that causes it to be cleared? What happens when manufacturing tries to put it in one of those pen thingies, and it needs to be 150 milligrams per mil, or 180 mgs per mil, which is a lot, right? That's higher than the concentrations we use to crystallize proteins, right? All of those properties have to be optimized. And so we're not just turning this wheel to optimize affinity, but we're also figuring out what epitope do we like and playing around with the geometry of the binding. And we're also trying to play around with those drug-like properties. And that's true for small molecules, large molecules, peptides, all of these things. Structure function is more complex than is typically alluded to when we talk about drug discovery. So a key part of that, knowing that we're going to have multiple iterations, is figuring out in the beginning of the project, in the middle of the project, at the end of the project, what's the right set of designs to test. And here, I'd like to just shout out really quickly to Jiwon Park and Natasha Tagasovska, who, you know, early on led a lot of our error and active learning efforts through the Frontiers group, working with Kyung-Young Cho and others. And I think we have a few, Clara, who gave a really nice talk yesterday, works quite closely with Jiwon as well. So, but here the idea is, if you have a good generative AI and a good scoring system associated with that generative AI, then you can make, you know, you can't make infinite things that are going to work, but you can make a phenomenally large number of things that work, right? So how do you figure out what to test? And here the idea is, let's make an active learning framework that knows about both the hierarchies of how we collect data, and I'll show you what that means in the next slide, but also the error and substantial error in some of these properties. And for us, the key thing is that a lot of late stage drug discovery properties are data limited. And so those oracles are really bad, really noisy, but it's still, they've shown that you can still use them to estimate the relevant diversity along that property, even if you can't estimate accurately what that property is. And, you know, the best, you know, a really old paper, but a sort of a nice example of estimating what you want. There's the predictive performance, you know, Fmax and all that kind of stuff. But there's ratios, there's populations, there's estimating diversity accurately along the prediction. In the social sciences, Gary King at Harvard did a bunch of stuff where he showed that even if you're estimating sentiment or political polarization, when you do this at the population level, you can actually do a lot of things to estimate diversity and overall balance across the population, even if you can't really get accurate point-wise estimators. And so the README paper by Gary King would be a great place to look for, you know, a 20-year-old paper that kind of has this idea. But this is the idea. We want to be able to deal with our properties, pick out the right number, and screen hundreds of thousands, or design high-throughput libraries. But even when you're designing yeast display or ribosomal libraries, you still have to be pretty careful about how you spend your DNA bucks there. Again, I promise to irritate anyone who knows statistics, and so I'm going to do that right now. I actually gave a talk at Amazon Web Services, and I said, who knows what a Pareto front is? And no one raised their hand. And I resisted closing my laptop and walking out. But they're nice people. They're smart, and they're better engineers than I am, but that was scary. And then the rest of my talk was unintelligible, so oh well. So if we have two properties, and here we have yield and affinity, and again, yield, you know, antibodies are how many domains? I don't know, 12, 15, there's a lot. So there's multiple constant domains, and then there's two arms, and then a lot of our antibodies are also multiple formats, like multiple arms. And so some of our antibodies aren't Ys, they're like a Y with a Y sprouting out of it, right? Those are like trispecifics and quadraspecifics. So how those things express in your manufacturing system or your small volume systems is actually pretty variable. And then affinity is also something we care about. So these are non-trivial parameters. If we're selecting designs, if we select designs that explore volumes of this predicted property space that we haven't explored yet, and we increase the volume of data points, if we think of the data points as being vectors that, along with the origin, compose a volume, then increasing the volume, the more you increase the volume, the more valuable that point should be. And if the origin is also a point, then you also increase the volume in a way that gives you a chance to see something better than you've seen before. Okay, so that's great, except for one thing, which is that very often the relationships between things that we see, observe, that we care about in any kind of prototype-driven design cycle, whether it's sneakers, antibodies, cars, whatever, is usually pretty hierarchical. And so what Natasha and Jiwan did is they figured out ways to navigate the fact that all of these things are composed in trees. And so here, if we can't express or synthesize a molecule, then we can't decide if, or we can't test to see if it binds. If it doesn't bind, then our somewhat impatient experimental colleagues move on to something else, Sometimes they don't. Sometimes we can beg for negative controls and off we go. But there's a lot of hierarchy there. And that's what that Park and Tagasovska paper is about. Property DAG is really about how do you do this. And one of the ways that they figured this out was to actually replace a lot of the outer products in these calculations with something called bind copulas, which is a way of getting behaviors that you usually get with ranks, but still preserving some of the numerical, preserving some of the variance that you see in these values. So they used some very nice mathematical tricks to figure out how to compute that. And when we actually do this on a number of rounds, and this is an example from that paper where we show 4 design rounds and show that with real antibodies, using this method, we were able to push the Pareto frontier outwards and increase volume, find better things, and sort of find the right balance between exploitation of the model and improvement of the model. And it's real data, so it doesn't look perfect, but each design run has at least a substantial part of it that pokes out of the frontier. And I was excited to see this because over my career, attempts to do model-driven experimental design or active learning, I don't usually get past one iteration before the grant runs out or something else happens. And so this was really fun to see, and it's a testament to antibody engineering and Yan Wu at Genentech being really committed to this project as well. All right, so I'm going to try to wrap up and leave enough time for questions here. But again, another part of this process that is difficult is that the drug discovery life cycle, going from caring about a disease to target to molecule to biomarker that you need these days to run a good clinical trial, that full process, even if you're focused on those molecular and molecular biology aspects of the problem, not the clinical problem, it's still quite long. And each time you get one of those blue or greenish arrows leading into the other one, there's a handoff that's traditionally been pretty lossy. So, one of the ways that we're trying to fix that is to try to make sure that when we're doing different activities in the drug discovery space, and here's just a number of things that we would do for small molecule, multi-parameter developability, potency modeling, generating new variations just to throw new attempts at that particular scaffold into the mix. A key thing that we need to do is make sure that we bring that together into a platform and that those data structures are things that we can share with the other stages of the drug discovery process. But across science, whether you're in academia or industry, as we move through iterations of technology, as we move from area to area, maintaining interfaces that are specific for certain kinds of tools is something that's been really, really challenging. Bioconductor, a lot of scikit-learn. There's been some really beautiful examples where platforms have persisted across a lot of projects and a lot of time, but when they're too close to edge applications, it's really difficult to at any scale maintain these things other than open source projects. And open source projects, the famous ones I just mentioned, they happened, but I think like evolution, if we replayed the tape, they might not happen again. So how do you make interfaces that are flexible when your technology stack is changing and when the technologies that people care about change throughout the project lifecycle as well? One of the ways that we're hoping to do that is with large language models. And to do that, we're training different large language models for different stages of the life cycle. And there's a really big effort surrounding foundation models at Genie. We've heard about from other posters and projects, some that span the target discovery process. But this is the Genie suite of models, and we are moving to get these so that we have molecules that understand molecules, but also workflows and reasoning over multimodalities. And so this is something that we're pushing. If you're interested in this kind of work, we do have positions open in this and the prior project. But we can find that with very small models, so models that are at least 3 orders of magnitude smaller than the state of our models, we can actually get reasonable performance or equal performance for target safety assessment. We also find that when we use benchmarks that, unlike this one, are not public, we do substantially better than those other ones. So the public models, the enterprise models tend to indicate that they've been trained on the benchmarks, any benchmark that you can find publicly. So it's hard to evaluate these things. For tool calling and reasoning, our model in dark green also seems to do about as well as the enterprise models. And here we're comparing to a model that, because it's more than a month old, is a bit out of date. But as we look at our newest versions of models and their models, one thing that is clear is that if you have a task that's specific to you and your institution, it's very likely that a little bit of fine tuning you can beat with a smaller model what these larger enterprise models are doing. All of this comes together into Agency, which is our platform for building and using agents at Roche. This is something that, has got a lot of attention in industry, but I think here we have a lot of pilot projects across our Center of Excellence. Again, this is something that involves Sarah Mustafavi, John Marioni, Ryan Copping, and the others that I listed on that first slide. But we want to be able to connect to models, serve tools, serve agents, and all of these different things. How we scale to a user group as large as Roche is also kind of a challenge. And then, the difference between what an agent is and what an LLM is, I think it's challenging to develop these types of projects when most people don't know that difference. But so I'll just share the short version of that, which is that an agent is an LLM trapped in an infinite loop. And then it just spits out decisions which you have to ignore or take. But here we've got a framework where we have a nice way of putting together public and internal data. We've got a lot of nice connection to our access control and our systems that allow us to know what data is safe to train on. We also license lots of other models at Roche, including Claude, which is primarily used for code development, and OpenAI, which we don't use that much, but is actually quite useful in certain settings. The agent marketplace is really focused on a small number of pilots at the current time. There's a couple things coming from Sarah's group that are focused on target prioritization and discovery. There's some pilots that are focused on data discovery across 125 years of Roche. And then we, of course, have some that are focused on drug discovery. With these tools, we can take users at any part of the project, and they can either use tools, query LLMs, or use additional agents, which I guess is like just saying, iterate the user to an autonomous version of a question that they've asked. This is something that we found really exciting, but again, I'm pulling back to why this is relevant for this talk. This is, for me, a way to cut through the Gordian knot of making different interfaces for different stages, and it's also a way in which we could imagine something flexible enough that it'd be meaningful to someone in the clinic and to someone in the drug discovery space and to someone in the target prioritization space. We've got a long way to go, and the field does as well, to figure out how to test these models, to deploy them properly, to make sure that we can limit them to just the tasks they're supposed to do, so to make selective subsets of these models. But I do think that this is going to be a really important framework to avoid what we've all seen in different projects, which is that the science has a finite scope, but the informatics can grow and grow linearly with the time that the project's been running. And that's just because, well, a lot of that has to do with interface and access issues. I'm going to skip the demo, but we have a sort of a 10-minute long demo where my head of large language models shows someone using that to design an antibody. But these are essentially up and running for target discovery and up and running for drug discovery. But there's a lot of continual struggles to figure out how to get the best core models and to figure out the fact that where we had computational demands related to training, now we have computational demands related to inference because these agents are just bubbling away in the background. The data layer is something that has to be right. If a human can't figure it out, then a somewhat perturbed version of a human or an agent certainly can't either. And then there's a lot of things we do as scientists that we do to explore as opposed to the core workflows. So how much do we embrace the long tail of methods we want to compare to, methods that are out on the internet, all the thing that we do when we explore methods. This is South San Francisco campus, Genentech. Come and visit us. If you're interested in working on either the orange or the green part of that box, come see us. There are positions in pretty much all of the pillars in the Computational Center of Excellence, and we'd like to talk to you or at least give you some really free bad advice about your career, even if it doesn't fit. So thanks.
This may be outside of or above your degree, I'm not sure who that Roche is thinking about this. But with the recent settlement or a settlement up in the air for some of Anthropic's use of training data from copyrighted material, is Roche kind of approaching its own internal Genie models with any particular care to avoid obviously like a problem where a model based on copyrighted materials would then have its outputs liable to the people who copyrighted it, right? So they might own the profits of a small molecule developed.
Yeah, that's a good question. And it's not above my pay rate, right? Because you're kind of asking, how do you make sure you don't violate massive quantities of copyright law when you do what you do? So we have an interesting advantage that Roche has been going for 125 years, and we've had subscriptions to every journal the whole time. Who knows? A lot of these journals are a lot younger than 125 years old. But everything that goes into Genie, the lawyers have scrutinized very carefully. And so our training sets are a lot smaller. We didn't just go nuts on Slim Pajama or the Pile. And then we do have relationships with code repositories to make sure that we have access to the copyright or open access subsets of those. But even just fine tuning on our own data, super powerful, right? And so if we look at our own source trees and things like that, it's your code plus the paths that still work for you. And so, it's not just about the size of the data set. So even if you do things properly and you don't violate mass quantities of copyright law, you can still get pretty cool things. And then if you're not a big company, like you're a smaller startup, the public, the amount of free to use, non-copyrighted material is still pretty amazing in terms of, just abstracts and the open access part, like NIH Commons and things like that. It's phenomenal. And so we're definitely not a bigger is better shop. And so that makes it a little easier for us to adhere to those, not guidelines, I guess they're laws. Well, they're laws for now. So we'll see how those lawsuits go.
So this example of Funkwine coming up with a non-canonical amino acid as it's like as a nice fit was very curious about. So I don't know creative biology well enough to know whether there could be a lot of things that would be more in the backbone that it could have come up with that would then not, but maybe those wouldn't have been synthesizable. Like how often does I guess I'm just curious about this in general, like how often this happens and whether you see things like this where you're like, okay, that's actually unreasonable. This is an example that seems quite reasonable.
This one does, yeah.
Yeah.
Well, there's a couple things. So the backbone can be pretty messed up and still realizable, right? You can add an extra carbon, that's a beta peptide. You can branch the side chain off the nitrogen, and that's a peptoid. So you can do quite a bit there, and there's ways you can staple it and things like that. But I think right now, some of that modulation of the backbone is future work. But one way in which that is super important is that if you take the backbone nitrogens and you methylate them or you branch side chains off them, they're no longer hydrogen acceptor donor. They're not involved in hydrogen acceptor donor pairs with water, and they cross the membrane a lot better. So, messing around with the backbone more is part of the chemistry of going from a binder to something that has a chance of having good pharmacokinetics. And so the idea is to make sure that that's built into the model at some point. But for now, we've focused on the biophysics degeneration. I think that kind of like tuning the chemistry balance, the number of backbone nitrogens that are free, that's something that we haven't really talked about, but it will be important.
And there's I guess I'm unclear if there's technically synthesizable, and then there's if it's standard amino acids, I can get E. coli to make it. But if it's something weird, now it's a much more chemical. Synthesis is more involved, I assume. So there's a cost.
That's right. Yeah. And then anything that's compatible with the cool, there are a bunch of assays that involve the flexozyme or the ribosome that's been messed with so that it can take lots of different loaded tRNAs. There's a good number of them. I was about to say there's about 600 of those, but I have no idea. I think the number's probably grown. And a lot of those are also packaged into assays that are provided by companies like PeptiDream. There's a couple different companies that provide them. And so you can take 17 or 18 amino acids at a time, and they can be any that you choose from this big library. But those are all C-branched reasonably. There's a small number of N-methylated or N-capped amino acids you can fit in there, but that limits what chemistry you can play with. And that's the major contribution to our training set. So the model's always going to like things that are encodable in the flexozyme system, just because it's going to see a lot more data from that. And for those of you who didn't know you were in a chemistry seminar, it's too late. You're trapped. Yeah.
How do you think about the unwanted immunogenicity, especially of these very unusual molecules for amino acids?
That's a good question. So the oracle that we have for immunogenicity is based on T cell activation and also some work that we did with Adaptive, which is a company in Seattle. But essentially, we have data sets about T cell activation for about 2 million things, proteins, or we have pieces of proteins displayed. And then we also have data sets about protease cleavage. Those are actually public data sets. And then we have data sets about how things are displayed in MHCs. So that oracle works fairly well, but it's probably way too focused on MHC1s. So there's and the most common MHC alleles. So there's a big push to figure out how to get that to be less focused on just those most common MHC alleles. But we found that method actually works fairly well at removing those liabilities. The other way in which we deal with that is that at Genentech, we have a humanized mouse, which is great, except that when you immunize rabbits or you do de novo, you get better binders than come out of the GenN mouse. But we have a humanized mouse, and then we also have the ability to do traditional grafting things. But what we hope is that we can have an oracle based on what will present and what won't, what will elicit immune response. And that's called GRAF2. And there's a publication about that. If you're interested in that, I can send a note with that link.
Hi, thanks for the great talk. On one of the last slides, the word managed fine-tuning came up. Can you explain that a little bit more?
I can. That's pretty easy in the sense it's really just fine-tuning models, but finding ways of providing a good interface so that people can fine-tune models without a lot of expertise in training large models. Yeah. It's fine-tuning with guardrails so that people don't accidentally spend too much AWS bucks. Yeah. That could be bad.
Timing is perfect. Let's thank our keynote speaker. Are you in the? Okay. Yeah. Right.
All right, I'll stop. I'll quit.
Thank you so much for the introduction. I'm actually going to change gears and talk about a system that probably most people here have never worked with, which is plants and their different pathogen threats. So as you're probably familiar, there's a wide range of different organisms that have basically evolved distinct systems to be able to kind of limit potential threats. This includes CRISPRs, respiratory immunity, as well as adaptive immunity, so think of your antibodies. But across all these diverse systems, most organisms actually have some form of an innate immune system that's primarily composed of protein receptors, such as pattern recognition receptors, as well as nucleotide binding repeat receptors, or NLRs, or nucleotide receptors. And so most of my work has focused on these surface localized receptors known as PRRs. They basically bind to a variety of different molecules, including proteins, carbohydrates, lipids, etc. So luckily, I kind of got a brief introduction into some of the biology. But this is a quick review. If let's say you have a particular pathogen protein, in our case, a plant protease will basically cleave and release a small epitope. In plants, many of the epitopes for most pathogens are around 15 to about 30 amino acids long, and they're about, generally speaking, structured. And these will bind to our plant immune receptor within this concave surface. I primarily focus on leucine-rich repeat-based receptors. This essentially have this leucine-rich repeat motif, so conserve leucine residues with multiple different any kind of amino acid in between. And as you have multiple different levels of leucine-rich repeats, you create this concave surface, which becomes a binding interface between different size ligands. And so when you get this interaction, you can get different immunogenic outcomes, ranging from a high response in a plant down to no response being non-immunogenic. And so you might be wondering, well, how do plants survive and deal with so many different diverse threats? They're sessile, they don't have adaptive immunity, and really the innate immune system is its primary way. So plant innate immune receptors are extremely diverse, and there's many encoded in a single genome. So plant species can have between anywhere between 50 up to potentially 1,000 different receptors encoded, much of which aren't characterized whatsoever. And just to give you an idea, if I want to go into the lab and potentially characterize a receptor and an epitope pair, on a good day, I can probably screen between 20 and 30 combinations. So the throughput is much, much lower than a lot of other fields. But as you might imagine, we want to understand what this diversity means functionally. And so my goal as a postdoc was to basically build a predictive model to basically better understand these interactions and what they might mean. And so when we initially started this project, other groups had basically tried to use AlphaFold as a proxy for interactions. So they utilized that IPTM score for protein-protein binding interaction. And we looked at cases where each of these different receptors has a particular epitope, and we looked at that known functional data. At first glance, using that threshold of 0.8, it looked promising. Those that were generally immunogenic had a high score. Those that were non-immunogenic had a low score. But what we quickly realized is that as you look at other receptor epitope combinations, particularly those that don't have a cell structure, this approach quickly falls short. And bear in mind, we only have an extremely small number of cell structures. These proteins are extremely difficult to work with in the lab. And so utilizing kind of a structure-based approach was clearly a non-starter. And so while we don't understand totally why this doesn't work, because right now we hypothesize that if we don't have a known structure that AlphaFold was initially trained on, it essentially tries to create those secondary structures in the ligand that leads to these really poor scores. And so my goal was to basically use a protein linkage model approach to fine-tune on to better understand these interactions along this diverse binding plane. And so to do so, the first goal, of course, we had was, of course, generate training data. And unfortunately, we don't have a centralized database to basically collect any of that. So my role the first 2 months of my postdoc was going paper by paper, supplementary information, every pretty much line by line, pulling out individual receptor sequences, their epitope sequences, ranging papers from early 1999. And in total, I pulled 1,337 combinations, which I know it's not in the millions, but in our field, this is considered like a remarkable amount of data, which in total encompasses 91 different plant species. And we primarily focused on receptor epitope combinations that, of course, were really well characterized. And I basically then took that data and classified into those three main groups. So this was based obviously on controls, as well as the assay types of how sensitive that particular assay was done, and as well as kind of the descriptions the authors obviously held about their data. And if you want to get a sense of how diverse this data is, we can look at protein identity within a particular group. So here you're looking at 25 different screw variants and how diverse they are within that pool. So we still capture a good amount of diversity based on other people's publicly available data. And so one of the second tasks that we had, thank you, was to extract the ligand binding at the domains. In reality, since these proteins reside on the plant cell surface, we don't care about the internal components. We only compare what actually interfaces directly with that ligand. And since we knew that the LR repeats were highly variable, so these are some of the different numbers of LR repeats that are composed in our reference base, we needed a pipeline to basically extract that content. And so what we utilized was a combination of AlphaFold as well as RNA annotation, because we recognized due to this hypervariability of these repeats, using standard homology or hidden Markov model-based approaches generally fell short. And so, familiar with AlphaFold. So what is LR annotation? And so this tool was actually developed by a PhD student in our lab. Yes, we have a PhD student in math in plant biology. It's kind of a rare thing. And so what he did is he basically used these three-dimensional structures and reduced them down into two-dimensional space. And these leucine rich repeats, essentially when you reduce them using linear algebra, they actually form a circular coil. Think of it kind of like a slinky. And so you imagine during that different production, you can easily then be able to extract the LR domain with extreme precision because other functional domains don't represent that same property. And critically for me, because you can basically track where it is along that coil, you can actually use it to basically interface what part of the repeat then binds silicon. And again, if you look across all of our diverse data sets, 93 species in general, this is a pretty good approach to be able to say how many repeats are necessarily predicted based on how much is actually manually verified. Great. So now we have some data. We have now a pipeline to pull out the components. Let's build our protein linkage model. And I'm not good at naming software. We needed MathML. I know it's not the best name in the world, but you got to start somewhere. And so basically what we did is we really utilized a lot of inspiration from other work based in human immunology by utilizing ESM2, of course, freezing most of the layers except for the last one, extracting the embeddings of both receptors and the epitopes, but critically doing a couple major tweaks. First, we basically adjusted the weights based on that structural context, knowing that those residues that were on the intercom case surface were obviously more critical to the binding interface. Secondarily, we knew based on previous work that, unsurprisingly as a biochemist, other properties were really critical because it would give that specificity of what LRV receptor paired with which epitope. And so we transformed these sequences into that sort of data. Then after some feature modelization, we then run through a film layer, max pool, and then do a standard report neural network into these three major classes. And as you might imagine, this is the first model, like any machine learning model, actually any computational model for predicting these interactions. And so we want to create a more simple version as a baseline to kind of see how this compared. And so we call it seq only. Again, not the best name ever, but it's quite simply just using the embeddings as our primary context which we're going to train on. And so we split our data into 80-20 training test set, and we try to stratify based on both first the receptor combinations as well as the immune outcomes. And using standard performance metrics, you can see that overall, when you provide that additional kind of multimodal context, overall, the model is able to learn those sort of combinations and better predict what the outcomes might be. And overall, so this group right here is our immunogenic group, this group is our non-immunogenic group, and our weakly immunogenic group are overall generally okay at predicting what the outcomes might be. But I'm most, of course, interested in how good it responds in a particular receptor epitope combination. And so we break down that test data based on what combinations were correct versus which ones were miscorrect or misclassified, so those were predicted in the wrong state. We really quickly see that using a language model approach vastly outperforms basically any sort of structural modeling-based approach, which is unsurprising in my eyes. And so this will give you an idea. So these are the only two receptors in this data set that have any solved structure. So with that, we then wanted to further verify this model. And so I kind of did it two different ways. So the first way is, of course, take this original trained test set and basically randomize which receptors were paired with which epitopes. Because in theory, if it's truly learning the context of those interactions, if I randomize it, it should decrease rapidly. And that's exactly what we see. So this is our original one stratified, but if we randomize those pairs, metrics really quickly drop. But of course, as a immunologist first trained machine learning engineer, I of course wanted to test it in the lab. And so we focused on one particular receptor epitope combination to help further validate this, primarily using CSP22 and this plant receptor core, and then using a ROS-based assay, which is our most high throughput assay we have on hand to kind of quantify immunogenicity. And the reason we wanted to use core as well as CSP22 is these are really good model systems or a model interface because they have so much natural diversity. So here you're looking at a handful of core homologs as well as substantial entropy along the different domains, and you can see how much variation is already naturally present. Similarly, you also see that in a subset of the CSP22 epitopes that we were further able to test. And so in tandem with that, it's also critical that if you look at the model structures of some of these different receptors, they're heavily structurally conserved. And so this is what we then tried to go test. So we trained on this particular homolog. We're going to further validate using two different physical versions. And so in order to collect that training data, validation data, we relied upon a fantastic undergrad, Tatiana, who was actually blinded to our past questions. So she was kind of like a perfect test subject, kind of collect that data and not know necessarily entirely what we were doing. So no biases in the data. And so she actually ran these processes across these two different plant species. Again, giving a timelines idea of how long this takes. This work took about 3 to 4 months to collect in total. Plants have long generation times, or hard but slower to grow. And then we classify them again by their immunogenicity, same approach before. And so then we take this data, and once again, we read through our predictive model in comparison to AlphaFold. And you can see very quickly we do far, far superior than a more generalized based approach. Here again, you can see these are immunogenic pairs, our weakly immunogenic pairs, non-immunogenic pairs, and our weakly immunogenic groups. And the only major group that we most poorly perform on is, of course, that weakly immunogenic group that's predicted as immunogenic. And our running hypothesis A, this is the smallest amount of data we have to train on, so very little context. B, I am technically categorically labeling quantitative data, so it could be, these are truly immunogenic and based on my own particular biases. But as an immunologist, I'm still happy with that misclassification, it's still useful. But once again, as you see compared to AlphaFold, overall the modeling is so poor, and only those that are right are those that are not immunogenic, clearly an artifact of just poor modeling in general. And so, I won't dive too much more deeper into the other, there's a lot more, but I'll summarize. Unsurprisingly, fine-tuned models are obviously far more accurate in some cases than generalized approaches, but we recognize ML is not generalizable necessarily at every receptor epitope combination. So if we held out one particular group, never trained upon it, and tried to predict it, it performed extremely poorly. This could partly be due to the limited data we have. It could be due to the architecture, design, lots of things, but it's definitely something we want to work toward in the future. However, ML really excels at basically screening in silico with homologous epitope variants at scale. And for us, when we have hundreds of thousands of receptors and epitopes in combination, this becomes a really good way to get a landscape of what the outcome might be before we even go to the lab. And so we hope, knowing that we can then integrate more of a generative-based approach to basically accelerate how we engineer these different receptors to a particular group of pathogens. And then finally, I didn't touch on it, but if you want to read about it in our preprint, we've actually found early on that ML might actually help us find convergently evolved receptors. So this is one receptor 4. Another group found this convergently evolved receptors for more of a brute force approach. And our model is actually able to kind of predict this interaction, even though the sequences were totally different. And we hypothesized how to do this shared chemical property, unsurprisingly, but this model can actually pick that up. Yeah, if you want to look at it, it's in GitHub. We also implemented a version of Google Colab for people who are not so computationally savvy. And with that, I think I'm right on time. Okay. Oh, water real quick. I'm really thirsty.
I think this guy's got. Tony, go. Really nice work. The thing I was going to ask, I was actually going to, before you showed your conclusion slide, I was going to ask the thing about like, can you generalize to new families to follow up, but then you took my question, so okay. So the thing I was going to ask instead was, is there any, so right now you're like, okay, you're using ESM2 instead of trying to use AlphaFold predictions, but like there is presumably also an option of trying to use both, right? Like trying to use some information about either known or predictive structure, accepting that maybe the model can learn not to trust the structure information markings are mostly relying on ESM. But so like, is there any value to that, or like it's just not? Yeah.
Definitely there's potential for an ensemble model down the line and trying to combine that interface. Right now, I'm only hesitant just because we've seen early on AlphaFold so poor that I worry it weakens it.
Right.
That being said, there are groups that have shown, although so there are groups that show that if you iteratively do AlphaFold, you can get better prediction scores during the iterative process, and so maybe some version of that. Yeah.
I guess the sort of related question is, are there things that AlphaFold's getting right that you're getting? I mean, you're doing better, much better clearly overall, but are there things that some things is it where AlphaFold's being better, and is there any patterns for that?
Yeah. So I haven't looked too deeply into like exactly the model interpretability of our model and like exactly what they can predict which first is most critical. And so I think until they do that, right now AlphaFold is slightly better at that when it gets that context right, because you can learn a little more directly, like, is there steric clashes, do those residues line up and make sense? Even if I do the model really, knowing we have such little data, I'm not sure I would trust all of it. But I think in time we'll get there. I mean, data sets are rapidly increasing, even though we're slow. So one in the back.
Great talk. So I'm actually very curious about the immune system for plants and how you split up the data. Do they actually work across different plants? I actually don't know. Yeah.
So because we're pulling from every possible characterized like receptor epitope from 93 species, generally speaking, if you actually test another plant species, it works surprisingly well, but it's not really representative of the species, it's more representative of the sequence. So the sequence is crazy far diverged, just like any other language model, it's messier. It's not really about that plant and that plant. So.
Yeah, could you comment on, having found a specific example where AlphaFold or like structured models do particularly poorly? Do you think there's broader claims of examples where a structure-free model might always outperform if that's like intrinsically disordered or open proteins or something?
That is a really good question. Okay, so I don't know broadly across other systems, but I do think in general, if you have these kind of hydrophobic, but not entirely hydrophobic, unstructured ligands and sort of binding pockets, it can sometimes struggle because, especially in our case, the interface is just so large. And so I think if you have other proteins where you have also large interfaces, this kind of unstructured content, many times these structural models just want to force that point, even if it's not actually biologically represented. But I can give you any explicit examples. I'll talk.
I guess if there's no more from the audience, I will ask one of you. Does your model rely on AlphaFold predictions at the very beginning to be able to extract the LLM regions? Okay.
Yeah.
Yes.
The only part.
Yeah. Other questions? I think I see one in the back.
Great talk. Could you remind me again what the downstream application of this work involves? For the recognition of this new novel epitopes, or like how does this generalize?
Yeah, so I would say it's very good if you have any sort of receptor fundamental like variant, any combination, assuming it's one of those 13 that we trade on. That it does exceedingly well, which is extremely useful for us because right now we don't have a computation method. Right, we have to do it in the lab. That's expensive. It's slow. No other way around it. The second part of it that I think that we clearly demonstrated your presentation is like potentially the convergently evolved space. So in the past, the recent year, many companies have had thoughts on that. For something likely, especially because these ligands are essential to a lot of bacteria, not I'll speak with the bacteria specifically, but the bacterial ligands are essential to life, right? Flagellin components that obviously complex with the ribosome. And so it's unsurprising over long evolutionary periods at the time that obviously multiple receptors have evolved or multiple plants have evolved a receptor to that particular ligand target. And so in the past, to find them, we would have to do these huge functional screens. It took 3 to 5 years. There's a whole PhD students project just to find a receptor. So if we can use this as a way to kind of screen through that context and reduce the functional work down to, let's say, thousands to, let's say, maybe 50, that's massive gains in being able to find new receptors, which was previously a huge bottleneck.
Great, thank you so much. Let's thank our speaker again.
Yeah. Are you using, remind me, are you using yours? Yeah, okay. I did so. Oh yeah. Yeah, probably easy. The easier thing I think is just sharing the whole desktop is probably the easiest thing.
Yeah. Well, for instance, the school protein was very big. So now we have about 6,000.
Yeah, the problem is that shows the presenter view on the.
Okay.
On there. I think the solution to this is that you start the PowerPoint and then share Zoom. So if you stop share, stop this as well.
Okay.
Yeah. So if you stop the share, and then and now share again.
Share again.
Yeah. And now you can pick that desktop. Yeah, I think that should do it. And then yeah, and then if you want to hide this, just so it's not you can if you want to hide this just so it's not in your way, you get more, and then yeah, hide this one, hide floating meeting panel. Oh no, sorry, here, and then it's hide floating. Yeah, exactly. Okay, I think that is all good.
All right, everyone, let's welcome our next speaker, Palash Sethi from the University of Florida.
Hi everyone, I'm Palash, a graduate student in the Zao Lab in the Department of Biology at the University of Florida. And I'm really excited to present our work on learning gene interactions and functional landscape from entire bacterial proteomes. using transformer-based models. There have been major advances in genome foundation model in the recent years. For example, EVO2 and high-end DNA. The majority of genome foundation models work at single nucleotide tokenizations, such as EVO2 or high-end DNA. There is another less popular category of models that work at the gene level, such as GNM. These models are trained on segments of the genome or metagenomes and can provide local contextual embeddings for the individual genes. These genome foundation models have been shown to be powerful for various tasks, such as variant effect prediction. On the other hand, if we zoom out and look at the whole genome, these models also have extremely local context. For example, the largest context window length these models have is up to 1 million, which spans about 1% of a typical chromosome and contains an average of just a few genes. As a result, with these models, we are confined to learning only very local interactions. So our goal is to build a foundation model trained on the level of whole genomes in order to learn long-range gene interactions and maybe to provide better functional embeddings for the whole organism based on its genomic content. These functional embeddings could then be used to perform tasks such as predicting organism-level phenotypes. There is also the potential of using these functional embeddings to study interactions between species, which has a long history in functional ecology. To achieve our goal, we turned our attention to bacteria. Compared with eukaryotic genomes, there have been fewer development of foundation models in bacteria. We argue that bacterial genome is a promising direction for whole genome foundation models. First, this is because bacterial genomes are much smaller compared to eukaryotic genomes, with only a few million base pairs and thousands of genes. Second, bacterial genomes are also much simpler and largely consist of coding sequences due to selective pressure to minimize the amount of non-functional DNA. As a result, the proteome of a bacterium still provides a somewhat comprehensive view of its genome, even without the intergenic regions. Third, there is a lot of data available for bacterial genome at both the species level as well as the strain level. For example, databases like RefSeq and Magnify make it very easy to gather that data set, to access that data. Last, bacteria are also relatively easy to phenotype with many standard chemical and morphological assays, and databases such as BacDive provide thousands of phenotypic measurements over 20,000 species. If you are able to make such a model, it would have a lot of applications. Important applications for bacterial proteome foundation model include genome annotation, antibiotic resistance prediction, natural product discovery, and even microbiome studies. While bacterial genomes are a good starting point for whole proteome foundation model, even these relatively small genomes pose challenges. Current models can capture context of, again, around 1 billion nucleotides, but most free-living bacteria have a median genome size of 4 million nucleotides. So even in bacteria, we're still far from modeling the whole genome context using the current models. And that is where our work comes in. Here we present BacPT, the bacteria proteome transformer, developed to capture whole proteome context length. And we applied this model to downstream applications, including identifying local genomic structures such as operons, learning protein-protein interaction at the whole genome scale, and predicting organism-level phenotypes. To talk about the data set, we trained BacPT on 33,000 bacterial proteomes from NCBI. We focus on high-quality RefSeq genomes that are assembled at the chromosome level. Our data set then contains a total of 92 million proteins across 548 families of bacteria. To robustly assess model generalizability, we followed a stringent train test split strategy in an unsupervised manner. We started by clustering all the proteins in our data set using MMCX2 to identify protein families at 50% sequence identity, resulting in about 33,000 protein families. So each genome was then represented by a vector of ones and zeros, denoting the presence or absence of each protein family. Finally, we clustered the genomes using SBB scan based on their binary representations. We selected the distinct cluster containing the genera Escherichia, Shigella, and Salmonella as our test set, which finally contains about 5,200 species. So this is what the model looks like. The input for BacPT consists of protein embeddings for all the genes predicted by the 35 million parameter ESM2 model. The bacterial genome is thus represented by a series of 480-dimensional embedding vectors, each corresponding to a protein in the proteome. These protein embeddings are then passed through the transformer layers. We trained the model using a strategy similar to mass language modeling. Specifically, we introduced progressively stronger noise to randomly chosen proteins and update the model weights using MSE loss between the reconstructed embedding and the original unmasked embeddings. So this is not like the typical language modeling task, which is trained on cross entropy, since we do not work at vocabulary, but instead we work at the embedding level. So this architecture is just based on Roberta with rotary position embeddings. It has 10 layers, 5 attention heads, has about 50 million parameters, has 5,000 context length, and it was trained for 1100 epochs over 12 days on 16 NVIDIA 800 GPUs. After training the model, we looked at a few metrics to assess the model performance. We first computed the mean Pearson R-square between predicted and the original embedding for randomly masked proteins on test genomes. We find that masked R-square steadily improves over epochs, so the model trains. We also observe that R-square for unmasked proteins decreases and plateaus pretty early as a result of contextualization. We also assess our model's performance by calculating how frequently the predicted embedding aligned with the original embedding of the masked gene instead of other background genes within the same genome. And we do this on both the local context level as well as the whole genome level. We found that our model has overall high accuracy at predicting the masked protein, beating GLM, the model which I showed in the beginning. We beat GLM on both the local context level as well as the whole genome level. We also see that when using the whole proteome context instead of the local context level context, the accuracy metric increases from 63 to 78. Next, to assess how well our model can predict the family of masked proteins, we bin all the proteins in our training set into 10,000 clusters based on the DSM2 embeddings. We saw that at a stringent level of cluster assignment, our model correctly predicts the gene family for masked proteins over 70% of the time. As the assignment level becomes less stringent, the accuracy goes down as expected, but still remains over 50%. We next assess the relative importance of local versus global genome context. To do this, we calculated the prediction mean squared error for a single masked protein while increasing the proportion of the genome visible to the model. We first found that overall, the local context seems to be more important than the global context, as the MSE systematically decreases when the visible portion of the genome is local, then randomly sampled across the genome. Yeah, we compared it across the random unmasking of the whole genome. We also observed that the MSE loss decreases at a constant rate as higher fraction of the genome becomes visible. This suggests that although our model preferentially uses local gene content information, it also leverages the whole proteome for generating contextual embedding for the individual genes. Finally, we assess if our model can differentiate between real bacterial proteomes from chimeric genomes generated by joining two randomly chosen genomes. To do this, we trained a linear probe on the mean embeddings across all proteins and benchmark against a bag of genes models using ESM embeddings. We see that BACPD has good prediction accuracy, whereas the ESM-based model essentially can only make random prediction because it is a bag of gene model at the end of the day. This means our model has learned gene context across the whole proteome and may be useful for downstream tasks such as metagenome assembly as well. After training, we test BACPD on multiple downstream applications. These applications span a variety of genomic resolutions, from operons to long-range protein-protein interactions to whole protein representations for complex trait prediction. We first set out to test if BACPD can identify gene clusters. Specifically, we focus on operons, which are highly conserved DNA segments containing clusters of adjacent genes that are co-regulated and co-transcribed. We hypothesize that BACPD may capture contextual representations that help distinguish operonic regions from non-operonic regions, as operons are conserved and exhibit distinct sequence patterns and functional associations. We trained a linear probe to classify BACPD embeddings into operons and non-operons, and see that BACPD is better than the ENCODE ESM2 embedding, as it captures genomic structure due to contextualization. Our findings also suggest that certain gene families are likely overrepresented in operons, thereby allowing the ESM-based model to make non-trivial predictions. We earlier saw that the model learned local gene interaction in predicted operons. Now we are scaling that up to assess protein-protein interaction across the full E. coli K12 proteome. To do this, we developed a method that summarizes gene-to-gene relationship into a single matrix. The idea was inspired by categorical Jacobian technique using protein language model. In categorical Jacobian, each amino acid in a protein is mutated into 20 alternatives, and the resulting changes in logics are used to build a contact map. That is for protein language models. Since BACPD doesn't use categorical tokens, we adapted this idea. Instead of mutating amino acids, we iteratively mask out each protein in the proteome using a zero vector. We then measure how this perturbation affects the prediction of the remaining proteins in the proteome. By calculating the L2 norm of the delta of the change, we construct a matrix that captures how each protein influences the rest of the proteins. We then compared this matrix called Jacobian matrix to different interaction scores from the string database. We see that Jacobian is highly correlated with neighborhood-based interactions. This interaction score was followed by fusion and co-occurrence scores. We also found that Jacobian has a moderate correlation of about R² 0.15 with gold standard experimentally validated PPIs. This suggests that BACPD captures both genomic proximity and evolutionary signals, which aligns with our earlier finding. While BACPD can model entire proteome, it tends to rely more on the local context. This Jacobian-based approach also gives us a highly scalable, unsupervised way to infer proteome-wide PPI interactions in bacteria. Functional characterization of bacteria at the organism level, based on genomic data, has significant applications, including phenotypic prediction and the study of microbial community interactions. Knowing that BACPD has knowledge of operons and protein-protein interactions, we wanted to check whether BACPD could leverage this information to enable enhanced prediction of organism-level traits using its functionally rich embeddings. To test this, we curated a genotype-phenotype dataset using BACTEL, spanning a broad range of functional and phenotypic characteristics, including enzyme activity, metabolite production, metabolite utilization, and even morphological properties. We use this dataset to train a linear probe on the mean BACPD embeddings generated from multiple layers of the BACPD model, and then we compared it against the mean ASM2 embeddings of the whole proteome. We see that BACPD embedding almost always increases the prediction performance. Out of the 30 traits we also benchmarked it against a model called Kratar, which is a PFAM-based model. We see that in BACPD outperforms Kratar in 22 traits out of the 30 traits, and achieves comparable results in 5 traits out of those 30. We also observed an average 5% improvement in F1 score with BACPD embeddings. And in some traits such as lipase, we have a delta F1 score of like 20% as well. We also benchmarked the trait prediction task against a common approach in bacterial trait prediction, which encodes the whole proteome as a vector of presence-absence of the PFAM domains of the constituent proteins in the proteome. To ensure the benchmarking is fair, we convert our proteome embeddings in the original gene order to structured embedding by replacing the PFAM presence-absence status with the context average embedding of the corresponding protein. We observed that BACPD's structured embedding outperformed the PFAM presence-absence method in almost all the traits. We are generating more results as of now, and I'm pretty sure we will beat PFAM and ASM on all the traits. To conclude, whole proteome bacterial embeddings using BACPD could capture whole proteome context length. It can also identify higher order genomic structures such as operons. It learns protein-protein interactions and predicts organism level complex traits. In the future, we want to extend this work on finding more gene clusters such as spliced gene clusters which predict natural products. We also want to use the function embedding for modeling the microbial interaction. We also want to use the GP maps, the complex trait maps for producing attribution maps that can help us in finding the causal genes for that particular complex trait. I would like to thank my lab mates at the Zaula and the funding in NIH and the biodiversity institute. Thank you. Thanks,
We're going to take questions.
Good work. Something about the performance metric 4. Yeah, so this was showing that predictions were improving when you saw more and more context. I was wondering, what does that actually look like, and how much context do you need to improve, and when does it plateau?
So as you can see in the figure, we don't see a plateau until the whole genome is visible. So at a fraction of one, that fractions, it represents the amount of fraction of the genome visible around the focal protein. So what we do is we mask a particular focal protein and then make the remaining context of the genome available to backreading. And as we keep on unmasking the whole genome, we see that the MSC decreases. That is why we claim that as more genomic context is involved around the focal protein, the model predicts better embeddings.
Well, what's the difference here between a local and random?
Yes. So, for example, if we on the x-axis, if we are showing around 25% of the whole genome around the focal protein, we wanted to compare it against random unmasking of 25% of the genome across the whole genome. And that kind of shows us that the local context around the focal protein is important to predict the embedding as compared to randomly unmasking 25% of the genome.
Thanks.
Great talk. I have kind of two questions. One is that you mentioned that you did a genotype phenotype map kind of thing. So is it like associated with the metagenomes, or is it something only for back PT to like take as an input or something?
Right. So we don't work with metagenome specifically. We work at the whole proteome level. So for the genotype phenotype map, the trait prediction task, we use a data set called BACTIVE, which has multiple phenotypes for the whole genome, for the whole genome of bacteria. So we don't work on like local metagenomic context, but we look at the whole proteome for using our model to predict the complex trait.
Got it. And the second question is that you mentioned in the future work you're thinking about BTCs. So can you like expand a bit on that?
Well, we have not started working on it, but biosynthetic gene clusters are specific cluster in the genomic region, in the whole genome that produces natural products, right? We are trying to come up with a metric that kind of looks at the whole genome, and for each protein, we have a metric associated with it. We are still coming up with it, so it's a future work. But our hypothesis is, if we are able to come up with a metric that tells how clustered together those genes are, how conserved they are across multiple genomes, maybe that could give us a metric for predicting whether these gene clusters are conserved together, and could be biosynthetic gene clusters.
There are kind of tools which kind of gives you the structure of that, like which genes are clustering together to form the BDCs.
Right.
But yeah, I'm just trying to understand like how exactly.
So the current rules, for example, like anti-smash and stuff,
Yeah.
they work on rule-based logic. There are specific rules that they use to identify particular BTC classes. We don't want to do that in a supervised manner for our model. We want in an unsupervised manner, could the model predict the clusters of genes and what kind of natural product it could be. If that makes sense.
Okay. Thank you.
Great, I have a question actually. On your slide about predicting whole organism phenotypes, I was wondering if you could speculate a little bit. Are some of these phenotypes harder than others to predict? So it seems like maybe some of these could be predicted just by the presence of one or two genes in the genome. Is that the case? Maybe your model does extra well on those that actually require a lot of context to predict.
Yes, exactly. So I'll use the second slide right now. So this is also similar to the first item that I showed. So we are comparing it against a PFAM model that just looks at the presence and absence of a PFAM domain in the proteome. We see that adding more contextualized information instead of presence absence of the PFAM increases the trait prediction performance. So I'm sure there are traits which require contextualized information, contextualized at the whole genome level to predict the trait as compared to just like presence or absence of a particular protein in the proteome. But we could maybe stratify according to like, what kind of traits are predicted better and what require contextualized information.
Great, thank you. Any other questions? Okay, let's thank our speaker.
And that's the first one, yeah. Right to begin. Let me start sharing. Okay, should all be good.
Stand up, you have two left. Our final speaker in this session is Jishnu Das from the University of Pittsburgh.
Good afternoon, everyone. I'm Jishnu. I'm an assistant professor at the University of Pittsburgh. I run a computational systems immunology lab, and we use machine learning and network approaches to study immunomodulation. So we work on both infectious and autoimmune disease, and the center of our universe is immunomodulation, how through the lens of machine learning and networks you can study these immunomodulatory processes in disease contexts. And before I jump into swing, which I'll be talking about today, I just wanted to talk about the fact that interpretable machine learning is really, really important. That's kind of the core of my research program. In Swing, I'll talk about how we are trying to make it interpretable and biologically motivated, but my lab has done other work too, where we really try to move beyond prediction to inference using high-dimensional multi-omic data sets. So I think a lot of the talks focused on interpretability and design aspects, and that really resonates with what we do. So now when we think about perturbations to cellular networks, the way I visualize it is there is really two broad levels. There is the variome, genetic perturbations, and there is the virome, pathogen perturbations on these cellular networks. And these perturbations really help explain what are the molecular phenotypes, alterations in the networks that lead to disease. And that is basically what led us to develop Swing. When you have genetic perturbations, there are very, very good variant effect predictor tools. For example, AlphaMissense does really, really well for coding mutations. But what are the edges or interactions in a network that are perturbed? Can we really get to these molecular phenotypes? So we developed this method to identify the impact of genetic variation on networks at a systems scale. So this is sliding window interaction grammar. If you're interested, this was published in Nature Methods about a month ago, and it's really a generalized interaction language model for peptide and protein interactions. So I'll get into what I mean by ILM or interaction language models. And this was work co-led by three amazing graduate students in my lab, Jane, Alyssa, and Prabal, and my partner in crime for this was Alok. So this audience needs no introduction to protein language models. Obviously, we know PLMs are awesome, and they can do many different tasks that we did not think were possible even a few years back. However, as powerful as they are, the fundamental premise is that biological interactions cannot be fully captured by passively using PLM embeddings. So we came up with an architecture where we encode the actual interaction itself rather than passively using PLM embeddings. And for the first part of the talk, I'll talk about what we have done with this architecture, but at the end, I'll show you actually what we miss when we passively use PLM embeddings. So the idea behind Swing is very simple. Exactly as I said, we are trying to represent the interaction itself. So this is a standard representation learning approach. And here in this schematic, you can have a peptide and a protein or two full-length proteins. What we are doing is, let's say you have a peptide and a protein, you slide the peptide along the full-length protein, and you are representing this using a biochemical property such as polarity. You could use any other biochemical property too, it doesn't really matter. So you line these up, so the peptide sequence has the amino acids, the target sequence has the amino acids, each amino acid has a number, and you calculate the difference. And then this is the delta biochemical metric, and you then come up with a k-mer list, which we embed using a doc2vec model. The size of the window, the size of the k-mers, all of these are tunable hyperparameters, but the fundamental idea is that this is an unsupervised representation of the interaction itself. It's not passively using PLM embeddings, it's not joining the proteins end to end, it's really looking at this paired representation. And then once we have this vector representation of the interaction, we now use classifiers for downstream tasks, and we ended up using XGBoost. It really doesn't matter. You can even use logistic regression. It almost works as well. The key is in this representation, biologically inspired representation of the interaction. And of course, the reviewer has asked us what would happen if you use a transformer. The answer is not too much better. Doc2Vec is almost good enough. And in fact, it's better because for some of the tasks, we don't have enough samples to actually meaningfully train a transformer. So the first thing, being immunologists, that we focused on is peptide MHC binding. This is, of course, there were other people who talked about this. This is a very well-studied and important problem, critical. So you have PMHC TCR and then downstream signaling. So this is a very well-studied and important problem. There are many methods that are good at doing this. This is just what we started with. So we first tried to learn the language of class 1 peptide MHC interactions. And what most methods do is they take all except 1 or 2 alleles and then evaluate how good the model is on held out alleles, methods like NetMHC PAN, MixMHC2-PRED. We said if we are really learning something generalizable, we should be able to train only on a small subset. So for class 1 PMHC interactions, we were able to train only using 8 human alleles. This is immunopeptidomics data, publicly available immunopeptidomics data from IEDB. And then when we test both in a cross-validation setting, but more importantly on completely unseen alleles, and we have a large number of completely unseen alleles because we trained only using 8. This number 8, we optimized, we really saw that we don't get a gain in performance when you move beyond 8. So we are able to do well. The same with class 2 PMHC interactions. We train only using 5 and can predict very well on unseen alleles. But there are many methods at really predicting these PMHC. So this is just the tip of the iceberg. What other more difficult tasks can we do? What really do we learn from this representation learning approach? And the first thing that we asked was, do we learn biologically meaningful relationships? So we did a computational ablation experiments for class 2 PMHC binding. The peptide lengths on average are between 14 to 20, but the overall length distribution is from 12 to 25. But it's well known that there is a core binding motif of length about 10. And when we did this computational ablation experiment where you start deleting amino acids, we see that there is almost no drop in performance till you get to about 10 amino acids. And then the model performance starts crashing. So what this basically means is that we are learning this core sequence de novo. We are not explicitly encoding it. Many methods explicitly encode it. So this is something that we are learning that's biologically meaningful, in line with expectation. But then we said, now let's make it more difficult. Can we generalize the language of peptide MHC interactions? And class 1 PMHC binding and class 2 PMHC binding are fundamentally, structurally, and sterically different. One is capped at one end, much smaller peptides, 8 to 12 amino acids. Class 2 is open at both ends, and the peptide lengths are much longer, 14 to 25 amino acids. We said, if we are really learning something generalizable, we should be able to train on class 1 and predict for class 2. This is a task that is considered so challenging that no method, to the best of our knowledge, has attempted this. However, a model trained only using 8 class 1 alleles remained predictive of class 2 peptide MHC binding, suggesting that we were really learning something generalizable in terms of the interaction. This was good. So just to recap, existing methods we do as well, like NetMHC PAN or MixMHC2-PRED on class 2, but we can go from class 1 to class 2 that no method does. And in fact, we said, well, if it's really agnostic to these structural and steric properties, we should also be able to construct a mixed model. 8 class 1 and 2 class 2, we mix them, and that still remains predictive. But what is the real goal of these models? The real goal is to predict on rare disease alleles. If we predict on common ones, that most methods can do. And to really test this, what we said is we now train a model on all the publicly available human data, but now can we predict for murine alleles in murine models of lupus and type 1 diabetes, things that the model has absolutely never seen. And then we do experimental validation to test how good the model is. So we worked with Alok and Mark Shlomchik to do this. And what we see is that a swing model that we trained, and this is a busy slide. So the three colors that you should pay attention to for the swing model are blue, orange, and pink. Blue is the class 1 only model, orange is the class 2 only model, and pink is the mixed model. They are doing well across the length spectrum in terms of predicting peptide MHC binding in completely unseen murine alleles. One H2IEK, this is a murine model of lupus, and the other IAG7 in a murine model of type 1 diabetes. The competing methods, net MHC pan, mix MHC 2 pred, are doing really poorly, and they only predict because there is a clear length bias in the middle of the spectrum. So we are learning things de novo, and we can translate this across organisms. Now we said, let's make the difficulty even harder. So we all know there are these awesome variant effect predictors, alpha missense, ESM1B, Eve, and so on. And they are really, really good at predicting pathogenicity, but they do not predict the disruption of specific protein-protein interactions. So we said, what happens when we now look at experimental data where we know that the wild-type proteins bind to each other, but a single missense mutation, so you're literally changing one letter in the vocabulary, disrupts the interaction. Can we pick this up? And the answer is yes. When we look at Mendelian mutation, population variance across the allele frequency spectrum, this is against experimental data, we are able to pick it up. So again, experimentally, it's been shown that the wild type interacts. Some of the missense mutations disrupt, others are not. We are only restricting it to cases where the wild type and the mutants were tested using the same experimental East2 hybrid platform. So this was really encouraging. And then we said, well, are we just encoding or learning from sequence similarity? And we did a proximity dropout experiment. So we said, now let's embed all of these proteins and then drop the two proximal clusters and see how much the performance goes down. The performance goes down from 0.8 something to 0.7 something, but we still have signals. So we are learning things well beyond sequence similarity. And in terms of outperforming state-of-the-art variant effect prediction methods, of course, these methods never claim that they can be used to predict the disruption of specific interactions, but we just want to make the point that in terms of really predicting these molecular phenotypes, swing outperforms ESM1B, alpha missense, or Eve. And because of this, often things that are predicted by alpha missense as benign, known to be actually deleterious. These are just two examples where we pick up corresponding molecular phenotypes, the disruption of these specific interactions. So we are really good at predicting the impact of genetic variation. And this is well known, of course, that when you change one amino acid, AlphaFold 3 cannot really pick up this. We are picking this up using sequence alone. So for the final part, I just want to talk about why we use this ILM approach. What would happen if we used PLM embeddings, biochemically informed embeddings? And there are these four conceptual bins. One is you could take just a foundation model derived PLM embedding. You could also take a biochemically informed embedding, the way we talked about it, and then you could passively use them. You could concatenate the embeddings of the two proteins, or you could embed the two proteins, take the vector sum, and play all of these games. So that's the first two bins. And then you have the ILM encoding, the representation, and then the actual embedding using the doc2vec model. And then you have these single context prediction tasks and multi-context prediction tasks. So for single context prediction tasks, Swing does as well as the best passive use of PLM embedding. The best passive use of PLM embedding depends on what case. This is the interaction disruption task. For the peptide MHC, it's different. But when we now go to these multi-context prediction tasks, going from class 1 to class 2, you see that all of these have essentially no signal, whereas Swing is able to generalize. This is going from class 1 to class 2, same for human to mouse. Again, this is the only approach that has signal. So it's picking up something generalizable, something that's translating across these tasks. And that is where the value of this biologically inspired interaction language model architecture comes in. So in summary, it's a first-in-class interaction language model that learns the language of protein-peptide interactions across contexts using only sequence. It's a few-shot model to accurately predict PMHC binding across HLA classes and species, can make accurate de novo predictions. It learns biologically meaningful information without prior knowledge, can predict the impact of missense mutations using sequence alone, and outperforms passive uses of PLM embeddings. There's a lot more that we can do. We are really excited about this ILM get structure, we can hopefully move beyond proteins, but that's work in progress right now. I get to talk about all of this science. It's really my lab that does all the cool work. So very grateful to have an amazing group of people. And before I end, I just wanted to say that we are recruiting for a faculty position at Pitt. Rachel and I are co-leading, co-sharing this search. So if you're interested, Pittsburgh is a great place to work. You get awesome students like the people on this project, from the CPCB program. If you're interested, please apply. The ad will go live very shortly. And then I would be remiss if I didn't thank our funders, as well as the organizers, for giving me the opportunity to speak, and all of you for your attention. Thank you.
We have time for several questions.
So you mentioned with the ILM, there can be different biochemical properties that you use. So are you usually just using, I miss whether you're usually using one, or you can use sort of a whole bank of different biochemical properties? And does that, I guess it depends on the task with what's most effective.
Yeah, that's a great question. So right now, in the examples that I showed you, we only used polarity. We also tried switching it to hydrophobicity, and the performance was about the same. However, you could imagine that and people asked us, oh, why not combine these? We said, well, it works already pretty well, but absolutely for harder downstream tasks, we are now working on a way in which we can have multiple scales, multiple properties, not just one, where they may be orthogonal.
Another details question. I was wondering if you could tell us a little bit about the training data for the more general protein-protein interaction model. Do you need proteins known to interact and their crystal structure, or?
Yeah, so that's a good question. The data set that we used was all these are E2 hybrid experiments where the wild type proteins were known to interact, and then the mutation may or may not affect. So this is experimental data. All that we need is the sequence. There is no structure at all anywhere in the method. So it doesn't matter if there are crystal structures, AlphaFold models, it really doesn't matter. But we did this sequence only in the interest of generalizability. There are a lot of cases now where you can infuse structure, like structurally infused language model embeddings. We are looking into that to see whether that can improve prediction. But as of now, this is only sequence. We don't need any structure.
Another one in the front.
Thank you for your talk. So in one of the results you shared, you reported the performance for different peptide size, and they have the variance performance results. I was wondering, what's the rationale of grouping the results based on the peptide size?
Yeah.
Instead of based on same peptides or non-same peptides. Yeah.
Yeah. So perfect. So that's a great question. So these are all by definition unseen peptides because these are de novo immunopeptidomics experiments. So this is a hard test. Like these are experiments that we did after generating the prediction. So neither our method nor NetMHC-PAN nor MixMHC2-PRED has seen these. And this is really the true use case because this is in a disease context. We are able to predict accurately across the length spectrum because in a lot of disease cases, what really matters is this maybe weirdly short or weirdly long peptide being presented, and that is binding this disease risk allele. This is H2IEK risk for lupus, IEG7 risk for type 1 diabetes. The point is, existing methods would do poorly in general, but more importantly, for the short or long, they would do completely terribly. Because we are using this representation learning approach, we are using the sliding window. The length doesn't matter. Whereas existing methods, because they are trained, there's a clear training bias because most of the peptides these methods have seen are in the 14 to 20. That's the average. But for, let's say, 9 or for 23, it's terrible. So that's exactly not reflective of the biases of the training data.
Thank you.
Hi, yeah, it's wonderful to always see sequence modeling models doing surprising things. So I'm wondering, you mentioned going beyond protein-protein interactions, and so one of the largest limitations there if it's going to DNA or RNA or even going to a small molecule and finding pocket predictions is still sort of unsolved. Just any thoughts on this would be nice.
No, thanks, Caleb, for the great question. I think we want to go to nucleotide like DNA, RNA, as well as other small molecules. We're just figuring out what representations we should use, whether it's the same thing, what metric. I mean, there, David's question about combining scales also comes in. With like here, you have 20 amino acids and pairwise differences. We get a perfect, like it's the degeneracy that we have with the polarity scale is just right. But when we move to other things, we may need to rethink the representation more carefully. But I think we believe the same ILM principle would still apply, but that's a great point.
Any other questions? All right, let's thank our speakers for this session.
Thank you.
Great. And we have I will do a 25-minute break and restart at 3:50. Thanks.
Oh, of course. Not a hard job.
Hey, how's it going? Are you presenting?
Yeah.
Yes. Perfect. Yeah, no, I mean, I also didn't get one apparently, so don't worry. Yes. Yeah, did you? Is this stuff? You either need to upload slides or be able to get into the Zoom. You can get into the Zoom, then I think we're good. Yeah, okay. You're registered as a panelist.
Yeah.
Yeah, okay.
Yeah, I got the right email.
You got an email, that's the main thing, yeah, perfect, yeah. Yeah.
Columbia, and so it's like the wrong email.
Oh, yeah, okay, but yeah, no, that's why. Yeah, I mean, the webinar stuff is good because it's basically like you can't get like Zoom bombed. Maybe that's not a thing, maybe that's the pandemic thing, but like, yeah, okay. So like, but you can't with the webinar. The good thing with the webinars, right, is only panelists can do anything. So then that you're sort of safe. The downside is panelists have to be registered or you can't get in. So yeah. 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 Right. Oh yeah, right. Because yeah, you're gonna.
I did actually register seafood.
Yeah. I have this. I mean, I sort of have this also a little bit, but I have like well, I have different. Yeah, I have different. I mean, mine are all Morningside, but there's still like multiple. And then I have this. And I'm right. Yeah. Well, I have this with class, right, where like people don't. Because you don't have you don't have a Google. Yeah. Yeah.
Yes, I can just.
Yes, it should. Fingers crossed. In theory, it should all. In theory, it should all work. And you're next, are you? Yeah. Okay. Yeah. I in retrospect, obviously, I should have emailed everyone beforehand.
Hopefully it's like
Great, yeah, so as long as I'm sure, either upload slides or like be able to log in. I'm sure,
yeah, I am pretty sure it's fine, but maybe that's just.
And then I'll just guide with your.
Yeah, it's definitely the safest thing.
As a.
You are sure, okay. Yes, yeah. Perfect. Yeah, got it. Nice, okay. Sounds good. Are you in the which DeepMind office are you in?
The Seattle
We just need to remember to turn that microphone on whenever you start. I can take this out of your way also. My admin's much better question. That's we'll give people like a minute. Okay. I think there's a couple of people from Ahmed's lab taking my class.
Oh, you're
Yeah.
doing functional genomics.
Right, yeah.
Meeting technically.
Oh no, really?
Great.
No, no. I mean, I do a hybrid, so it's all recorded. So in principle, they can,
We have Zoom as well, so.
yeah. They can just have 2 Zoom meetings going at once. Have the Zoom and the group meeting going at once.
We have bad audio acoustics though, unfortunately.
Yeah, that seems tough. I tried doing it once, being on 2 Zoom meetings. It doesn't work very well. Yeah, like however low energy one of the Zoom meetings is, it's still like
It's not the same.
yeah,
It's not the same.
yeah.
So, but I think they'll go.
Yeah,
I have
upgrades?
I have a master's student who's working with me who's taking it. She was actually
Yeah.
close with a project, so she
Yeah.
might and ask you.
Right. Yes. No, I think she talked to me actually. Yeah, because I was trying to say, like, I'm very happy to have people do whatever project they're doing. If they're working in a lab, do that for the class.
In terms of like how genomics have your
Yeah, I mean,
everything. And
I was trying to tell I've had people do, I mean, the only thing is I don't know this stuff very well. So like, but I mean, I had some on the first year I taught to like some RL for like protein structure, protein folding or something. It didn't really work, but like, I don't know. I mean, usually those people doing that stuff, like, usually if they're doing something a bit outside the scope of the class, it's because they're interested in it and kind of ambitious. So it's usually a good thing. Okay, I think we should probably.
Yeah. Okay.
Yeah, I think we can get. Yeah, I think also if we get going, then there's actually one thing I want to double check is the sound quality.
Very chill.
Is the sound working in the overflow? Because we're about to start. Does the sound working here?
I think it's off. Maybe just press this.
Oh. I must have. This is together.
I don't know.
I think we should get going. I'll okay.
Well, welcome everybody to the.
She's loud. I'm telling you, this is good.
What? Yes, she's louder than me. That's
Yeah,
amazing.
yeah.
So, I'm David Campbell from Cold Spring Harbor Laboratory, and welcome to the last of the oral sessions from the meeting. And our first speaker will be Julia Rogers from Columbia University.
Everyone, so hi, I'm Julia. I'm currently a postdoctoral fellow at Columbia University, and I'm working to decode the biophysical logic of cell signaling to explain and program cellular behaviors. To do so, I'm developing integrative data and physics-based modeling frameworks that directly link molecular variation to emergent cellular behaviors. So what you're currently looking at on the screen is actually a network of protein-protein interactions involved in cell signaling that are all mediated by modular peptide binding domains. However, this network is actually far from complete because it contains interactions needed by only a fraction of known peptide binding domains. So today, I'm going to tell you about how I've developed a machine learning approach that combines statistical mechanical theory with advances in representation learning to universally predict the interaction affinities of domain peptide interactions and ultimately construct a complete and quantitative signaling network. So this network is composed of interactions needed by 8 domain families, and here each node corresponds to a protein and it's colored based on its domain composition. So for example, one node corresponds to group 2, which is an adapter protein that can form a complex with the epidermal growth factor receptor, its sauce, to regulate cell growth and proliferation. Group 2 has an SH2 domain that combine a phosphotyrosine motif on EGFR, and it also has two SH3 domains that combine polyproline regions on SOS. And these are two examples of interactions between modular peptide binding domains, or PBDs, and short linear motifs, or SLIMs. And for this talk, I'm going to use the term SLIM and peptide interchangeably. SH2 and SH3 are two families out of an estimated 100 to 200 in humans, and here I'm showing examples of 24 different families. They combine a wide variety of different types of peptides, including those that are post-translationally modified through phosphorylation, methylation, or acetylation. The number of experimentally measured interactions varies widely across domain family, and I'm indicating the number of experimental measurements based on the size of these circles. While some domain families are very well characterized with over 10,000 measured interactions, others are much more poorly characterized, within the order of only 100 to 1,000 measurements. Across all domain families, though, the number of measured quantitative interaction affinities is much smaller, and I'm indicating that number based on the size of these dark wedges. Yet it's quantitative differences in interaction affinities that enable complex signaling logic. Experimentally measuring domain peptide interaction affinities at scale is challenging due to the weak and transient nature of these. So to overcome this challenge, I developed a machine learning approach for universally predicting domain peptide interaction affinity directly from amino acids. At the molecular level, we can describe domain peptide interactions using statistical mechanics. In solution, a domain and a peptide will exist in an equilibrium between a bound and an unbound state, where the probability of observing the domain bound will depend exponentially on the change in free energy upon binding, where I've taken the unbound state as a reference, and it will also depend on the concentration of peptide in solution, where a standard state ensures proper units. Typically, interaction affinities are quantified by a dissociation constant, or a KD, and I can express the probability of observing the domain bound in terms of KD. Weaker interactions have higher KD and require increased concentration of peptide to achieve saturation. When the peptide concentration equals the KD, then you're equally likely to observe the domain in a bound or an unbound state. And this KD is related to the change in free energy upon binding. While determining an exact functional form for this change in free energy upon binding is effectively impossible, I can learn an empirical approximation from experimental data using machine learning, and in this way, predict interaction affinity directly from amino acid sequence. In my model, I decompose the change in energy upon binding into contributions from individual domain and peptide residues, and also pairs of interacting residues. Additionally, I learn an entropy term to account for the degeneracy of unbound microstates. And each of these three energy terms are then computed using neural networks. So one of the first questions in developing this model was, well, how should these amino acid sequences be encoded within the model in order to compute the change in free energy upon binding? Well, across all domain families, there's some shared interaction motifs at the molecular level. For instance, hydrophobic interactions, electrostatic interactions are going to be present, but they're going to be patterned in different ways. If we map sequences to a shared representation space, then we can potentially capture such similarities and compare interactions both within and across domain families in a biophysically meaningful way. To do this, I've leveraged advances in representation learning, specifically protein language models. And so I first embed protein sequences using a protein language model, and then I use the representations for individual residues to compute the single site energy terms, and combinations of residues representations to compute the pairwise interaction energy terms. The second question in developing these models, how are we going to infer the parameters of these neural network free energy functions from data? Firstly, we can train the model on abundant qualitative interaction data that provides a binary readout indicating if an interaction was observed or not at a fixed peptide concentration. Conclusive binder readouts are indicative of stronger interactions, so train the model using an objective function that penalizes predictions of higher KD value, whereas non-binder readouts are indicative of weaker interactions, so I'll penalize predictions of lower KD value. Domain peptide interaction affinities have also been tuned to ensure the dynamics of cell signaling, so I'll penalize predictions that fall outside of a known physiological range. Alternatively, I could train the model on quantitative affinity data using a negative log likelihood loss to penalize predictions that deviate from the true value. And by using a multitask learning objective, I can train the model simultaneously on both of these tasks. Where here, alpha is a hyperparameter that trades off learning to predict affinities versus classified binders. And by using this multitask learning objective, I'm able to train a model on all available interaction data to construct a quantitative statistical mechanical model, or QSM. And so the movie I just played you is actually from an overfitting experiment. So it's a very accurate predictor of affinity, but we wouldn't always expect this to be the case, which begs the question, when can the predictions be trusted? So in this model, I actually also provided prediction of the absolute error, and to do this, I train a shallow neural network ensemble, where I take the ensemble mean to be a prediction of the binding free energy, and the standard deviation ensemble I take to be a prediction of the absolute error. And so when I train the model of quantitative affinity data, my objective function actually has two terms, one that incentivizes the mean of the ensemble to match the true experimental KD value, and a second term that incentivizes the standard deviation to match the true absolute error. Okay, so do QSM models constructed for individual domain families accurately predict affinities? Here, I'm going to show you predicted KD values in log scale on the x-axis versus the experimental values on the y. A perfect prediction is indicated by the dashed line. In the case of the WW domain family, the predictions are reasonably correlated with the true values, and they're consistently of the correct magnitude. I can construct models for 24 different domain families, and across the board, we see that the affinity predictions are of the correct magnitude, and in a third of the cases, I achieve Pearson correlations above 0.7. And so this really illustrates how by leveraging statistical mechanics to construct a multitask learning objective, I'm able to train a model on all available interaction data to construct a quantitative predictor. Well, how trustworthy are these QSM predictions? In addition to predicting the KD values, the model also predicts its own absolute error, which provides an estimate of model uncertainty. One way we can evaluate these uncertainty estimates is by constructing calibration curves, which plot the empirically observed confidence level versus the model's predicted confidence level. Perfectly calibrated uncertainty estimates would fall on this dashed line, whereas when these curves fall in the bottom corner, it's indicative of the estimates being overconfident, versus in the top corner, they're underconfident. And so often we'll just summarize calibration based on the area between these two curves and quantify a miscalibration area. In the case of these two models, they're actually quite well calibrated, and they both have the same miscalibration error. However, this doesn't necessarily mean that the error predictions themselves are quantitatively accurate. Achieving that is a much harder task. And in fact, while both models predict error estimates that are positively correlated with the true values, they're arguably better for the PTP model than the WDR model. And so I bring this up in part because I think it has implications for using these uncertainty estimates in, say, active learning frameworks for guiding data acquisition, or for Bayesian optimization frameworks for designing frameworks. And so these are just two of the models out of all the 24, consistently see that the error estimates do positively correlate with the true values. And so this is useful to have this estimate in guiding downstream applications of the models. Okay, well, are QSM free energy functions universal? To begin to answer this question, I'm actually going to switch to evaluating the model for its ability to identify interacting versus non-interacting domain peptide pairs, as this allows me to evaluate a much larger and more diverse set of domain and peptide sequences. I'm going to evaluate model performance on this classification task with the area under the receiver operating characteristic curve, and I'm going to plot these AUROC values in a heat map. On the x-axis, I have training domain family, and on the y-axis, I have test domain family. So the diagonal results are results from models constructed for one domain family and evaluated on it. These are quite accurate classifiers, with AUROCs often approaching 1 in many cases. But I'm really curious, if I construct models for one domain family, are they transferable to others? For reference, I'm going to outline domain families that bind the same types of peptides in these colored boxes. And indeed, there are cases where models constructed for one domain family are modestly transferable to another. This happens especially for polyacrylamide binders that are found in this green box. We can also see it outside of these colored boxes, which is really exciting because it suggests that even if it binds a different type of peptide chemistry, the model is learning something that allows it to generalize to other domain families. And so by leveraging a protein language model to map sequences to a shared representation space, I'm able to construct a universal predictor. QSM predicts interactions between domains and peptides. But many signaling proteins are composed of multiple domains or multiple peptides, and this can engage in multivalent protein-protein interactions. Can I use QSM to compute the probability of a bound state ensemble versus the unbound state, and thus identify protein-protein interactions? To evaluate the model on this task, we assembled a set of low-throughput confirmed interactions and evaluate the model's recall in this set as a function of false discovery rate. For comparison, high throughput experimental approaches using affinity purification mass spectrometry are used to hybrid assays recall about 10% of these interactions at a 1% false discovery rate. A model we previously developed recalls twice as many interactions, and QSM recalls 4 times as many multivalent protein-protein interactions, really illustrating its potential for discovering new interactions and constructing a protein scale signaling network. So when I started, I showed you an interaction network that was actually constructed with the model we previously developed and limited to 8 domain families. Now using QSM, I can expand to many more domain families. And so to summarize the development of QSM, by leveraging statistical mechanical theory, I'm able to construct multitask learning objectives that allow me to build a quantitative predictor of binding affinity directly from amino acid sequence. By using an error-aware objective, I'm able to train a shallow neural network ensemble that provides a model estimate of its own uncertainty that can guide the use and deployment of these models for scientific applications. And by leveraging advances in representation learning, specifically protein language models, I'm able to construct models with models transferability. And so I'm now really excited to use QSM to construct truly quantitative models of cell signaling, and then use them to decode the biophysical logic of cellular information processing. So before I end, I want to give you a sense of some of the questions I'm now thinking about answering with these models. Many of these proteins are modular and composed of multiple domains and peptides, and they can adopt an ensemble of different conformations. The prevalence of an active versus an inactive conformation is often regulated in part by intramolecular domain peptide interactions. And I can use QSM to compute the probability of these different conformations, where each conformation is distinguished by a different set of intramolecular domain peptide interactions. What's exciting about this is that I can do this at proteome scale. And so this then allows me to ask questions such as, how is protein conformational plasticity tuned for signaling logic? Even just with these very simple models, I can compute measure conformational plasticity based on the Shannon entropy of their distributions, and these estimates actually match some of these proteins' known functions. So for instance, among low plasticity proteins are kinases that are known to act as digital switches. They exist predominantly in a single inactive state until an interactor switches them into an active state. Among high plasticity proteins or adapter proteins, they adopt an ensemble of many conformations with roughly equal probability, such that each of their binding sites is transiently exposed and they can form effective hubs. This then begs the question, how do different interactors shift these conformational ensembles? In other words, what are the multi-input computations performed by these different modular proteins? At the level of domain peptide interactions, I'm interested in using QSM to assess how sequence variation tunes domain peptide interaction affinity through in silico deep mutational scanning. And I'm also now using the models to ask the question, how specific or promiscuous are different domains across the proteome? How is domain specificity and promiscuity balanced to ensure that you have specific responses to different stimuli, while also enabling pathway crosstalk to calibrate cellular scale responses? And so with that, though, I'm going to end by acknowledging first my postdoc advisor, Mohammed El-Qureshi, a number of students, both past and present, who really contributed to enormous data curation effort to curate the literature to construct data sets for training these models. Students who've also worked on benchmarking general purpose protein peptide interaction predictors on these data sets. Ashley is also actually working on me to develop a deep learning model for predicting the conformational ensembles of multi-domain proteins at atomistic resolution. And Dave has been working with me on a GPCR specific model. Thank you all for your attention, and I'd be happy to take any questions.
Yeah, so we have plenty of time for questions.
So, hi, great talk. So I'm curious about whether you are interested in or planning to generalize this to model protein interaction with other kinds of ligands that are not peptides.
They're not peptides. So
it's a harder problem.
It is a harder problem. I mean, we already heard some great comments about this in some of the earlier talks, right? And how potentially you can leverage these different kind of modalities, be it say small molecule and peptide, to hopefully learn some general physical chemical principles that generalizes across peptides and small molecules. So actually, in the case of the GPCRs, a lot of the data we pulled from is coming from like Campbell and Drug Bank. turns out because they are a therapeutic target, there's been a lot of work developing, say, support peptides or modified peptides. And so this is a natural case where we're interested in doing a multimodal model. We haven't done it yet, but it is in the works. I think how we, embed these different modalities is going to be important. How we source the data is going to be important. And I think it's hopefully going to be a useful test case for then making a more general purpose model. So definitely something we're thinking about, but don't have anything specific yet.
So doing that one.
Yeah, go ahead.
Yeah, thank you. Yeah, really fantastic work. I have one, I think, technical question and one sort of bigger picture question. So the technical question was more on the, you were showing a lot of stuff, so I missed with the actual model architecture, and you were focused on the kind of the thing you're predicting side, right? Kind of the likelihood. So the architecture itself, I'd like to understand a bit more of that, whether it's not using any structure, I think, but I wanted to understand that. And then the more big picture question is, obviously cells themselves have structure, right? We have all the old school organelles surrounded by their lipid membranes, and now we know about like condensates and other cellular organization. So how you would think about incorporating that, because the which proteins get the chance to interact with each other is determined by that stuff.
These are great questions. So you're right, I glossed over the architecture because most of the innovation, even I've really incorporated kind of physics is actually in the objective functioning. The model is actually very simple. They're just, so there are 3 MLPs. I think one key difference between a number of models is the fact that this is a residue-based model, as opposed to there's no sort of aggregation. I think that's useful, especially with these small, so maybe this is kind of the best slide. They are literally MLPs. We do actually also, so I glossed over, there's post-translational modifications that are key to regulating a lot of these interactions. These are not explicitly captured by language models, and so there is an upstream encoding of these protein language models. It's actually a residual network, so it provides a residual update to the language model embeddings. I think there's room for innovation on the architectural side. So here we just concatenate embeddings to these pairwise interaction terms. You can imagine starting to use attention or cross-attention mechanisms. This already worked quite well, I think, because we actually do have a fair bit of data in aggregate. And so that's why we haven't done that there. But again, I think there's lots of room to innovate here as well. It's not structure-based whatsoever. There's a couple of reasons for that. One, because many of these interactions, they are transient and weak. It's not clear that there is even a single structure. So you can bind peptides in different orientations. You can have different modes of binding. So these are typically order of, say, 10 micromolar is actually like the sweet spot for signaling. There's not necessarily structures available, and I don't think it's necessarily the right way to think about a lot of these interactions. Also, to apply this acronym scale, having a sequence-based model is much faster.
Sure.
And so that's part of it. But you can actually, what's nice is, I'm not going to say that these are the interaction energies between residues, but the model has learned something, and so you can map these onto structures and sort of ask what has it learned. Your second question about localization, this definitely plays a role. And it's something that I do have some future ideas for how we might start to leverage these models, but then incorporate kind of the constraint of the physical localization. I'm not sure it's going to be explicitly included in this model, but it's something we could think about. I really view this as generating what is biophysically possible.
Yeah.
And then we can take, say, information about, even if it's estimated protein abundances, because it's quantitative, we can start to say, how does this shift the different complexes that you form in different contexts? How does, say, localization influence it and whatnot? But this is just really the starting point for that. So a lot more to do there.
Okay, great. Well, with that, let's thank our speaker again.
I'm doing PDF, which is the safest of all the options.
Figured it would be.
Yeah. Okay, should be good, I think.
Awesome. Thank you. Okay, perfect. So our next speaker is Peter Shaw from Google.
Hi, everybody. I'm Pete Shaw. I'm a research scientist at Google DeepMind. Thanks everyone who stuck around for the last oral session of the day here. So I'm going to present this work on behalf of my fantastic colleagues listed on the slide here, and talk to you about why, if you're working on a task like protein function prediction or a related task, you should consider retrieval augmented models. Nice. Okay. All right, so what is protein function prediction, and why do we want to do it? I guess many of you are probably already familiar and maybe already think this is important. If not, let me briefly try to convince you. So given a protein sequence, we want to predict its biological function. And function, of course, can be represented in many different ways. In this paper, we stick to these three categorical labels from different ontologies. So enzyme commission numbers, gene ontology terms, and PFAM families. So a few things that are important to note here. So more than 100,000 sequences a day are added to various online databases, and far less than 1% of these have been experimentally characterized. So some of these uncharacterized proteins could, address important problems in chemistry, medicine, biology. So it's important to build computational tools that can help predict the function of some of these uncharacterized proteins. And I'll say one thing that was exciting for me in this collaboration was a bunch of my colleagues on this project at Google have actually shipped some of these functional annotations to fill in gaps in these protein databases. So from a machine learning perspective, what are the key challenges here? So we kind of have what looks like a straightforward sequence classification task, but we have these two really key challenges that make it hard. So the first is kind of out of distribution generalization in the sequence space. So oftentimes we're interested in predicting the function of a sequence that might look pretty dissimilar to any sequence that we've characterized in the past. And then the second challenge is more on the output space. So, sometimes we have these rare functional labels, new functional labels are being added to these ontologies. So we have kind of a few shot learning challenge in some cases. So these are going to be two kind of running themes through the presentation here that I'll call back to. And sorry, let me first briefly also give you a admittedly slightly simplified overview of kind of prior work in this area, which falls into kind of two broad categories largely. So the first category of methods are based on similarity-based search. So in this case, we have this protein sequence, and we have a database of sequences with annotated function. So for a similarity-based search approach, we find similar sequences using, a tool like BLAST, which is going to compute these local alignment scores between our query sequence and the database sequences. And then we get a set of similar annotated sequences, and we use some sort of heuristic, like assuming that our query sequence should have the same labels as the most similar annotated sequence. And this is great. These techniques have been widely used for decades. They really capture a lot of domain knowledge and can be quite effective in some cases. So more recently, of course, there's been a lot of interest in neural models for this task. So here we're kind of using really the same information, just in a different way. So we're going to tune a neural model, get a set of neural model parameters that captures this information about the sequence to function relationship. And then to make predictions, we're just going to put this sequence, or sometimes with additional information, into the neural model input and get a predicted set of labels. This is, of course, really conceptually appealing. These highly nonlinear models can capture, at least in principle, can capture really complex and subtle patterns related to function that maybe tools like BLAST don't. But there's an interesting sort of empirical fact, and we aren't the first folks to kind of notice this, but BLAST remains a very strong baseline compared to a lot of these neural models on these functional annotation tasks. This really kind of was one major motivation for this work, was thinking about how do we combine aspects of both of these types of approaches to improve the accuracy and robustness of protein function prediction. All right, so the methods that we explored are largely inspired by two techniques that have worked really well in the natural language processing space. So these are retrieval augmented models and in-context learning. So there's a lot of prior work in the language domain about how retrieval augmented models can better capture tail knowledge. They sort of remove this sort of bottleneck in needing to represent all of the knowledge necessary to do a task fully in the model's parameters. And there also can be useful for few-shot learning. So this in-context learning, where you condition on a few examples of the task you're trying to do, and the model at inference time can learn to perform this task. All right, so here's an overview of the system that we built. Let me just walk through this briefly. So given a protein sequence and a candidate label, we retrieve exemplars with annotated function, and we retrieve exemplars both with and without this candidate label. Then we, in our case, just concatenate all of this information and put it into a transformer model. Then we ask to make a binary prediction for whether this candidate label should apply to the query sequence. Again, a few things to note. So we retrieve these exemplar sequences conditioned on both the query sequence and a candidate label. So this does mean at inference time, we need to make separate predictions for each candidate label of interest. But it has this kind of major benefit that we can select the most relevant exemplar sequences for learning this specific class boundary that we're trying to classify. So it's kind of like a meta-learning setup where what the model actually learns is to infer the class boundary from this provided context. And this is a bit different than prior work. There's lots of prior work on retrieving unlabeled sequences or multi-sequence alignments for the purposes of learning better protein representations. So this kind of in-context learning setup drives a few different design considerations, particularly around the training recipe for these models, which I'll talk about next. So we have two stages of training. I'm actually going to talk about step 2 first, since it kind of motivates some of the pre-training changes that we implemented. So for fine-tuning, basically we want to construct training examples that look like what the model sees at inference time. However, there's one notable difference. So at inference time, we try to find the closest exemplars to the query sequence, assuming that these are going to provide the most relevant information to make the classification decision. But at training time, we sometimes bias this sampling to select less relevant exemplar sequences. This is, again, related to that key challenge that we want to be able to generalize even in cases where the exemplar sequences are dissimilar from the query sequence. So with this technique, we can improve the model's out of distribution generalization capability by kind of making the training time look a little bit more like the scenarios that we're going to see at inference time in a non-IAD evaluation setup. And now to talk about pre-training, I guess a now sort of standard method is to use these masked language modeling approaches to pre-train these protein sequence models. A fairly standard setup from prior work is to pre-train on a single sequence at pre-training time. Obviously, in our case, during fine tuning, we're going to want the model to be able to compare, multiple sequences. So we incorporate that into the pre-training recipe as well, and we see this gives a bit of a boost. So we're going to sample a sequence pair with higher than random similarity and ask the model to jointly predict masked residues across both sequences, as well as predict a similarity score, which can be deterministically computed between these two sequences. So, pre-training in this recipe kind of encourages the model to not only learn everything it would learn from pre-training over a single sequence, but also to kind of implicitly align and compare these multiple sequences. All right, so that's basically our method. Very simple generic model, and then this training recipe that teaches it to effectively use this additional retrieved context that we're going to give it at inference time. So let's see, I guess, how well this all works. The key question for us was really, does providing this additional context help the model? And also, of course, how does this compare with other prior work, keeping in mind those kind of two challenge cases that I mentioned earlier. So we took 9 benchmark tasks across EC-Go and PFAM prediction. If you can't read the small text, the task metrics vary here. There's some function of precision and recall. Higher is better. And the blue bars are our approach. ProText in the green is the best previously published neural model for each task. So we see that ProText outperforms these strong baselines. And what's particularly interesting is the 3 splits on the left are random IID splits, but these 6 on the right are non-IID splits. So these are splits where the train test split has been designed to restrict the sequence similarity in the test set. So these are cases where the test sequences are dissimilar from those available for training the model. So of course, performance is maybe generally lower on these settings for ProText, but it's even lower for the other approaches. So I think this is notable. One question people might have is whether this retrieval augmented setting is still useful, even if the exemplars are dissimilar from the query sequence. And, empirically, this is actually where we see it's most helpful. Here's comparison to a BLAST baseline, which is a surprisingly strong baseline. In many cases, these were previously published from prior work, but in some cases, we reported a stronger BLAST baseline than was previously available. So just, I don't know, kind of another secondary finding is just how strong BLAST remains. And here's our ablation where we build a version of that same transformer model, just as a more standard sequence model without any exemplar sequences or retrieval augmentation. So this really helps us isolate the contribution, the core contribution of retrieving on exemplars, and we see performance dropping quite considerably. So I think this is nice. One might hope that some of the other advances from prior work, looking at incorporating structural features, text-based information, might even stack with some of these retrieval augmented methods, and that would be interesting to explore for future work for sure. Now to look at cases of rare functional labels. Again, we see the improvements are largest when fewer training examples are available. So this is the performance across functional labels binned by kind of how many examples for that particular, in this case, PFAM label were available in the training set. And we even see that it can generalize to labels that were held out during fine tuning. So this really reinforces the idea that the model is really doing some form of in-context learning to understand the class boundary of interest. All right, so to wrap up, I think the key contribution is, of course, that retrieving and conditioning on relevant annotated proteins can improve accuracy and robustness of function prediction. I guess, it's kind of a buzzword now, but like agents with tools are an increasingly popular instance of inference time retrieval. So that could be interesting to explore. And then I guess just to end on a question, it's interesting to think about what other types of information could be useful to retrieve at inference time, either for protein function prediction or other related tasks. So I will end there. Thanks again to my colleagues on this project, and hopefully we have time for questions.
Yeah. We have plenty of time for questions, yeah.
Thank you so much for the talk. I was curious how the mix of positive versus negative exemplars affected the results, and if you ablate just the positive or just the negative.
Yes, so we don't have those ablations in the paper, but I can talk to you some of our initial experiments and general trends. So a majority of the experiments in the paper just simply use two positive and two exemplars with the class label and two without. For the PFAM experiment specifically, because PFAM families are mutually exclusive for a given subsequence we were classifying, there we actually included only 4 positive, which was sufficient. In general, we're using a dense transformer model, so scaling beyond 4 sequences starts to become a little problematic just from the amount of compute you need. We did some experiments in the appendix as well with alternative architectures, like this fusion and decoder architecture that lets you scale kind of non-quadratically with the input length. So we tried to increase those numbers, okay, what happens if you have 8 exemplars, 16? We didn't see considerable improvements. There was some, so maybe that could be interesting to explore too.
I think we had another one in the back, yeah.
Very cool talk. Just wondering, in terms of the pre-training where you input the pairs of sequences together, how much did you see that improved performance on your tests?
Yeah, so this ablation is in the paper. The improvement, let me just actually bring this slide back up. So the overall improvement of doing sequence pairs versus single sequences was relatively significant. The additional advantage of predicting this similarity score was relatively smaller. It was significant, but the effect size was smaller. So I think like maybe you're kind of already teaching the model to do a rough similarity prediction, even just by virtue of if it's going to do residue prediction well, it's incentivized to, because the sequences have higher than random similarity, already do some form of comparison between the sequences.
Yeah, we have time for one more over there.
Hello, thank you for the great talk. So I was interested what you think about, so the 2 sort of like positive examples and 2 negative examples seem super low. For this case, especially be like kind of generalizing along the like in context sort of like examples. So I wonder if like you did any experiments that we get at this idea of like maybe those extra sort of like tokens are just like giving essentially more effective parameters for the model to like learn a little bit better and sort of make inference, similar to how we think about like reasoning models, that their sort of thinking tokens aren't really doing a whole lot, like the content of those thinking tokens really isn't doing a whole lot. It's really just kind of giving the model more sort of like parameters to kind of search over. So do you have any comments on that? Did you guys do any like experiments that kind of get at that?
Yeah, that's a great point. I like these papers that kind of in the thinking model context, do these reasoning tokens, are they helpful? I mean, I think they generally do conclude that they are helpful above and beyond just adding capacity to the model in the thinking reasoning model case. In this case, we did not run that ablation specifically. I would expect, just given the effect size of conditioning, of ablating the exemplars entirely, that there's something going on here beyond just adding additional compute. We have other, maybe to support this a little bit empirically, we do have a comparison between, we evaluated two different size transformers. I believe like a base and a small, so roughly like a couple 100 million parameters versus maybe 50 million parameters. And the effect size there was much smaller than the effect size of this exemplar ablation. So I think that, that's a great point. It'd be interesting to look at, but I think there's probably something going on here more than just the added compute capacity from the larger context. Thanks.
Great. So we have to move on, but let's thank the speaker again. Great. So our next speaker is Sam Slajeski from New York's own Flatiron Institute.
Cool, thank you so much. Yeah, I'm Sam. I'm a postdoc at the Flatiron, and today I'm talking about learning protein dynamics at proteome scale. There we go. So it's actually nice to follow the previous talk because I don't necessarily have to convince you that proteins are really important and understanding their function is really important, right? They're these workhorses in the cell, and thanks to the sequencing revolution, we now have hundreds of millions, if not billions, of new protein sequences. And our task is, how do we figure out what these proteins do? If I give you an arbitrary sequence, does this have immune functions? Does it have catalytic functions? Is it involved in some sort of scaffolding or structure, or does it have signaling properties? And I think AlphaFold has rightfully been revolutionary in this prediction of sequence to structure, but I think it's important to remember that structure is a means, not an end. We care about understanding a protein structure because there's this tight link between the shape of a protein and the function it performs. So by being able to accurately predict structure and the shape, we can get to these accurate predictions of the function of a protein. However, oftentimes, a single structure is not enough. You can take the sequence and you can pass it through AlphaFold and you can get kind of this weird shape, but you might not be able to understand exactly what this shape does or how it works until I start telling you that this domain here kind of moves laterally back and forth. You have a domain on the bottom left here that kind of moves rotationally. You have this blue domain that moves kind of in and out, and then you have some corkscrew motion in the top right. And now you can start to build a clear picture of the function of this protein and actually how it works. So this is not a unique example. Nearly all proteins have some sort of dynamic function, be it allosteric regulation, where you have some kind of off-target binding that induces conformational change. If you have transmembrane receptors that need to have helical reorganizations to transmit a signal across a membrane, you have proteins with these kind of long, floppy, disordered tails that are important for recognition and become structured upon binding. And you have cryptic pockets, which are maybe not immediately noticeable, but then become open upon binding and are dynamically accessible. So I think if you take one thing away from this talk, I hope it's that dynamic sites are functional sites, and that by understanding what parts of a protein are dynamic, how they move, and how they move in concert with one another, kind of these functional or dynamic domains, we can start to piece together the functions of proteins. Just some brief background. For a long time, the primary approach for modeling protein dynamics were molecular dynamics simulations, where you kind of integrate the equations of motions over several time steps and put together a trajectory of protein structures. In recent years, people have kind of hijacked AlphaFold to try to get it to generate multiple structures by somehow modifying the input MSA, either doing alanine scanning, subsampling, or clustering the MSA and getting multiple sequences out. And then recently, there have been a lot of methods that take kind of single and pair representations or some sort of sequence representation and train a denoising diffusion or flow-based or some other score-based model to directly sample these structures. And all of these methods are really powerful, but they have a fundamental limitation for what I want to do, which is the speed of their throughput. So I'm showing here just some back of the envelope estimates. I didn't actually benchmark all atom molecular dynamics simulation for AlphaFold database. But you can see that even in kind of the most optimistic
estimates for some of these sampling-based methods I'm showing here, BioEmu, doing a whole human proteome would take on the order of 100 GPU days. If you want to scale to kind of larger data sets, you get into GPU years. And this is orders of magnitude faster than actually running all atom MD. So I'm interested really in high throughput prediction of dynamic properties that allow us to investigate things like differential splicing of isoforms, to affect the impact of missense variants or somatic mutations and how they change the dynamics and function of proteins. And actually to go beyond the human proteome, I have collaborators that work on coral biology. So we have whole new, take a proteome, multiply it by however many new organisms, in addition to thinking about designed or engineered proteins and understanding how their sequence affects their dynamics. So we built a model called Rocketship for fast structural heterogeneity profiles. And our key insight here is that you don't actually need to sample full structures to have kind of information or informative things about the dynamics of a protein that can be used to help understand the function. So we predict a one-dimensional descriptor of function. Remember I said earlier that dynamic sites are functional sites. So we can predict this RMSF, the root mean square fluctuation, which is a scalar measure of how flexible is this residue. We also predict two-dimensional, basically correlations in motions. And this, I think, is really valuable for trying to understand allosteric networks, which have to rely on kind of intramolecular communication. If you have motion at one part of the protein that affects motion at another part of the protein, you would expect them to have correlated motion. So we predict these two-dimensional descriptors. And then we predict what I call a sort of 2.5-dimensional descriptor, where we take a simulation trajectory and we slice it. We take each of those sequences and tokenize it using foldseek, this method for kind of for any given residue, classifying it as the type of shape that it is. And then you can compress that into a distribution. And what we get for each residue is a distribution over the types of three-dimensional states that it has been seen in over the course of this simulation. So by focusing on these really kind of dense information-rich descriptors rather than sampling full structures, we are able now to scale to the proteome and beyond, making predictions on the whole human proteome in less than 20 minutes on a single GPU, and in theory, scaling to all of AlphaFold database in less than a year on a single GPU. You can, of course, parallelize this. And I think what's really interesting is that by specifically kind of hyper-focusing on these dense information-rich descriptors, we can actually do better at predicting things like local flexibility or correlated flexibility compared to just kind of sampling structures from an ensemble. This, I won't go too much in depth, but this is enabled by rich pre-trained features. So we use a ESM3, a kind of multimodal language model that is pre-trained on sequences and structures. So this allows us to get embeddings of the protein that are very representative of similar sequences and similar structures. And then we train them on these large-scale MD data sets. I'll have the detail of the simulations up here for those that are interested, but I think the two key things I hope that you take away from this slide are we tried to be really principled about how we did our splits to make sure that the model was generalizing to new and diverse folds. So we actually used foldseek to cluster the structures and use different foldseek clusters to determine our splits. And the second is that while these databases are some of the largest available right now, and they cover diversity in terms of different types of proteins, these simulations are quite short. And so this is a limitation that you'll definitely see, where these simulations are very unlikely to access kind of larger domain motions. But a second either key insight or key assumption, depending on how much you believe it, is that the local fluctuations that can be observed in these shorter simulations are still informative of things like allosteric networks, where understanding kind of pairwise correlations in motion help you chain together these intramolecular communications. So here's just kind of our top line performance. We compare with BioEmu, which is a sampling-based method, and Dyna, which is another protein language model-based method. Those of you that are familiar with Dyna will note that it's not actually designed for this task. So I think it's important to be fair, but we were basically just trying to evaluate the tools that we currently have available. How might you use them to predict these types of things like local fluctuations and correlated fluctuations? So we were able to achieve the best performance in terms of error for predicting RMSF, as well as the two-dimensional Gaussian correlation coefficients. The IMSD is actually a measure of network similarity. As you'll see, we treat these 2D correlations as allosteric networks, as well as these kind of distributions of structural states, the 2.5-dimensional profiles. But what I really want to highlight is that we're able to do this with a fraction of the runtime and a fraction of the parameters. We even go so far as to train a sequence-only model that's really small, fewer than 2 million parameters. And while it is worse, it's really, really fast and is able to achieve somewhat competitive performance. So we wanted to go beyond just looking at simulated data and see how well this would correlate with some experimental data. I will not pretend to be an expert in NMR or NMR data, but within the Dyna paper, they're looking at these kind of longer timescale motions in the microsecond to millisecond timescale. And one thing that I think is really exciting from this paper is they curate this database of NMR measurements. And because they're looking at the longer timescales, they focus on these R2 to R1 ratios and the Rx measurements, and largely leave these Het NOE measurements, which kind of correlate with shorter timescales, relatively untouched. What we wanted to see is, can we take this nicely curated data and see whether our predictions of RMSF correlate with these experimental Het NOE values? And we find that they do. There's kind of a sign flip here where lower Het NOE correlates to more flexibility. So the correlation here is negative, but they are quite correlated. And using this threshold of 0.65, we're able to do good binary classification. And I'll note that this is basically zero-shot prediction. We're not fine-tuning on any of these experimental measurements. We just take a sign flip, and I'm showing here, this is one of the proteins that in the original Dyna paper, they highlight their prediction of the longer timescale motions. And we show here that we are able to fairly well recapitulate these shorter timescale motions with our rocket ship predictions. The next thing we wanted to look at is the MDCath data set, which actually includes simulations at 5 different temperature points. Unfortunately, there are not really methods out there right now that can predict the relationship between temperature and flexibility. So we didn't have a ton of baselines to compare to. We look at there's a paper from De Ribeiro and Lima showing that B factors tend to display this kind of exponential dependence on temperature. So what we can do is take our RMSF at the lowest temperature and scale it with this exponential scaling relation in this paper. And we can find that the predictions from rocket ship are actually, as the temperature gets kind of further away from this base temperature of 320, we're able to maintain fairly accurate predictions of the relationship between temperature and flexibility. Finally, we wanted to look at a case study, and we chose to focus on KRAS. KRAS is a very frequently mutated oncogene, and it's implicated in diverse cancer types. As a result, it's become a popular therapeutic target, but it's really challenging to target because of its highly dynamic and allosteric behavior. It kind of acts as a molecular switch where it only relays signals downstream after being bound by GTP. So what we wanted to look at is if we can model the allosteric network of KRAS using rocket ship. We predicted, as I mentioned earlier, these 2D correlations in motion and actually treated this directly as a network. We were then able to perform clustering on this network and identify substructures within the protein with correlated motion. What I'm showing you here is just kind of comparisons between these metrics derived from a simulation as well as those predicted, and you can see that they correlate quite nicely. But what I really want to focus on is the substructures that we were able to pull out unsupervised from these predictions of motion, where we're able to identify this C-terminal tail that not only has higher flexibility, but if you look at the structure profiles in the bottom, kind of displays a discrete series of structure tokens. There's this pair of in kind of greenish teal here, a pair of anti-parallel helices that move in concert. And what I thought was the most interesting is this blue cluster here, which we've labeled as pocket. It's maybe a little bit hard to see on the structure, but in the bottom, there's this flexible loop. And sure enough, you can go to the PDB and find a structure of KRAS with the ligand bound exactly in this pocket. And we were able to kind of pull out this set of residues as a unique cluster because of the way that their motion was correlated. Oh, yeah, can just. Finally, we wanted to look at, there's this really nice data set from Wang et al. Published in Nature last year of a deep mutational scan of KRAS, where they systematically mutate every residue and look at the change in folding free energy, as well as the change in binding free energy with a bunch of partners. Here, we're looking just at the change in folding free energy.
And our hypothesis was that residues which are central in these allosteric networks are really important in kind of information propagation within the structure. And so high centrality within these predicted allosteric networks would correlate to residues that result in a large change in folding free energy when mutated. And this is what we find both looking at the wild type KRAS and identifying the residues that are central in this network, as well as when we perform our own in silico deep mutational scan, and we look at the variance in predicted centrality as it correlates to the variance in folding free energy. So we think this is kind of an exciting general technique for understanding residues that are important for allosteric communication within a protein. Finally, I just want to close on some thoughts about the outlook of predicting dynamics for proteins in general. I've kind of set up this competition, right? You have to compare to these baselines and other methods on speed and accuracy, but I really think that they are compatible and can work together as part of this funnel or this pipeline where you have this massive sequence space that you need to search to try to understand important properties or understand variant effect. So you can take these much more rapid protein language model-based methods and identify proteins with flexible domains or mutations that have large impact on flexibility. You can then go and use these sampling-based methods to kind of sample diversely from the ensemble and potentially use these as seeds to run kind of more in-depth or longer molecular dynamics simulations. In addition, I mentioned earlier kind of the length of these simulations being a limitation, and now a lot of people feel like every month another kind of molecular dynamics data set comes out with longer simulations. But I think it's important that we can do this in a tailored way. So there's been people talk a lot about lab in the loop frameworks, and I think it would be kind of equally smart to do a sort of simulation in the loop framework where you take these models and you kind of quantify their uncertainty and you say what types of proteins, what parts of the proteins are we least confident in, and use that to do targeted new simulations. So we're not just burning GPU hours simulating tens of milliseconds of proteins that we can already do very well on. You can then take this new data and fold it back in to improve the quality of those models. So with that, I want to thank Sonia, my co-author on this paper and one of the co-group leaders, along with Pilar of the Structured Molecular Biophysics group. I'm funded by the Simons Foundation. Thanks to everybody else that kind of gave feedback on this work. Thank you to the organizers for the opportunity to speak. The code as well as pre-trained models are on GitHub, and we have a preprint on bioRxiv. I'm happy to take questions. Thanks.
Yeah, great work. I have two questions. One, what's going on with the inverse scaling for the structure time series prediction? It looked like the larger model and the one with structure information was doing worse.
You're talking about
It's the mini
here.
Yes.
Yeah, so the large model is doing well for the RMSF and the GCC. The KL divergence, I'm not entirely sure on this. This is something we're still looking at. I think you may have noticed in the talk, we focused primarily on these first two derivatives. I think that there is a lot of the tokenization is a really powerful strategy for kind of compressing information. But we're still not entirely sure because we don't have, because we're treating every residue independently and we don't have kind of correlations between pairwise structures. You could imagine that sampling from these distributions would not actually give you physiologically meaningful structures. So
yeah, I was sort of, I guess, mistaken about the way you're doing the sampling then, but I thought it would be quite nice if you could do, from structure token to decoding to get a coarse grain.
Yeah, I think that's where we're thinking about going. I think that's a great idea. The limitation right now is, yeah, the kind of independence of each position. It might actually just work okay for some cases, but you have this risk of getting kind of non-physical structures.
And then the second question I had was, have you tried applying this to, pretty standard like sequence structure information benchmarks like ProteinGym or clinical variant effect prediction?
Clinical variant is kind of next up on my list. We're really interested in doing something that can take advantage of the genome scale. I have results that I didn't show here. We do a very
kind of preliminary, we take UniProt labels and we show that the regions of proteins that are labeled as disordered have higher RMSF predictions across the entire human proteome. But I'd say that's still in progress.
Wait. Thanks. Thanks for the talk. AlphaFold 3 and a lot of these other structured methods, they have outputs that report their uncertainty in the localization of atoms and the pairwise, predicted pairwise pairs. And I was wondering if those were surrogates for flexibility. Have you looked at those? Is that true?
I haven't played around with those myself. As we were kind of doing literature review, I came across a lot of papers that looked at this sort of thing. And as far as I can tell, the jury is still out. There are papers that report both that say kind of PLDBT correlates super strongly with flexibility and you can use it. Some that say you should just kind of treat it as a binary thing, right? I think AlphaFold DB has these thresholds of like 0.75 or something is very good. And if it's less than that, it's probably an IDR, some sort of intrinsically disordered region. And then there are some papers that say you can't trust these. So I think that probably the true answer is somewhere in the middle where a lot of the times when there will be high uncertainty, it's because the training data, the PDB has that same protein in several different conformations. But it's also likely that you could get high uncertainty because you just haven't seen this in training at all. And it is actually well structured, but it's just out of distribution.
Great. So let's thank the speaker again and move on for the last opportunity.
Okay. Sorry, remind me.
It's Michael Brusidiakina. Should be.
It's the one that says "Look, Mom."
Look, Mom. Yes.
Yeah, there we go.
That's better than final.
Yeah, it's a good. Yeah.
Thank you.
Should be good.
Great. So welcome to Michael Brusidiakina.
Pretty close, Brusidiakina.
UNC.
Great. Yeah, so thank you all. Thank you for staying for this last presentation. I'll try to keep this somewhat lighthearted since I know you guys are very tired. So yeah, I'm Michael from UNC. Let's see. Space. Oh, perfect. Yeah, so I think to kind of frame everything, we're at a really cool place now in terms of kind of drug discovery. So many brilliant computational biologists, such as yourselves, are kind of understanding biology in a new way, and this is resulting in a lot of potential really cool drug targets to treat diseases with, say, small molecules. And then at the same time, we can target a lot of these targets much more effectively using tactics like PROTACs or kind of beyond rule of 5 molecules. However, pharma companies still, by and large, tend to focus on kind of extremely well-validated targets. And why is this? I posit that a lot of the issue here is that finding small molecule binders is really hard. So, the kind of traditional way of doing it, if you have a new target, is to use high throughput screening, which can be slow and expensive. And then even the hits it gives you tend to be poor binders that require a lot of optimization. And then this kind of creates a chicken and egg problem where it's hard to justify an expense like this for kind of new targets. But at the same time, the best way to validate a new target is to find a binder that modulates the target and kind of see what ends up happening. Does it actually have an effect on the disease? So this obviously leads to the question, can we computationally identify binders? And there's a lot of interest now in kind of using some sort of pure AI method to solve our problems, find new binders. The problem is there's kind of well-known generalization issues with a lot of these pure kind of ML methods. And there's not going to really generalize to a lot of the kind of cool new targets that we're interested in, like transcription factors or even like RNA. What about kind of physics-based approaches? So kind of there's a traditional scoring functions like AutoDocVina or Glide make kind of very fast predictions based off of like some very simple energy function of like a protein ligand complex. These make so many assumptions that they're not particularly accurate. On the other hand, we actually do have ways of reasonably accurately gauging protein ligand binding affinities, but they're with free energy simulations. But these are kind of way too slow for anything kind of large scale. So it's 1 GPU per day per compound for if you're trying to get an absolute binding free energy prediction. Oh, okay. Well, basically, I kind of want to, yeah, I guess dive deep into what's going on in these free energy calculations. Like, why are they so slow and why are they so accurate, I guess? As you can see, binding free energy is like this complicated, like 10K dimensional integral. I'm not going to go into this too much. But you can kind of estimate this by like a bunch of using like a ton of MD simulations by kind of alchemically removing the interactions between the receptor and the ligand and like computing the work done over the course of like these many MD simulations. And if you do it, you can oftentimes get a pretty good answer. So this leads to the question, can we combine these two approaches? Since machine learning and physics are kind of have these kind of two very different problems of like generalization versus this kind of speed accuracy trade-off. So at a high level, something I've been interested in is like, can we use physics simulations to create a protein-specific data set for an ML model? So like a naive idea would be to run these free energy simulations of the target and complex with kind of random putative ligands. This would take way too long, and there wouldn't be like nearly enough data for this to kind of work. Can we do this more efficiently? And I posit to you that the answer is yes. So there's this cool technique called force matching, where you can basically take a data set of forces, frames and forces from molecular dynamics simulations, which you can obtain relatively cheaply, and use kind of a neural network to kind of remove degrees of freedom. So you're predicting the forces on some subset of the atoms without the knowledge of where the positions of the other atoms are in the system. So you're kind of trying to implicitly figure out where these atoms are. And you can kind of show that if you do this, you're kind of training a neural network to give you the kind of conditional free energy of the system, conditional on the current I guess atoms. This has been kind of looked at for this is pretty frequently used for kind of coarse graining force fields, so like integrating over kind of all the atoms except for like the alpha carbons in a protein. However, I'm interested in this question of like, what if we removed all the atoms but the ligand atoms, which is a pretty large increase, a large decrease in the system size, about 1000x. So this would make things a lot faster. So here's kind of the overview of the method. Take a protein. We've been using kind of about 30 to 60K kind of random small molecules sampled from zinc, kind of put randomly, semi-randomly in the binding site. I can go, the details are a bit tricky. I can go into them if anyone wants to ask. But essentially random positions of the binding site. You also want to make sure that your ligands are, you also have samples outside the binding site, so you get the solvation free energy as well. We've been using kind of 4 nanoseconds of MD simulation, which is pretty short, but kind of a lot of these interactions, well, yeah, it's I think better than I think you can still get some good stuff from here. And we're kind of freezing the ligand in the binding site relative to the kind of rigid body degrees of freedom of the protein binding site. You can get the forces. This takes costs about $800 if you use like some really cheap GPU services. From there, we basically just train a neural network. And I can go into the architectural details later if people are interested. It's not particularly spicy, but the idea is we just use force matching to kind of train a neural network. But at inference time, we're interested in kind of the energy values that it gives us. And what we've been doing is just doing a very simple calculation for virtual screening for the sake of speed of like taking, once you have a docked pose, like estimate kind of binding free energy as like this kind of force matching potential of the ligand in the binding site minus the ligand outside the binding site. That ignores the kind of ligand, kind of the free energy of ligand reorganization. You can, in principle, do that much faster too, but I haven't looked at that yet. How do we know if this is working? So what we did is we choose targets with kind of many known binders in Kemble, with and without crystal poses. Generated the data set, making sure to exclude anything too close to kind of anything with like a known, any known binders, essentially. Train the models. We can also, sorry, but basically we additionally generate kind of property match decoys as like kind of negative data because like we don't generally have like data on non-binders for these proteins. And then we use kind of two different docking methods, unidock, which is kind of just a really fast version of kind of autodock VINA, which is a kind of traditional physics-based, traditional docking method, and then diffdock, which is a kind of fancy AI method for getting the 3D pose of the ligand of the binding site. And the hypothesis here is that diffdock was presumably trained on all these targets. It's known not to generalize very well, but the hypothesis here is it should be reasonably accurate on these targets specifically. Put a pin in that. Then we kind of, let's just compare it to the baselines. So the kind of three metrics we care about here are like kind of enrichment factor. So like the maximum enrichment factor you can get on this benchmark, which is basically like a kind of question of like, how much better are you at random at selecting these kind of binders in an actual virtual screening context? So that's something that I'm most interested in. Also AUC. And you can also ask like, can it re-rank docked poses correctly? So each of these docking methods, unidock and diffdock, will give you a set of possible poses of the small molecule in the binding site. And the question is, if you take the best energy from all these methods and rank them, does it pop out the actual, like the true pose, if the true pose is in there? Interesting. We used 6 targets. So here are the kind of results. So for crystal poses, where you're comparing, like for the set of true poses, or for the actives, you have true poses. For the decoys, the kind of presumed non-binders, you have just docked poses. If you use unidock, VINA does actually quite poorly. Nina, this kind of neural network approach does better, and we're getting kind of much better results. Though it's worth noting that these are the sample sizes here are low enough that these error bars are pretty big. So take, I guess, everything with a grain of salt. But we're better along all the metrics. Intriguingly, if you do this for the diffdock, but this is just using the diffdock for the decoys, all the methods do way better, which is kind of interesting. I'll put a pin in that. That's not what you would expect. However, if you look at kind of the docked poses for the, so instead of for the active molecules for each target, you're looking at the, you don't have the true pose, you just have the kind of docked poses. We don't really see any improvement in performance for the LFM methods. Same thing with the diff dock. Everyone improves with the diff dock docking about equally. So these kind of results are kind of interesting and raise some questions. So first of all, why is DiffDock really improving the performance of all these models, even when you're using crystal poses for the actives? Hypothesis. Oh, okay, I'll get that. And then there's also the question of why do we have such a steep performance drop when we go from known crystal poses to actives to docked poses? So my hypothesis for the first question. Oh, and there's kind of two related questions, which is why does Vina do worse when we use true poses? And then why does Nina have such like a high AUC relative to its enrichment? There's kind of two hypotheses here. One is that DiffDock is giving great poses for actives, but horrible poses for decoys, which makes it really easy. And I think this is due to kind of the nature of DiffDock is that it's kind of doing really well on data it's trained on, but it wasn't trained on random molecules in a protein binding site. For the second questions, there's kind of, my hypothesis is basically that these models that we're training are much more pose sensitive. So I've looked, yeah, it seems that Vina and Nina are actually surprisingly good at giving good scores to actives, even if they have an incorrect pose, but for different reasons. And I can kind of hypothesize about why later if anyone's interested. But because of the kind of physics-based nature of how this was trained, if you have a wrong pose, it's going to give you a bad score. And this is also related to the fact that it pops out the score. It re-ranks dock poses much better. So you could hypothesize about using diff-dock poses for the actives and unidock poses for the decoys as maybe a more realistic scenario where you have a better docking method. And indeed, we see that the LFM methods appear to be doing better. When you do this, though we still see Nina has a quite high AUC, I think because it's still ranking incorrect poses relatively highly. So kind of moving forward, we have some promising results for this method, but it's kind of seems to be pretty limited by kind of the docked poses that you give it. If your docking method, if the poses aren't very good, this performance, you won't see as great a performance boost. However, the cool thing is this, I think, is quite solvable. So this LFM energy function can also be used in a docking scenario where you're explicitly minimizing it, just like you would do with, say, a Vina or Glide force field. I haven't done that yet because we need to make it faster, essentially, but I think it's totally doable, and I'm excited that it's something I'm working on now. And then I think just kind of philosophically, I think this is a general method that isn't kind of limited to experimental data. So I see these as kind of initial results. There's so many things to tune about this method, and I'm hoping we can get much better results by, changing model architectures, changing simulation parameters and whatnot. So I think there's lots of room for improvement here. And then finally, I want to start using this on novel targets. So there's a couple collaborations I'm in now with experimentalists. If any of you have targets you'd like me to try this on, please reach out. I don't want to go into details, but we actually have found some binders for some pretty weird targets with this method, which is I'm excited about. And I'd love to acknowledge my lab as well as David Coase and the Coase lab. They've been providing me with a lot of advice. So thank you. And I guess any questions?
Yeah, we have time for a question or two.
Nice work. The thing I was missing is when you're training, you're training for a single protein, right?
Yes, yes. So
yeah. So many ligands, and then you train. And I was also missing because you're predicting both. It's like a field, or it's actually like directly the forces. Yeah, I missed what the gradient as well.
Yeah, so you're predicting I guess an energy, but when training you're using like autograd to like you're regressing on the forces essentially.
Okay. So but you don't have to there's not some like double backpropagation. There's not some like it's like regular backpropagation.
Yeah, I guess I guess there is some double backpropagation. So you're getting the forces. So yeah, it's you're predicting the energies, I guess, backpropagating to get the forces, and then backpropagating through those forces to train the model. Yeah. Yeah, it's a
yeah. Is there any chance of generalizing across?
So
it doesn't make sense this stuff, actually, because you're trying to,
I think potentially it's maybe if you limit it, you could imagine aligning all the kinases and potentially trying to generalize across the kinome. Yeah.
Put in some embedding in the protein as well.
Yeah.
Okay. Any other questions? Okay, well, great. Well, let's thank the speaker one more time. And before I hand this back over to David, because it's the end, it's the last session, I just wanted to make sure that we give a hearty thanks to the lead organizers, David Knowles and Peter who have done an amazing job.
We've done an amazing job being patient with AV stuff, I also want to do thanks, I guess they're not in the room, but especially to Sarah Curtis and Kristen Weatherly, who were amazing with doing a ton with helping set up, and then again to all the volunteers from my lab and Peter's lab who have been running around helping clean up things. On that front, those of you who have posters, if you could please, when you're done with the poster session, it's not difficult to achieve, I know, to like knock over the poster stands, but if you can knock them over in such a way that they then fold up and end up next to the InstaDeep station, and the little stand things as well. They're like, if you've ever set up a camp, like a tent, they work like that. You like pull it apart. That would be fantastic. And similarly, yeah, I think we if people can aim to be out around 7, it's because their staff wants to be able to leave. So thank you so much all for coming. Thank you to all the speakers and presenters. It's been great. Yeah. Thank you again to sponsors for food.